pathophysiology involved, but how are their functions regulated? This part is not very well understood. So we performed a discovery experiment about 6 or 7 years back, where we did bulk RNA-seq analysis of flow-sorted CD4 T cells that we isolated from the wild-type mice, either from the control kidneys or post-IR kidneys. And as you know, these kind of discovery studies throw at you so many different genes, but one of the genes that caught our attention was called TIGIT. And as you can see, its expression was extremely high on CD4 T cells that were isolated from post-IR kidneys compared to the control kidneys. But we didn't know what TIGIT actually means, so we studied a little bit and we came to know TIGIT is T-cell immunoreceptor with Ig and ITIM domains, and it's considered like a new generation immune checkpoint molecule. And it's mainly expressed on activated T cells, NK cells, and Treg cells, and it interacts with its ligand called CD155, which is presented by APCs and potentially other immune cells also, and renal epithelial cells. So this interaction between TIGIT and CD155 has been shown to attenuate T helper 1 and T helper 17 mediated pro-inflammatory responses, and at the same time enhance Treg mediated responses. And in addition to TIGIT, T cells also express other partners. It's a much more complex pathway than I've shown here. And I'm going to talk a little bit about some of our recent data about some of those things and how ischemic injury affect those molecules. But much of this information on the understanding about TIGIT pathway has been derived from cancer-related and multiple autoimmune-related diseases, but its role in acute kidney injury was not known. There are multiple mechanisms that have been proposed through which TIGIT is supposed to function. It could be both T-cell intrinsic mechanisms or T-cell extrinsic mechanisms. In T-cell intrinsic fashion, TIGIT has been shown to directly inhibit TCR-mediated signaling and dimerization of CD226, whereas in T-cell extrinsic mechanism, TIGIT-CD155 interaction has been found to enhance anti-inflammatory cytokines like IL-10 production by APCs. Another important reason to identify and study novel immune checkpoint molecules like TIGIT is the recent realization that immune checkpoint inhibitor therapy that targets traditional molecules like PD-1 and CTLA-4 often results in acute kidney injury. And this shows that T cells play an important role in human AKI, but also emphasizes that we need to identify novel immune checkpoint molecules in order to minimize renal related adverse events. So with this background and preliminary data from bulk RNA study, we hypothesized that immune checkpoint molecule TIGIT plays an important role during AKI pathophysiology. So we wanted to first validate our bulk RNA-seq data. For that, we performed the same kind of experiment that we did for bulk RNA-seq study, where we had the wild-type mice subjected to ischemic injury, and then we performed flow cytometry on the kidney mononuclear cells that were isolated with Percol-based methods. And as you can see, there was significant TIGIT expression on CD4, CD8, and double negative T cells, shown here in the red bars, that were isolated from post-IR kidneys compared to the kidneys from either sham operated or control mice. So this validated our bulk RNA-seq data and also showed that during ischemic injury, TIGIT expression increases at both mRNA and protein level. We have also tried to assess TIGIT expression in human kidney T cells and understand how ischemic injury affects TIGIT expression in human kidney T cells. And we have an active collaboration at Johns Hopkins University, fortunately, that gives us these valuable renal cell carcinoma nephrectomy samples. And for this particular study, we got the sample either before the renal artery clamping or after the renal artery clamping. The renal artery clamping, post-renal artery clamping samples underwent different times of ischemia. And what we found from this study is that TIGIT is expressed on human kidney T cells, and that ischemic injury increases TIGIT expression on human kidney T cells. We have also tried to explore the single-cell RNA-seq data from Kidney Precision Medicine project, which is freely available to explore. And when we try to look at TIGIT expression comparing healthy controls versus AKI patients, we found there was significant TIGIT mRNA expression in AKI compared to healthy controls, and it was mainly restricted to T cell and NK T cell compartment, which I am showing here with the red arrow. We then wanted to understand how TIGIT expression affects T cell functions. So for that, we directly compared TIGIT negative versus TIGIT positive subsets of CD4, CD8, and double negative T cells. I'm only going to focus here on CD4 cell in the interest of time, but we have similar findings on CD8 and double negative T cells. And as you can see, TIGIT positive cells, which are shown here on the right-hand side of these plots, had higher interferon gamma, TNF-alpha, and L17 compared to the TIGIT negative subsets, both at baseline and after ischemia reperfusion injury. What was interesting that we found was that TIGIT negative subsets were actually making less pro-inflammatory cytokines after ischemic injury, whereas the expression of these inflammatory cytokines was not affected on TIGIT positive subsets. So we also found that these TIGIT positive T cells in the kidney have an effector and central memory phenotype, both at baseline and after ischemia reperfusion injury. Since Treg cells have been shown to express large amounts of TIGIT, we were interested to see what is the situation in kidney during IR. We first looked at baseline levels, and we found 60 to 70% Treg cells in the kidney express TIGIT, but during ischemia reperfusion injury, this population declines rapidly. We next wanted to understand how TIGIT is involved in AKI pathophysiology, and for these studies, we were very fortunate to get TIGIT knockout mice that were kindly gifted to us by Dr. Vijay Kuchu from Harvard University. And once we had these mice, we again performed the bilateral ischemia reperfusion injury and followed the mice up to 72 hours. And then we assessed kidney injury by serum creatinine or histology. And as you can see, TIGIT knockout mice, shown here in the red bars, had reduced serum creatinine compared to the wild-type mice. Also, when we looked at their histology, they had fewer necrotic tubules compared to wild-type mice in their outer medullary region, suggesting that TIGIT knockout mice are protected both functionally as well as structurally from ischemic injury. We have also tried to understand the role of TIGIT in cisplatin AKI model. Cisplatin, as you know, is an important anti-cancer drug with significant kidney effects and is used as a model for AKI in mouse. So for this study, we used single intraperitoneal injection of cisplatin, and then we followed the mice up to 72 hours. And like the IR model, we checked kidney injury by serum creatinine or histology. And similar to our IR model, we found in this model also the TIGIT knockout mice were both functionally and structurally protected, suggesting that TIGIT likely plays a pathogenic role in AKI. As I said in the beginning, there are other partners of TIGIT that T cells express, and in addition to CD155, other ligands are also expressed by antigen-presenting cells. But we don't know how ischemic injury is affecting all these things and how TIGIT is interacting with some of these molecules. In addition to TIGIT, T cells also express PD-1 and other immune checkpoint molecules. So we did some recent studies that show that the co-stimulatory counterpart of TIGIT, which is called CD226, its expression goes down on kidney T cells and NK T cells, but we don't see any change in the co-inhibitory counterpart of TIGIT, which is CD96. In terms of the ligand, when we looked at the whole kidney using Western blotting, we found there was significant CD155 expression, and to some extent, CD112 also increased. We didn't see any statistical significance on CD113 expression. So we next wanted to know what type of cells are making CD155 and 112. So we performed flow cytometry, and we found that this is mainly increased on CD11B positive cells, though other cell types also express these ligands. The expression of these ligands is not affected by IR injury. And next, we wanted to understand how TIGIT is interacting with other immune checkpoint molecules like PD-1, CTLA-4, 2B4, and some newer ones like LAG-3 and TIM-3. We have not been able to find much difference in more of these, but what we have seen is that in addition to TIGIT, only PD-1 expression changes, and it increases after ischemic injury. This was not a new finding, but when we looked at TIGIT positive and TIGIT negative subsets, we found that PD-1 expression was very high on TIGIT positive CD4 and CD8 cells, both at baseline and after ischemic injury. So we wanted to then understand how TIGIT is regulating T cell functions during AKI pathophysiology. So we performed an unbiased single-cell RNA analysis of CD45 positive immune cells that were flow sorted either from the control kidneys or post-AKI kidneys after 24 hours from wild-type and TIGIT knockout mice. And initial analysis of our single-cell data was able to identify all major immune cells in the single-cell data. And when we looked at TIGIT expression, it was mainly restricted to T-cell and NK cell compartment. When we focused further on the T-cell cluster, we found that TIGIT is mainly expressed by T helper 17 cells. Then we performed additional analysis and differential expression analysis, which showed us multiple genes that were either upregulated or downregulated. But to make some sense out of this differentially expressed gene signature, we performed gene set enrichment analysis. And amongst other things, what we were able to see was there were some metabolic differences between T cells that were isolated from T helper TIGIT knockout mice compared to the wild-type mice, both at baseline and after ischemic injury, as shown here in the bottom panels in the red color. So there were genes that were enriched that were related to multiple metabolic pathways in the TIGIT knockout mice. Recently, we have also used a flow-based approach where we can investigate key metabolism-related proteins to assess either glycolysis, oxidative phosphorylation, or fatty acid oxidation. We have used the same, thank you. We used this flow-based approach, and we found that GLUT1 expression was significantly increased on T cells that were isolated from TIGIT knockout kidneys. Recently, we have started using seahorse-based approaches to directly measure oxygen consumption rate and extracellular acidification rate, which are the endpoints of oxidative phosphorylation and glycolysis. And as you can see there, the OCR and ECAR are quite distinct between TIGIT knockout and wild-type T cells, suggesting that TIGIT likely plays a role in T-cell reprogramming in these mice. We are currently performing additional experiments, and we found that there is increased glucose uptake in TIGIT knockout T cells and increased ATP production from glycolysis in these cells. I'm not able to show that data right now. So to conclude my presentation, I think we have shown that there are T cells that play either pathogenic role or protective roles depending on the T cell type that you're looking at and the time after injury that you're looking at. And I think some of our recent data clearly shows that TIGIT expression increases on kidney T cells after ischemic injury. And these TIGIT positive cells have a pro-inflammatory and effector and central memory phenotype. And knockout mice are protected both in the ischemia reperfusion and after cisplatin injury models. And it seems like TIGIT is likely playing a role in T-cell metabolic reprogramming as a mechanism, but we are still exploring this pathway. And it likely interacts with its main ligand CD155 and potentially also with PD1. Again, we don't have any conclusive data on this right now. We are still doing some experiments. But either targeting TIGIT directly or some of the ligands or inhibiting the interaction between these signaling pathways could be a potential strategy for AKI therapy. So I would like to acknowledge a lot of different people, especially my mentor, Dr. Hamid Rao, and our postdocs in the lab, especially Radhika Kapoor. She did some of the recent work related to TIGIT story. And Shishir Patel, Tara and Reo for helping in all our experiments, and prior lab members that helped me really start this project. And all my collaborators at Johns Hopkins University, and Dr. Vijay Kuchru for providing the TIGIT knockout mice for making our life easy to understand TIGIT role in AKI pathophysiology. We have recently started collaborating with Dr. Shikhar Mehrotra also at MUSC related to a different project, and I would like to thank Dr. Ankit Saxena also for his continued support with understanding T-cell and flow-based technologies. With that, I would like to thank you all for your listening, and I can take any questions.
Thank you, Shanti, for this very well designed and very well presented talk. And we will invite one or two questions from young people, the young students, because they should be the one to ask.
Sorry, I can't really hear you.
Hello.
Sites are there. In your cartoon,
Yeah.
the cell signaling pathway, inside the cell, there is a tyrosine kinase, the docking sites are there for
Right.
phosphorylation, dephosphorylation. Have you, because either by docking or undocking, you require phosphorylation or dephosphorylation. Did you study that pathway? That one? I have a doubt actually in that. Maybe the phosphorylation is the one of the key, and maybe controlling the injury, etc. This way I have a small doubt, that's all.
Yeah, I think what you're trying to understand is more about how TIGIT signaling works, and I think what you're saying is that through the ITIM domains, it interacts with the other intercellular signaling. We haven't done that part. We are more interested in how this pathway can be targeted for AKI therapies.
This can be continued.
I think you get
Any
expansion,
participant?
everything, am I? So you get expansion of double negative cells, and what are the antigen that is attracting huge number of double negative cells in the kidney?
It's a very interesting question. I think because these double negative T cells or even T cells are involved very early on and they expand very early on.
Theoretically it won't be there in circulation.
Yeah, we think in that early time point, the antigen is not involved because it will take long time for them to get activated through an antigen-dependent manner.
Okay, but apparently,
But in later time points, yeah, that antigen-dependent
Then they should
mechanism
have-
also.
they should have TCR that time.
Yeah, they have TCR.
Okay, thank
But
you.
in the early phase, most likely antigen is not involved.
Hi
Okay.
there.
A young girl is waiting there. I should take
Hi there. There is one question here. That's a great talk indeed. I just wondering because TIGIT is believed to be exhaustion molecule. It should be associated with the TIM3 and other CTLA4. But one thing just mentioned that TIGIT positivity is associated with pro-inflammatory cytokine. How this can be justified?
This is a very good question and it has really haunted us a lot. There are a couple of things. We don't know the final answer right now. If you remember, I showed some data that we didn't see any other immune checkpoint molecules getting affected. We've only found TIGIT or PD-1. If you look at the tumor infiltrating lymphocytes, they usually express all of those and they have a exhausted phenotype. So that's one thing that's different. And there is some literature that shows, I removed those slides because I thought that I will have less time, that shows that actually in the TCR gamma delta population, TIGIT positive cells have more cancer killing properties.
But as you mentioned,
Can you continue it later, please?
Sure.
She is standing for.
Hello sir, I'm Pooja Kumari from IIT Kharagpur. Sir, your talk was very nice. Sir, I have one question that since nowadays the co-blockade therapies are, it's like very interesting now and people are researching on it. So as you are showing here that PD-1 expression is also there in this scenario. So whether for the future TIGIT PD-1 co-blockade could help here in treating these kidney injuries?
Yeah, so that's the next step we are doing. Actually, those are some of the experiments that are ongoing in our lab right now. Yeah, but in this model, we are facing a lot of issues with the antigen, antibody-based experiments because it induces inflammation and immune response much more strongly, probably because there is already some immune activation.
With this, we conclude the session, and it has been.
Thank you.
Okay, and here. Okay, there is another talk. What is? No, it is the conclude. And we enjoyed, both of us enjoyed the session. Lovely talks, lovely interactions, and thank you organizers to have given this opportunity to us to chair the session, and very kind introduction of us by our young girl for that matter.
Thank you very much, chairpersons, for graciously leading this session with such clarity, depth, and guidance. We extend our heartfelt gratitude for your valuable contributions to this session. Now I would like to request our respected chairpersons to kindly present our esteemed speakers with mementos as a gesture of our heartfelt appreciation for their insightful and inspiring talks today. I request Professor Sanjeev Noel to please come up on the stage. I would also request our speakers and chairpersons to kindly remain on the stage for a group photograph after the memento presentation. I also request Professor Peter D. Katsikis to please come up on the stage and receive your memento. Thank you very much, sir and ma'am. Now I would like to invite Now I would like to invite Professor Amit Avasthi to kindly come forward and present tokens of appreciation to our respected chairpersons. We are now moving forward to our next technical talk segment. It is my pleasure to invite Dr. Hemant Kumar Palani, who is Assistant Professor of Hematology at Christian Medical College, Vellore, to deliver the technical address titling India's First Point of Care POC Trial for CAR T-cell therapy as a representative of MNNIT Biotech India.
pointer? Timer soon. टाइमर नहीं हो पाएगा, टाइमर नहीं हो पाएगा.
So good morning everyone. So today I'm going to talk about the point of care manufactured CAR T cell therapy clinical trial, the results of a phase 1 trial which we done at CMC Vellore. So a little bit about the introduction of CAR T cell therapy to the students over there. CAR T cell therapy is an established and accepted standard of care for relapsed refractory B cell malignancies, especially B cell leukemia and lymphoma. The advantages of this CAR T cell therapy is higher response rate and no need of HLA typing unlike hematopoietic stem cell transplantation, and it has a potential to target as many different cancer antigen as possible. So for example, in CD19 CAR, it has two important domains. One is the extracellular antigen recognition domain and the intracellular T cell activation domain. So it has the advantage of both monoclonal antibody and cytotoxic T lymphocytes. So one gives rise to the high affinity and specificity, and the other gives the cytotoxicity and proliferation. Yeah. So this is just a live cell imaging of CAR T cells stained with green fluorescence and against the tumor cells in red, and the cytox blue stains the dead cells. So this is just a table comparing the list of 7 US FDA approved CAR T cell clinical products available worldwide in comparison with 2 Indian products, Quatimi and Nexcar 19. As you can see, the majority has 4-1BB co-stimulatory domain. So what is the main challenge of this CAR T cell therapy? So I would say the major challenge in the CAR T cell therapy is the cost. So the access to this CAR T cell therapy is very expensive, and it has significant logistic challenges in centralized manufacturing and remain difficult to access even in developed countries like US. As you can see, there is a issues with the reimbursements, insurance and additional expenses. So what we believe is this decentralized or point of care manufacturing using an automated system in academic or tertiary care centers is an alternative to centralized industry driven manufacturing. As you can see in this cartoon from American Society of Transplantation and Cellular Therapy, in the long term, the point of care strategy is the only way forward to lower the national and global inequities for CAR T cell access. If you can see in detail, the cartoon represents the difference between the current centralized manufacturing and the potential future point of care manufacturing, where the sample is collected and transported from hospital to manufacturing industry, where the manufacturing process and quality control takes place, and again shift back to the hospital for the patient infusion. Whereas in the point of care strategy, where all the process can be done in the point of care center itself. So the vein to vein time, once the sample taken from the vein from the patient back to the vein, the CAR T cells, this we call as vein 3 to 5 weeks in centralized manufacturing, whereas just 1 to 2 weeks in point of care manufacturing. And in this point of care manufacturing, there is an option of infusion of fresh product, whereas in centralized manufacturing, there is only a frozen product. And there is no need of bridging therapy in point of care manufacturing, whereas centralized manufacture, since there is a long waiting time, there is a need of bridging therapy, where the quality control is done only by the company, whereas in point of care, we can done at the hospital setting itself. And importantly, all the research, innovation and optimization is not possible in centralized manufacturing. It can be done in the point of care. And importantly, the cost. It's just 1/10 of the cost compared to the centralized manufacturing. So at CMC Vellore, we do almost 300 transplants per year. So we wanted to establish a point of care manufacturing for CAR T cell therapy. So which manufacturing platform should we use? So there is a final yellow one is a prodigy. There is a increase in cell numbers in bag as shown in the orange one. Interestingly, there is a significant difference in the phenotype of the product, where the CAR T cells, the yellow one, significantly give rise to higher central memory and naive phenotype of these CAR T cells compared to others, like 46% compared to 13 and 16%. So this naive and stem cell memory is important for the greater persistence of these CAR T cells in vivo. In addition to this flow cytometric data, the gene expression profiling also suggested the CAR T cell generated from this prodigy developed product shown more naive and less cytotoxic than others. So we have chosen this prodigy based CAR T cell manufacturing, and we have started our phase 1 clinical trial evaluating the safety and efficacy for relapsed B-cell leukemia and lymphoma. So we have used a second generation lentiviral vector from MNNIT Biotech, since it was already shown superior efficacy in plenty of clinical trials done worldwide. So this was just a workflow of CAR T cell manufacturing using a Clinimax Prodigy system, where CD4, CD8 T cells are selected, followed by activation using T cell transact. Transact is nothing but a CD3, CD28 nanobeads. After activation on day 1, the cells have been transduced with lentiviral vector. From day 1 to day 9, it's just an expansion process. On day 9, the fresh product will be infused back to the patients, and we have a set of release criteria assays before giving the product to the patients. So the table represents the standard release criteria assays or different quality checks. These are laid down based on the US FDA standards. So I'll just highlight few, starting with the viability. It should be more than 70%. The transduction efficiency, that's a percentage of transduced cells, should be more than 15%, and obviously more than 90% of CD3 positive T cells, and it should not be having any contamination, bacterial or viral contamination or endotoxin negative, and copy number PCR should be less than 5, and VSVG, replication competent lentivirus, should be negative. So there is a plenty of regulatory approvals to be obtained before starting a clinical trial for CAR T, starting with IRB, IBSC, RCGM, Review Committee of Genetic Manipulation for importing the vectors, GTAC from ICMR for safety, and CDSCO for clinical trial approval. So this is just a study design, cartoon showing the study design. So on day -9, after apheresis, we start the CAR T cell manufacturing in the lab. On day -6, we have the first QC, which gives the go or no-go signal to enroll the patient into the clinic and start the patient for flow turbine and cyclophosphamide conditioning for CAR T cell infusion. On day -2, we have a release criteria assays, whatever I have shown in the table previously. We will do all these assays. Once the product completes and finish the QC assays only, then we'll reach the product to the ward for the infusion. So it's all fresh out product. The vein to vein time is only 9 days, and there is an option of cryopreservation, but we have given only a fresh product to all the 10 patients, and all the cases were being done only within the hospital. So this cartoon shows we have enrolled the patient, 6 ALL patients and 4 lymphoma patients, and this is a 3+3 dose escalation trial, starting with 0.5 million per kg with a cohort 2 of 1 million per kg and third cohort of 2 million per kg. This is a table summarizes the quality control data of all the 10 products. I'm not going into the detail, just to highlight few. The median age of the patients is 45 and the viability is 97%. The percentage of transduced cells is almost 38%. And the copy number PCR, it's all within the range of release criteria or acceptance criteria. So coming to the characteristics, so as I shown previously, the more than 90% of the cells are CD3 T cells, In contrast with BALL, there is a lymphoma cohort showed there is an increase in CD4 CD8 ratio of around 2.3 when compared to 0.6 in BALL. So in consistent with our previous NIH data showing the increase in central memory cells, even we could see there is an increase in central memory population. The gray bar represents the central memory and the red bar represents the effector memory T cells. This is a characteristics of individual products, and this is a mean of all the products. As you can, for example, if you see the product 07 compared to the starting T cells, the final product showing the increase in central memory as shown in the figure. So we also done a functional analysis of CAR T cells by exposing this manufactured CAR T cells to target and non-target cells, basically CD19 positive cells and K562 as a control. And as you can see, there is a significant increase in cytotoxicity in target cells compared to non-target cells. In addition, we also done an antigen restimulation assay where the CAR T cells are stimulated with target cells with increasing ratio at the different times for over a week, showed increase in proliferation of CAR T cells. This represents the antigen specific functions and proliferations of manufactured CAR T cells. So we also done a profiling of cytokines. As you can see, I'm not going to all the graphs, just to focus on one. IL-2, there is, as it is exposed to the target cells red bar, there is a significant increase. When it exposed to non-target cell control, the level is significantly down. This shows the specificity of a manufactured CAR T cells. And this just a bar increase in line graph showing the percentage of CAR positive cells in vivo. This is a percentage of cells post infusion into the patients. As you can see, there is a median increase in around 2 to 4 weeks post infusion of the patients. Coming to the outcome, this is a swimmer's plots represents the outcome of all the 10 patients. Each bar represents a patient. The red bar indicates the complete remission. The green represents a progressive disease, and the yellow one is a partial remission. So out of 10 patients, 8 patients had a complete remissions for about 1.5 years of median follow-up. And BLL patient, there is a bone marrow minimal residual disease by flow cytometry is negative for almost a year. Coming to the toxicities, only 2 out of 10 patients had grade 2 CRS, and no neurotoxicity has been observed in any of the patient, and hematological toxicity more than grade 2 has been seen in only one. So importantly, coming to the cost, so we also done a detailed costing analysis. The Sankey diagram illustrates the healthcare resource utilization of patients undergoing the CAR T cell therapy as a inpatient and outpatient care. So the mean expenditure of this 10 patients was calculated to analyze the cost drivers for the patients treated, where as you can see, the major cost drivers were hospital care, lab investigations and medications, which comes to around $12,000. And we also done a bottom-up micro costing analysis, showed the production of CAR T cells of about $35,000, which give rise a total cost of $47,000 US dollars. To summarize, the manufacturing CAR T cells with Prodigy, which give median viability of 97% and the fold expansion of 15-fold, and the transduction efficiency of 38%, and T cell differentiation subset shows an increase in central memory T cells, which gives more persistence. And toxicity wise, there is only one grade 3 CRS has been observed, and this phase 1 data established a safety and efficacy with excellent clinical outcomes. So coming to the status of the study, we have completed our phase 1 clinical trial. We are awaiting regulatory approval to start phase 2. So all the data whatever I have shown has been published recently in a molecular therapy oncology, as you can refer to the in detailed data. So in conclusion, the point of care manufacturing is a potential way forward for reducing the cost and to improve access by scaling up more centers. This same platform can be used for multiple cell and gene therapy products in combination with local manufacturing of vectors and CGT reagents. It can further reduce the cost. In India, we need a combination of centralized and decentralized manufacturing systems like Prodigy. So not only by the words, this is just an aerial view of our CMC campus, as each arrow highlights the location of the laboratory, starting with the workflow of sample from apheresis to GMP, followed by QC, and followed by a ward for the infusion. And this is one of the CT scan of patients before infusion and 28 days after infusion. So finally, I'll to acknowledge Dr. Vikram Mathis, who is a clinician PA of the study, and manufacturing team, QC team, and clinical team. And I also would like to thank ICMR for the funding and MNNIT Biotech for organizing and sponsoring this talk, as well as the sponsors for our phase 2 clinical trial. And this is our GMP facility at CMC Ranipet campus. Thank you. Yes. Yes. No, that patient didn't respond, so we have removed.
The way I would like to see this problem, because you are injecting an antibody which has a different variable part, right? So theoretically, one would expect that expansion of anti-idiotypic antibodies, and that might interfere with this kind of therapy. So those negative data, the one did not responded, but at the same time it has shown high IgG titer. I would expect that you might get some anti-idiotypic antibody which is probably interfering with the T-CAR functional activities. So this is kind of my curiosity. Probably that can create some problem in CAR T-cell therapy.
Yes.
You have any comments on that kind of
No,
issue?
we didn't look for the IgG levels for that patient. So what I would have shown it might be a different patient. So we have removed that patients from the cohort.
So that is not the one
Shown
IgG
an
titer.
increase. No, that's
The
not.
color was same, so I was
Okay.
wondering this is
Okay.
same patient.
No.
Okay, thank you.
So, nice talk.
Thanks.
So do you think if a patient is transfused with frozen product or non-frozen product, there will be difference in the outcome?
So studies have shown there is a difference. So fresh product is always better for greater proliferation and persistence. So there is a data out there. There is a publication comparing this.
Okay. Okay, thank you. So another one is your antibody sequence is humanized or it's a mouse one?
It's a mouse, murine SCFE.
It's mouse one?
Yes.
And what is the relapse rate of this construct?
So out of this 10 patients, there is no relapse till now for past 2 years.
Okay, thank you.
Yeah. Thank you.
Thank you very much, sir. As we now move forward to the next plenary talk, it gives me great pleasure to invite our distinguished chairperson to kindly come forward and take their seats on the dais. We are honored to welcome Professor D.N. Rao, who is former professor and head of biochemistry department at AIIMS New Delhi. A former scientist at ICMR, Professor Rao's research spans molecular immunology, infectious disease, and reproductive immunology, focusing on immune regulation and host pathogen interactions. He is widely recognized for his remarkable contributions to immunology, molecular biology, and vaccine development. Accompanying him, we have Dr. Alpana Sharma, who is former professor at Department of Biochemistry, AIIMS New Delhi. Dr. Sharma is an accomplished academician and researcher with vast experience in biochemical and molecular studies. Her research focuses on tumor immunology, autoimmune skin disorder, and the tumor microenvironment in cancers. With our esteemed chairpersons now on the dais, I would like to request Professor Rao and Dr. Sharma to kindly take charge of the session and invite the speakers for their respective talks.
I will introduce Vikas, is my friend actually. Short to them. I request Professor Joshi Jacob, my great friend actually. I think he's from MRI School of Medicine, Atlanta, USA. And every time, every IAS, the seminars, he always used to come and used to give a new insight in his research. I think this time he's giving a dermatology, I think, looks like. I invite Dr. Joshi Jacob to initiate the talk. Dr. Joshi, restrict to the time, okay?
Good morning. It is such a joy to be here with you today. And let me talk to you about the work that is currently ongoing in my lab. And I'm going to talk to you today about this skin disease called hidradenitis suppurativa. So it's a debilitating, extremely painful skin condition, as the pictures show you. And if you ask me where do these lesions appear in the body, that's shown in this slide. So if in both males and females, you find in regions that are shown in the pink, and if it's in females only, it's shown in the dark. So it's usually in the breast area, in the armpits, and in the groin, and on the buttocks, okay? And so if you look at the incidence of it, it affects women more than men. It affects people with color more than people with less color in Caucasian population. And it's an extremely painful condition. And the patient that have it says it feels like hot knives that are stabbing under my skin, or the pressure is so unbearable, it's like a balloon that's about to pop. It's incredibly painful, okay? And the most distressing symptom is that there is this thick, foul-smelling pus that comes out of these lesions. And since the lesions happen in parts of your body that's important for your social interactions, what happens is your life kind of falls apart, and there's increased opioid addictions, and also 3 times more rates of suicide in patients that have this. And the cause is unknown, and currently there is no cure. There are drugs that you can give for symptomatic relief, but there is no absolute cure. So what else do we know about this? It is onset with puberty, number 1. The incidence is about 0.4 to 1.7%. And this is based upon not by going out and taking like 10,000 people and saying who has it and who doesn't, but by looking at the insurance claims that have this code as the diagnosis. So the point is it could be significantly higher. The incidence could be significantly higher than what is known. It's often misdiagnosed, and I'll tell you why that is. And the diagnosis is made by physical examination, and normally it takes 7 to 10 years to get a correct diagnosis. So how do we go about figuring out what's wrong with these patients? So they have to go to a doctor, and there are 3 things that you need. 1, you need to have the typical lesions that are shown in on that end. Second, it has to be in typical locations like I showed you before. And finally, there has to be chronicity and recurrence. They have to see this twice in a row that's 6 months apart. When these 3 things come to pass, then it's diagnosed as a hidradenitis. So if you are in a major city like Allahabad, you're okay, but if you're in the village, then your GP does not know what is wrong with you, and it takes a while to figure it out. So if you look at the progression of the disease, the first step that happens is, this is the skin. The first step is a hair follicle gets occluded. And as a consequence, what happens is you have pro-inflammatory cytokines and chemokines that are released, and that causes a lot of inflammatory cells to migrate into the skin. And immune cell migration happens, and you have inflammatory nodules and abscesses that form. And now the skin gets fibrosed, and then you can see the follicles, they all become one. They become tunnels that have incredible amounts of pus in them. And then what happens is that the inflammation continues, the lesions get worse, and then when you look outside, you find these lesions that have this pus that seeps out of them. So if you look at what is the cure for these, there are 3 monoclonal antibodies that are used. The first one is it's against TNF-alpha, and the other 2 against IL-17A. So essentially, what you're trying to do is you're controlling the inflammation. It's symptomatic treatment. It's not a cure. And when you give this medication, what is considered a cure, or they say, okay, he's been fixed. It's not a cure. If you reduce the number of lesions by 50%, that's you're counted as a success. So if I had 10 lesions and it goes down to 5, it's considered like, okay, that drug works. And so technically, there is no cure at all, okay? So I'm just going to skip these 2 slides. And so the question that we have is, what causes it and what drives it, okay? And so our central hypothesis is that HS is a virus-induced autoimmune disorder. And we think that antibodies made against the virus cross-reacts with self-antigens, and it drives the hidradenitis. And we'll get into it. So the first experiment that we did was to do what's called a Vir scan. Essentially, I don't even have a pointer. No, it's okay, I got it.
Pointer.
Okay, so no, I have. So if you look, we take healthy cohorts of patients, HS or healthy controls, and take their serum, and now you do what's called a Vir scan. From 1 drop of blood, you have this phage library that expresses all known viral proteins that infect humans. The proteins from all viruses are encoded in it. You add the antibodies to it, and then you fish out the phages that are positive, then you sequence it, and then you compare the 2, and you figure out, is there any virus that associates with this particular disease? So when we do this experiment, what we find is that the biggest jump that we saw was Epstein-Barr virus. If you look at the p-value, it's insanely high. And so what we find is that people with HS have high levels of antibodies against Epstein-Barr virus. EBV, as you know, is a ubiquitous virus. It causes infectious mononucleosis. 95% of you and me are infected with this virus. We have antibodies against it. And it infects B cells, as you know, and it persists latently in it. And then you have bouts of reactivation in them. So the question is, if 95% of you and me are positive for EBV, why is it that a small percentage of people get hidradenitis suppurativa? And now, when we look at the data more closely, we look at epitopes that are bound by antibodies of people with HS and normal controls. We find that people with HS see epitopes in EBV that you and I do not see, okay? And that's shown in this slide. And this slide shows you, these are the common epitopes. If you look at everybody, you find that all of our antibodies can bind to parts of the EBV genome. But if you look at the HS patients, now you find there are specific epitopes that are seen only by those with HS and not by the normal controls, okay? So essentially, there are 2 things that are happening. We all have antibodies, but we don't get HS because we do not respond to forbidden epitopes in the EBV genome. And we think that those forbidden epitopes have cross-reactivity to human antigens, and that's what aggravates hidradenitis suppurativa. Okay? All right. And so the next question is, are there plasma cells and B cells in the HS lesion? And the answer is yes. So the treatment, when it gets fibrous, is to surgically remove the skin and put a graft back. That's the only solution. So once it gets to that level, there's nothing else you can do. So we get these samples from the surgeon. This is 1/3 of the sample that I got. So we chop it up into pieces, and then you can do a single-cell RNA-seq with the samples that we get. And when we look at it, what you find is that you find that majority are keratinocytes. That's not surprise. But you have T cells, B cells, and plasma cells, and also other cells. So we were intrigued by them having B cells and plasma cells, and particularly plasma cells because they are the differentiated antibody-producing B cell counterparts. And we wondered if they had a role in the disease at all. So then what we did is we looked carefully at the B cell and plasma cell populations, and you have the B cell population, you have 1, 2, 3, 4, and 5 distinct plasma cell populations that we see based upon their digital signatures. And I'm going to summarize the data, and when you look at it, it turns out that these skin B cells that we find in the skin, these are actually memory B cells, number one. And then you have these antibody secreting cells 1 through 5, and you can actually put them like in a differentiation pathway. And when we look at this, they are actually class switched, and they're somatically mutated memory B cells, and they're differentiated plasma cells. That's what you find in these lesions, number one. Okay? The next thing we wanted to know is, are there many, many clones of these plasma cells and B cells, or are they oligoclonal or polyclonal? And you can actually look at this if you look at the molecular fingerprint of antibodies, by looking at CDR3, you can tell if who's the parent and who's the daughter and so forth. So we can figure that out. And that's exactly what we did. And when we do that, and also when we look at, if you take the whole isolated cells and say, how many VH genes are there? You find that there's only like one major one at all. And the rest of them are just not represented. So it's almost like almost a monoclonal population that you see in them. And when we look at the somatic mutation pattern, what you find is that even when they come there, they're already mutated. And once they are there, they mutate just a little bit, but it's not that they come there as naive and then they start mutating in the skin. We see them, they're mutated, and they continue to rest there. Okay? And so the next question we want to know, are these memory B cells and these plasma cells related to each other? Did the memory B cells give rise to these plasma cells? And when we look at that analysis, obviously you know that it has to be related because there's only like one clone. And so when we look at it, so this is a complicated slide. This is their memory B cells, and all the other things that we see in there, they're all related to each other, which means that it is the memory B cell that gives rise to each of those individual plasma cell populations. So to summarize, what I've shown you is that skin memory B cells in HS lesions differentiate into all these different plasma cells, and they're all clonally related. They are the child of the same memory B cell that is undergoing differentiation. Okay? And then we did an experiment where we had a patient, a female patient. She had 2 HS lesions on each of the 2 breasts. So then what we did is we took one and the other one, and we analyzed them. We wanted to know, are the B cell clones the same at 2 distant locations, or are they different? Okay? And surprisingly, what we find is that, so what this complicated slide tells you is that if you analyze the left breast or the right breast, both of them, they have the exact same antibody in them, heavy and light chain. So even though they are 2 separate anatomical sites, the clones that we find are identical, which means that they came from somewhere else, they moved into the HS lesion, and they start dividing and differentiating into plasma cells, and that's why you find the same clones in 2 lesions. Okay. The next experiment so I showed this before. So when we compare people with HS and without HS, we find that the people with HS respond to EBV much more, and that to selective epitopes. In the next experiment, we wanted to know, do these lesions have EBV, active EBV in them? And the next data I'll show you, but we will not talk about it, the answer is yes. So if we look at the lesions from these patients, 57% of the patients have EBV we can detect it by RT-PCR. So there is EBV in the active, there is antigen in the active lesion, number one. And now we wanted to know, what are these antibodies specific for? So I told you the lesions have memory B cells and plasma cells, and they're monoclonal, they're almost, very limited clones. If we recreate the antibody, what are they specific for? And I told you before that when we look at the whole serum from these patients, they target forbidden clones within the EBV genome. So we tested this, and we take these monoclonal antibodies, and then we run them against EBNA1, which is one of the latent genes in the Epstein-Barr virus. We find that the HS patients bind to it, and the controls don't. And then when you look carefully at the EBNA1 antigen, this shown here is the full EBNA1 antigen from here till the end. And if you delete this glycine alanine repeats and make it short, then the antibodies, they bind to this region and not to this truncated version, which tells you that there is a part of the genome that it's targeting. And it turns out that in multiple sclerosis, people have shown that there are antibodies that target EBV, and it's in the same area, this glycine alanine repeat region, that the HS antibodies also target. Okay? And I'm going to skip that one. So we did a VIR scan. We also wanted to know if you take serum from HS patients and normal controls and run an autoantibody panel, do the HS people see autoantigens? Do they recognize autoantigens? And when we look at that, so it's called a Hue scan, just like we did before, we find that the highest bind is COL-A1, which is a pre-collagen molecule. So you have the precursor of type 1 collagen, there are two kinds. So COL1A1 is one of them. And we find that the antibodies from this HS patient bind to it. So we wanted to know, in this monoclonal antibodies that we recreated from the HS lesions, do they bind COL1A1? And to my surprise, these are all 24 monoclonal antibodies, and when we look at reactivity, we find that they bind COL1A1 beautifully. It's interesting because they bind to the precursor COL1A1, but they don't bind to the type 1 antibody at all. So we have two things that we show. The same antibody molecule, the same monoclonal antibody that we recreate from the HS lesion can bind to two antigens at the same time. They can bind to an EBV antigen and to a human self-antigen. So it is almost like in these patients, when you have this cellular destruction, you have EBV, you make this monoclonal antibodies, now you have two antigens there. You have the EBV antigen and the self-antigen from the cellular damage, and now you have an abundance of antigen, and the B cells that proliferate, and they cannot stop. They're like that Energizer bunny in the commercial. They keep going again and again and again, and you cannot stop it. So that's why if you detect it early, you could stop it, but if you come in very late, then it's going to be extremely difficult to stop. And with that, I come to the last slide. So 95% of the adults have been exposed to the EBV. Why don't we all get HS? We don't get HS because only some people respond to forbidden epitopes in the EBV, and that cross-reacts with human self-antigen. And that's why those people get it, and the rest of us don't. Okay? And so our data does not show cause and effect. I'm not saying that EBV causes HS. I'm not saying that at all. I'm just saying that in the HS lesion, when you look at it, we see pathogenic antibodies that can recognize EBV as well as self-antigen. Okay? And so what is the working model? So the working model, we don't know how it starts, what's the initiating event, but we know that a hair follicle gets occluded, EBV reactivates in the skin, EBV-specific autoreactive memory B cells will migrate from rest of the body into those sites. It doesn't matter whether it's one breast or the other or both. You have the common precursors that come in there and start differentiating into plasma cells, make antibodies that can react to both EBV and self-antigen at those two sites. And the cell destruction leads to release of self-antigen and drives autoreactivity. Okay? And so what we are doing now is we are trying to see possible therapies include antiviral drugs to curb EBV reactivation, antibody therapy or CAR T cells to deplete B cells and plasma cells, and also to make decoy EBV receptors to block HS-induced pathogenic antibodies. So the planned studies are followed. Number one, measure the antibody affinity of HS antibodies to EBV and to self-antigens. So I'm in the process of doing that. Two, do B cells and plasma cell deletions, they change over time. So now we have patient cohorts where they come in and we take punch biopsies now, then they come back after 6 months, we take it again, same site, and a year later, and we want to know, do they change over time, or do they always remain the same, right? And we want to know which came first, the EBV reactivity or the self-reactivity. So we have experiments that are doing that, and I will not get into the details of that. And we want to map the forbidden epitopes recognized by these HS antibodies and self-antigens by cryo-EM. Where do they bind? What else do they look like? Can we have a predictive value by understanding these? And HS diagnosis take 7 to 10 years. So we want to know, can we make these forbidden antigens and develop an ELISA kit by which I can take all of your sera, all of your EBV positive, and tell you with some precision how many of you are going to progress to HS or not. And if you can diagnose it early, then you can initiate treatment early, and the outcomes are much, much better. I told you that there are these FDA-approved drugs that target inflammation. It works 50% of the time, and it works even better if you target the disease much earlier. And finally, can these forbidden epitopes serve as potential therapeutics? So that's where we are. And I want to thank the people in my lab that did the work. So the work was initiated by my MD PhD student, Gordon Dale, and also Carla, and they have both left the lab. And I'm grateful to my dermatology collaborator, Dr. Ornstein and her team, Mehul Suthar and her team, Bo Liang. Bo Liang and I, we do the cryo-EM studies. And also to Sudhir and also Paul Johnson from the Emory University. So a year ago, 2 of my students graduated. And then I had to decide, do I take another student and then have another 5 years? The problem is, I may not look old, but I'm actually quite old. If my life was an IPL game, I'm in the last over with 3 balls remaining, okay? So I did not want to take another PhD student because that's another 5 years. So I recruited this other guy. He works really hard. He comes in every day. We get along fine. And he does very good experiments with his hands. And his error bars are narrow, and he's actually, he's very well trained. And it's a joy to work with him. And so what is I told you I'm at the end of my career, so what's the plan? So plan A, we have submitted our revised manuscript for review. Merck wants to fund this, so I have a grant that I submitted to them that's under review. I'm going to submit a grant in February 2026. I wish to develop a blood test for HS. And I want to develop forbidden epitope mimics as therapeutics. And then get into cryo-EM affinity studies and acid. I do that by myself now. So this is plan A. If it works, that's great. So if it doesn't work, then you need to have a plan B, right? So what's plan B? If plan A does not work, I think I'll just go to Kerala and make Malayalam movies. That's what I'm going to do. So with that, I'm going to stop, and I thank you for your time.
Thank you, Dr. Joshi. Such a lucid talk, and you have unfolded the uncommon dermatological disorder.
Yes.
So really very good and excellent talk. And as the organizers told, we are running 1 hour late, so we can take just one question, just one.
Yes. Make Malayalam movies.
We'll watch. This plan A will work.
Okay, thank you. Thank you, Chairperson, for allowing me to ask this question. Basically, it's not a question. I'm just wondering, you have explained it so nicely, the connection between EB virus and the disease. And you have shown there is a molecular mimicry and there is a cross-reactive antibodies there. The amazing part is why it is only monoclonal antibody generated against that particular antigen epitope which you have shown. You very cleverly, you did not explain why it is happening in only few people when we all, 95% of us, are exposed to EB virus. And you did not even specify what are the speculative, chances of whether it is genetically predisposition, genetic predisposition.
So let me answer the first question. So the question is, why is it that only some people have to make these antibodies to this forbidden epitope? So most of us can clear EBV by hitting. So if you are shooting a deer, right? If you shoot it on the leg or on the arm, you can make it go down. But sometimes you have to hit it at the heart. You have to keep shooting till you hit a critical part. So in some people, the antibody response against all the other things that you and I can use to clear the virus is not good enough. So you have to keep like epitope spreading and hit like the heart of it, which are forbidden, and that's the reason why you get it.
And the second one is that why it has to go and locate at specific locations and not other places.
So the HS is known to be a disease in the folds of the skin. So I don't know what starts it off first. I only know that once it starts, it migrates there.
Joshi, thank you very much for a good talk. Most autoimmune diseases have more than one autoantibody. It's true of type 1 diabetes, lupus, or RA and all that. You showed us 5 clones of plasma cells.
Yeah.
Are they all making similar antibodies, same antibody?
No, they are encoded by different antibodies, but their target is the same. That's why I did the other patient serum also. When you take the whole patient serum, they also target, the top hit is the Col1A one, the pre-collagen one that we see from the entire, not just monoclonal, but the entire plasma that we get from.
Yeah, I think it will be important to know what markers they carry. Are any of these long-lived varieties of plasma cells?
No,
plasma cells have a short life,
but
some of them are long-lived.
So when we look at the patient serum, we find the antibodies that are very durable and they persist for a very long time.
And the other one, of course, the chairpersons will not allow me to speak. We'll talk about some. Are there any biomarkers?
First question, we'll be the ones to answer that. Absolutely, I agree.
Thank you, Dr.
Thank you, Chairpersons. Your presence has truly enriched this proceeding, ensuring the session flowed smoothly and meaningfully. We are grateful for your time, your expertise, and the thoughtful way you hosted the discussion. Now I would like to request our respected chairpersons to kindly present our esteemed speaker, Professor Joshi Jacob, with a memento as a gesture of our heartfelt appreciation. And before you all depart, please stay with us on the stage as we will proceed with the group photograph. Now I would like to invite Dr. Ambak Rai to kindly come forward and present token of appreciation to our respected chairpersons. Thank you, sir and ma'am. As we now move ahead, we now come to a very special and proud moment, the presentation of IIS Senior Scientist Award. This session will be conducted over the next 25 plus 5 minutes. While we truly recognize that a lifetime of contributions can't be captured within any time frame, but we humbly request your kind cooperation in adhering to the allotted time duration so the conference flows smooth. May I have the honor of inviting Professor Amit Awasthi, who is president of the Indian Immunology Society and the professor at THSTI Faridabad. To join him on the dais, we invite Dr. Kaustuv Nayak, who is the secretary of Indian Immunology Society and senior scientist at ICGEB New Delhi. We request both of you to kindly come forward to lead the felicitation ceremony and present the IIS Senior Scientist Award to our distinguished awardee.
Thank you.
I request everyone to please stay in the hall as parallel sessions will start after the tea break.
Thank you very much. This is a privilege to invite this Seniors Oration Award, which is one of the important award for IIS. So you know IIS has 3 different categories of award. One is the Senior Oration Award, Mid-Career Award, and the Young Scientist Award. So we receive application, and I just like to briefly tell audience, the people who understand this and the newcomers should know this. So Senior Oration, we invite applications through the website and through the circulation to all the members, and interested participant or candidate apply this, and this thing comes to the IIS EC, and EC looks at the application, and this application is forwarded to 3 independent reviewers, and these reviewers are discreet. They don't know each other, so this is how done in a very confidential manner. And then this reviewer gives the independent report to the president, and then I put together the score, and then it's based on the average, we decide who is the winner of this award. So this year, Professor Pradeep Sen, who is the chief scientist at CSIR Institute of Microbial Technology, IMTech, and he has done his PhD from IICB, has done postdoc in US, and he's in the CSIR IMTech for quite some time, and he has done some original immunology work. And I would just like to mention few, because immunology at the core at the Indian Immunology Society. So he has done some original work in the host pathogen interaction and published some original papers. And one of the interesting thing what he has also identified, the TIM3 you know is the checkpoint molecule which were identified and discovered in Harvard Medical School by Professor Vijay Kuchru. And he has done a lot of work on TIM3 in the autoimmune disease and the cancer. And Novartis has taken this anti-TIM3 antibody as a combination therapy with PD-1, PD-L1, CTLA-4. So this is licensed to Novartis. Now, Professor Sen has shown that for the very first time that, TIM3 is doing a lot of job through the dendritic cells, which was not really known originally from Professor Vijay Kuchru's paper, because he was focusing primarily on the T cell. So from his paper, subsequently the other story come out from the Harvard Medical School, which got published in Nature, where the same work what he has shown using the Leishmania pathogen through the dendritic cell, on the similar line, the study got published that dendritic cells, TIM3 is very important. If you delete TIM3 from the dendritic cells using Kree-Lock system, you get a totally different outcome. This is the one story what I would like to congratulate you for your original work in the immunology. This is very original. The second thing is that you also developed the biosimilar, which is also a requirement, because we are talking about industry and academia. So now we are moving toward the biosimilar space where you are developing new antibodies, and this is a TNF-alpha. And we know TNF-alpha is really a culprit for many of the autoimmune disease. And such discoveries coming from his lab, which I think is really remarkable. So I will not really waste too much time. I request you to really kindly start your talk. Thank you. And in this session, as customary, there is no question, because this is a Senior Scientist Oration Award. If you like to have any question, you can talk to Professor Sen at the tea or lunch.
it will be seen properly. It's not coming. It's not coming. That's the issue. Can you just hide this one? Hide the presentation. No, hide the presentation. क्यों? No, no, hide the presentation. If mode one. No, 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 no. This one. Go here. Hide the presentation. Yeah. Yeah. Good morning, everyone. And first, I would like to thank Professor Avasti for your kind introduction. I would also like to thank Indian Immunology Society for considering me for this particular award. And IMMUNOCON always remains as a great platform for me to speak about my research. Now, coming to my research, for many years, my laboratory is engaged in working on different aspects of the visceral leishmaniasis. And very recently, we have started identifying the molecular events leading to immune suppression and thereby establishing the disease visceral leishmaniasis. Before going to our findings on visceral leishmaniasis and immune suppression, I'd like to give the brief introduction about the disease, visceral leishmaniasis. Visceral leishmaniasis is a global health problem. It is second deadly parasitic disease in the world after malaria. The disease is prevalent in 89 countries across the world and mostly in the Latin American countries, Africa, and Southeast Asia. This particular disease is prevalent in India as well because 54 districts in India is endemic for visceral leishmaniasis, which is mostly centered in West Bengal, Bihar, Uttar Pradesh. But currently, the disease is getting spreaded from the previously known endemic region, and now you can see a lot of visceral leishmaniasis cases are coming up in Jammu Kashmir, Himachal, Uttarakhand, and Kerala, Gujarat, Madhya Pradesh. In fact, a very recent case of visceral leishmaniasis appeared in Thiruvananthapuram in Kerala, and in 2022, in Bihar, it is almost 20 deaths have been reported. Now, the spread of the disease from endemic region to non-endemic region is not unique for India. It is equally applicable to other developed countries like USA, UK, and Britain, and France. This is because most of the people army people who are deployed during Iraq war or Afghanistan war, when they are coming back, they were carrying the disease. In addition, you can see there are a lot of post-Kalajar dermal leishmaniasis cases, which people believe that they are the reservoir of the future infection, and keeping the worry that there might be outbreak of the particular disease. In the absence of effective vaccines for the leishmaniasis, generally sodium antimony gluconate remains as the first choice therapy. But in the year 2000 to 2004, there was several drug-resistant cases evolved. And then the people started working on miltefosine and proteasine B as a proper therapeutics, but also people are encountering drug-resistant cases. So the bottom line is that visceral leishmaniasis won't go away. And also we should, keeping in the mind that we should come up with the proper therapeutics for treating this particular disease, and that requires proper understanding of the disease biology. Now, as I was mentioning, the disease biology understanding is very much important. Immune suppression is an important hallmark of the disease. It's a really huge problem. People look into this problem from different angles. Some people believe that it is because of the T cell dysfunction. Some people believe that it is because of the increased T cell, regulatory T cell reactivity. Some people believe that it is macrophage dysfunction, and we believe that might be dendritic cell, which is the potent antigen-presenting cell, may play a role in this process. This is because previous reports from my group, as well as other group, established that DC plays a key role in initiation and regulation of anti-Leishmanial immune response. So this particular fact is summarized in this particular cartoon, what we published in Microbiology Spectrum, where infected sandfly, when they bite the healthy individuals, they deposit the parasite in the dermis, and these parasites are getting cargoed by the dendritic cells to the local draining lymph node, and this is when they present this antigen to the T cell, effectively induce anti-Leishmanial immunity. But what happens if the diseases are now functionally impaired? Can they elicit the anti-Leishmanial immune response? It is not yet known. This is because of the fact that it is not yet known whether Leishmania donovani at all can immunoregulate dendritic cells, and if it is so, how it can mediate the effect. So to understand the effect of this particular, this first part, that whether it immunoregulates dendritic cell, we did in vitro study where we incubated DC with Leishmania donovani promesticode for several durations and then stimulated with LPS and looked for costimulatory molecule expression. We found that in response to LPS stimulation, there is significant upregulation of the costimulatory molecule expression. But infection with LD promesticode progressively reduced the costimulatory molecule expression on dendritic cell. And in agreement to this particular finding, we found that LPS-induced pro-inflammatory cytokine secretion was gradually reduced when we infected the DC with LD promesticode. So this suggests that LD inhibits DC activation and maturation. Now the question is how LD generally mediates this effect. So in 2006, I was coming across one paper published by Thomas Lasky's group stating that Leishmania major, which is a cutaneous Leishmania parasite strain, they express phosphatidylserine on their surface, and that phosphatidylserine expressing parasites are apoptotic in nature, and thereby it is suppressing host immune response. And this enables the parasite to keep the phosphatidylserine negative fraction to remain viable well. But it is not yet known whether Leishmania donovani is really expressing phosphatidylserine on their surface or not. So to investigate this issue, we isolated Leishmania promesticode from the stationary phase and immune stained with the antiphosphatidylserine antibody, and we found that a significant fraction of the Leishmania donovani promesticode is phosphatidylserine positive. And also, this fraction was more prevalent when we isolated LD amastigodes directly from the infected hamster, suggesting that Leishmania parasite express phosphatidylserine on their surface. And we are first thinking about whether these parasites were really apoptotic or not. But in fact, our hypothesis came out in another way. The parasites are, when we sorted annexin 5 positive fraction from the culture and did the tunnel assay, we found these parasites are really viable. I'm not showing the data. Subsequently, when we sorted this parasite and maintained it in a culture, those annexin 5 positive parasites were really replicating well. That indicates that the parasites, phosphatidylserine expressing Leishmania donovani parasites are not apoptotic, rather they are mimicking the apoptotic bodies. And that is why they are fooling the host immune response. Now the question is, if phosphatidylserine is getting expressed on the parasite surface, what could be the possible receptor on the host cell that can help them to interact with the parasite? One of the receptors that was previously known is T-cell immunoglobulin mucin protein 3, commonly known as TIM3, which is a checkpoint inhibitory receptor expressed on the dendritic cell. TIM3 was initially discovered by the Th1 specific surface protein, and this TIM3, however, later on was found to be expressed on the innate immune cells, including the dendritic cells, and was thought to promote the pro-inflammatory response. But in contrast to the prevalent idea, we found that TIM3 in fact acts as an inhibitory receptor on dendritic cell, and this particular observation was reproduced in many other labs, including Professor Vijay Kushru's lab. And now it is believed that TIM3 in fact acts as an inhibitory receptor. Now, if you look at the structure of this TIM3, you can see there is IgV domain which is having phosphatidylserine binding motif. So this indicates the possibility that phosphatidylserine can bind onto the TIM3 receptor. And now the question is really whether it is happening for the Leishmania parasite as well. Now, for this, we immunostained the Leishmania promestigotes with the TIM3 Fc chimera, and then we did the immunostaining with the secondary antibody, looked for how many parasites are getting immunostained with the TIM3 Fc chimera. We found that there is a significant fraction of the parasite getting stained with the TIM3 Fc chimera. Now, really, if the TIM3 is interacting with the parasite, can it interact with the phosphatidylserine or not? So we did another experiment where we pre-incubated the parasite with TIM3 Fc chimera, came up with the antiphosphatidylserine antibody-mediated immunostaining, and looked for whether this pre-incubation is minimizing the antiphosphatidylserine-mediated immunostaining. We found, indeed, it is happening. In the absence of TIM3 Fc chimera, the phosphatidylserine-expressing parasite are really giving the positive result. But when we pre-treated with TIM3 Fc chimera, there is a significant reduction in this immunostaining. So this suggests that TIM3 is interacting with the phosphatidylserine on LD. Now, the question is whether TIM3 is playing a role in disease suppression then and during LD infection. To look into this possibility, we silenced TIM3 expression in DC and analyzed the effect of this TIM3 silencing on pro-inflammatory cytokine secretion and co-stimulatory molecule expression. We found that LD promestigote infection significantly reduced LPS-induced pro-inflammatory cytokine secretion as well as co-stimulatory molecule expression here. But when we silenced TIM3 expression in the DC, we found there is the inhibitory effect of LD was withdrawn. Now, since NF-κB is the major pathway leading to the DC activation and maturation, we wanted to check the involvement of the NF-κB signaling in this pathway. We found that in response to LPS stimulation, there is significant increase in the NF-κB activation. But LD infection reduced this LPS-induced NF-κB activation, silencing of TIM3 expression withdrew the inhibitory effect of LD on NF-κB signaling. So the conclusion was that TIM3 is required for LD-induced inhibition. Now, if TIM3 is mediating the suppressive effect of LD on dendritic cell, can it have any impact on the disease biology? So we silenced TIM3 expression in DC in vitro, and then infected the DC with the LD promestigote and stimulated with LPS. And after all this in vitro manipulation, these diseases were adaptively transferred into LD-infected mice. And what we observed, in the absence of any DC transfer, there is a significant hepatosplenomegaly and the parasite burden in LD-infected mice. But when we adaptively transferred the LPS-treated DC in LD-infected mice, the hepatosplenomegaly is getting reduced, as well as the parasite burden is getting reduced. When we transferred LD-infected mice, LD-infected DC treated with LPS, we found that there is a significant increase in the hepatosplenomegaly and as well as the parasite burden. Interestingly, when we silenced TIM3 expression in DC, infected the disease with the LD promestigote and treated with LPS, and when we adaptively transferred these in LD-infected mice, there is a significant drop of hepatosplenomegaly as well as parasite burden. So this suggests that TIM3 of DC contributes to the disease pathology. Now, how does TIM3 inhibit dendritic cell and suppress anti-leishmanial immune response? To address this question, we looked for the role of interleukin-35. Interleukin-35 or IL-35 is an immunosuppressive cytokine which was initially reported to be produced by the regulatory T cell. It belongs to the IL-12 family consisting of IL-12, IL-23, as well as IL-27. And it is a heterodimeric cytokine consisting of IL-12P35 of IL-12 and EBI3 subunit of IL-27. But the production of IL-35 by dendritic cell is really contradictory, because some of the people believe that it can be produced by dendritic cell, and some believe that it doesn't produce by the dendritic cell. So we wanted to confirm whether dendritic cell really can produce IL-35, and if it is so, whether it is getting regulated upon LD infection. We found that for this we performed the in vitro experiment. We incubated the bone marrow derived DC with LD promesticate for different time periods and looked for IL-35 expression based on the double positivity of these two subunits, and here this red color indicating the fraction. We found that this IL-35 expression is getting elevated from 12 hours onward and persisting till 60 hours. But co-expression of both the subunits doesn't mean that IL-35 is really getting produced functionally. It is required to ascertain whether these two subunits are physically interacting so that the functional IL-35 is getting produced. So we performed the confocal microscopy analysis and looked for whether these two subunits are getting co-localized, and indeed we found that yellow signal is showing that these two subunits are getting co-localized. To further confirm whether these two subunits are physically interacting, we performed the immunoprecipitation assay, and co-immunoprecipitation assay indicates that these two subunits are indeed interacting physically with each other. Furthermore, the FRET analysis showed that IL-35 subunits are interacting, suggesting that LD induces IL-35 expression in DC. I'm not showing any data. We have further confirmed via ELISA used specific for IL-35 heterodimer, and we found there is a significant IL-35 production by DC in response to LD infection. Now, we have done a series of experiments in vivo suggesting that DCs are the early producer of IL-35, not the T cells, not the other splenic cells during Leishmania Donovan infection. If it is so, whether DC-derived IL-35 can influence the reactivity of the neighboring dendritic cells or T cells. Specifically, we wanted to look for whether DC-derived IL-35 can induce T cell IL-35 production, as well as whether they can regulate T cell proliferation. We found that in response, when we incubated the freshly isolated DC, incubated with infected DC supernatant, the LPS-stimulated co-stimulatory molecule expression, pro-inflammatory cytokine secretion, and also you can see that NF-kappa B signaling is getting dropped. Whereas addition of the interleukin-35 neutralizing antibody significantly withdrew the inhibitory effect of this infected DC supernatant. This suggests that DC-derived IL-35 can induce the suppression of the dendritic, neighboring dendritic cell. Now, our further evidence showed that infected DC supernatant induces IL-35 expression in the T cell. However, addition of the anti-IL-35 antibody withdrew the inhibitory effect. In addition, IL-35 present in the infected DC supernatant suppresses the T cell proliferation in IL-35 dependent manner, suggesting that IL-35 from LD infected DC stimulates IL-35 expression in the T cell and suppresses the activation of the neighboring DCs and the T cells. Now, if DCs are producing IL-35 and suppressing the DC and the T cell function and generating the immunosuppressive environment, can it have any impact on the disease biology? To determine this, we transfected the DC in vitro with anti-IL-35 neutralizing antibody. And these diseases were adaptively transferred into LD-infected mice. Our primary goal was that when this disease will be transferred into the LD-infected mice, there is a possibility that within the splenic environment, they will interact with the Leishmania parasite, which will induce IL-35 expression within the dendritic cell, this adaptively transferred DC. And then this IL-35 neutralizing antibody will bind this IL-35 and neutralize there. And how it is impacting on the disease biology. We found that when these IL-35 transfected diseases were adaptively transferred into LD-infected mice, there is significant drop of the hepatosplenomegaly and the liver and the splenic parasite burden. So suggesting that DC-derived IL-35 promotes disease pathogenesis. Now the question is how this IL-35 is getting produced within this dendritic cell by the upon LD infection. A series of experiments have been performed. I'm not going through the data here, but in summary, this particular data shows that LD interacts with the TIM3, and that is inducing the signaling mediator Bruton tyrosine kinase and at the downstream STAT3, and STAT3 then binds to the promoter region of IL-12, P35, and EBI3. In fact, we have identified a couple of the STAT sites in the IL-35 subunit promoters, and we have confirmed that indeed STAT3 is binding there. So when we block TIM3 with anti-TIM3 antibody, then we found that this IL-35 production mediated by the STAT3 is getting significantly reduced. And also when we suppressed STAT expression via siRNA, there is a significant reduction in the IL-35 expression. So this suggests that LD stimulates IL-35 expression in DC via TIM3, BTK, and STAT3. This finding marked STAT3 as the downstream effector mediating IL-35 expression. So the question is, if we can block STAT3 with some inhibitor, can we reduce the IL-35 expression in DC? And as well as whether that can prevent the disease pathogenesis. In fact, we came up with some FDA-approved STAT3 inhibitor with remarkably reduced STAT3 phosphorylation and also IL-35 expression. And when we adaptively transferred this STAT3 inhibitor-treated DC in LD-infected mice, there is a reduction in the hepatosplenomegaly as well as the parasite burden. However, IL-35 overexpression in this STAT3 inhibitor-treated DC withdrew the inhibitory effect of this STAT3 inhibitor. So this suggested that blocking IL-35 production in dendritic cell with an FDA-approved STAT3 inhibitor attenuates disease pathogenesis. For the sake of publication, I'm not disclosing the name of the STAT3 inhibitor, but nevertheless, we've observed that this particular inhibitor is having the potential. Now, once getting satisfactory result, we wanted to check whether if we deliver this particular drug orally in the infected mice, can it reduce the parasite burden? And we delivered this STAT3 inhibitor orally 3 times every week till 54 days, and we found that this STAT3 inhibitor actually reducing hepatosplenomegaly and the liver and parasite burden. So this suggests that FDA-approved STAT3 inhibitor can be a potential candidate for the visceral leishmaniasis therapy. Now, summarizing my total finding, we can say that during LD infection, when LD is interacting with the TIM3 via phosphatidylserine, TIM3 interaction, TIM3 signaling is getting activated. At the downstream, it is augmenting the Bruton tyrosine kinase activation, leading to the STAT3 activation, and this STAT3 is then binding onto this IL-12, P35, and EBI3 promoter region, leading to the enhanced expression of the IL-35. IL-35, in turn, it is inducing the disease suppression by blocking NF-kappa signaling, and also it induces IL-35 expression in the T cell, suppressing the T cell proliferation, and also type 2 T cell response. We have done series of experiments confirming that this particular pathway, how it is promoting type 2, disease promoting type 2 T cell response and suppressing type 1 T cell response. I have not included this data for the time's sake. And another thing is that this particular observation or immunosuppressive mechanism, in my opinion, it might not be limited for leishmanial infection. It has a broader implication for the other pathogenesis of other infectious diseases. I'd like to conclude my talk by acknowledging the help of my students, Mr. Sandeep Kumar and Shubham, who came up with the IL-35 story. Manish Mishra, he identified STAT3 as a signaling mediator and promoting the disease pathogenesis. Mohammad Nashad Akhtar, he first identified phosphatidylserine expression on the Leishmania parasite and how TIM3 is doing the immunosuppression. And this work was funded by Global Challenge Research Fund by UK, SCRB, and CSR, and I'm thankful to my institute, IMTech, for providing the infrastructure necessary for this particular work. Thank you so much.
Thank you very much for your enlightening talk. With this, we will be honoring this very prestigious Senior Oration Award, and IIS is really great to confer this award to you. Thank you.
Thank you so much.
The award carries a citation, a check of rupees 30,000 and a medal.
Thank you, Professor Avasthi and Dr. Nayak, for leading this meaningful ceremony and for your continued guidance, inspiration, and unwavering support to the immunology community. Now we will be taking a short tea break. After the tea break, the parallel sessions will begin at their respective venues, MP Hall, Seminar Hall, LHC 3 and LHC 4. I kindly request you all to reach the respective venues after 10 minutes of allotted tea break. Also, I request all the speakers who have not submitted their presentations yet, kindly submit it to the technical team to ensure the smooth flow of conference. Thank you. Enjoy your tea break.
हेलो 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 अंदर आवाज आ रही है हेलो 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 चेक हेलो 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 चेक 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 Hello, hello. Hello, hello.
मैं ये ट्राई 5 मिनट वेट कर रहा हूँ वरना चिल्ला रहा हूँ जल्दी से ट्राई कर कि कोई भी माइक से चल जाइए
Check, check, check. Hello, hello. Check, check, check. I request everyone to kindly assemble back to their respective sessions at their respective venues. Hello. Check. Hello, hello. Dada
Hello.
Niri. Hello, 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 hello. Hello, hello. Hello, hello, hello. आवाज आ रही है? Check, check, check. Check, 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 check. Hello, 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 hello. नहीं आ रही. Hello, hello,
Hello.
hello. Hello, 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 hello. Hello, 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 hello. बाहर आ रही है? Hello. Check, check, check. Hello, 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 hello. Check, 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 check. Check. I respect everyone to end the tea session and please assemble back to their respective sessions of their respective halls. I respect everyone to end the tea session and assemble back to their respective halls.
Check, check, check. Hello.
Now I request every dignitary. Hello, hello. Now I request every dignitaries to move forward to the auditorium as we are running late. Please cooperate and move towards the auditorium.
Hello, check.
Good afternoon, everyone. I hope you all had a refreshing tea break and are ready to begin the next round of exciting scientific discussions. We now commence the parallel sessions of Day 2 here at IMMUNOCON 2025, and it is our privilege to host you in the MP Hall for Session 1A themed Basic Immunology. This session will take us back to the very foundations of immune science, exploring the principles and mechanisms that shape every branch of immunological research. It gives me great pleasure to extend a warm welcome to our respected chairperson for this session. He will introduce our distinguished speaker and guide us through this upcoming talk. We are honored to invite Professor Santosh Kar, who is former professor from the School of Biotechnology at JNU New Delhi. His expertise is in immunology with research focusing on diseases like filariasis, malaria, and tuberculosis. He has been the founder director of Regional Medical Research Center in Bhubaneswar and a former chairman of the Center of Biotechnology at JNU. To accompany and lead this session, we were scheduled to have two chairpersons, Professor Santosh Kar and Professor Vatsala Mishra. However, due to unavoidable circumstances, Professor Mishra will not be able to join us today. The session will continue under the guidance of Professor Santosh Kar only. I would request Professor Santosh Kar to kindly come on the dais, please.
I have a problem. Yes, sir. Yes, please. The problem is that you have given me 7 people to now. Yeah. And how do I control time? You have to give me a thing and a timer.
Buzzer and timer. Yes, sir. We will provide that.
Do I go by my timing or you have a timing announce there. You can switch on that. So, 15 minutes. And I'm alone handling.
We are truly honored to have such eminent scholar with us today. With our esteemed chairperson now on the dais, I would like to request Professor Santosh Kar to kindly come forward and take charge of this session.
Good morning, everyone. Can we now start the session on basic biology, basic immunology, where we have 7 speakers. I will read out the name of everyone, one after the other. The first speaker is Dr. Girish S. Kirimanjaro. He is an associate professor in the University of Pennsylvania, Department of Veterinary and Biomedical Science at the Pennsylvania State University in US. His interest is diverse role of selenoproteins in B cell development, homeostasis, and humoral immunity. I think for a change, we have been talking a lot about T cells. So it is a good thing for you to tell us about B cells. You have about 15 minutes time, but just before 2 minutes, I will press a buzzer showing that your time is up. If I am very strictly keep time.
Sure.
If you don't keep time, then everything will not be finished in time.
Absolutely.
So the floor is yours, Doctor.
All right, thank you for that introduction. And first I would like to thank the organizers for inviting me to share some of our unpublished data here. And also thank you for referring that this is the first B-cell talk. And then this is not the first time. In most of the conference, it is mostly T-cells. So I'm the odd man out here. So, all right. This one? All right. So this is my first time in immunology, Indian Immunology Conference, so I thought I would just introduce my lab. My lab works on various aspects of host pathogen interactions, and then two of the projects are related to the role of micronutrient selenium on bacterial pathogenesis, which I'm not going to talk about, and the second one is the role of selenium and selenoproteins on humoral immune responses, particularly the B cells. So for this audience, what is selenium? Selenium is an essential micronutrient which was discovered about 200 years back and considered to be toxin for longest time. And in the last 50 to 60 years, now it has been considered as an essential micronutrient. This is used, normally we get this through our diet. And then the daily recommended dose is about 55 to 70 microgram per day. And then this is important for various aspects of human and animal health. Actually, the agriculture farmers, particularly the beef cattle people, already know the importance of selenium, and the importance in humans is being slowly recognized now. So the selenium is mainly used or incorporated as the 21st amino acid, non-classical amino acid selenocysteine. And most of the biological functions of selenium is mediated, at least in mammals, through this amino acid selenocysteine and their incorporation into selenoprotein. So we do need optimal level of selenium. The selenium deficiency leads to multiple diseases. The classical disease is called Kishan's disease, and another one is Kashin-Beck disease. Both have cardiomyopathy and then problems with the bone development and the joint development. And then in the extreme case, the excessive selenium causes what is called a selenosis. And then the selenosis is pretty common in northern part of India, particularly the Punjab and Haryana area, where the soil is highly rich in selenium. So this is a U-shaped, inverted U-shaped curve where you need an optimum level of selenium. Both deficiency and excess causes various disease. So just to give an idea of where the selenium deficiency occurs, so selenium deficiency is pretty common in sub-Saharan Africa, as well as many parts of US, and then the diet in Europe is also significantly lower in selenium. And then, of course, the biggest region where the selenium is highly deficient is in the big part of China, where many of the viral diseases are associated with the selenium deficiencies. And as I said, India has a different problem. The northern India has a higher selenium content, and then the Indian government actually buys all the grains produced in this region and discards them. And then you may be aware in this summer in Maharashtra, there was an extensive case of alopecia and then other disorders because the selenium-rich grain were brought into their food source. So, and it was traced back to a region in Punjab. So as I said, the biological function of selenium is primarily mediated through selenocysteine in mammal. It is a 21st amino acid. It's incorporated into many of the proteins. And then this is encoded by only one tRNA, which is, we call it as TRSP. That's the uniqueness of this. And this is actually recognized as the stop codon. It's a beautiful biochemistry in terms of how selenocysteine is co-translationally synthesized, and then it is incorporated into the protein, which I'm not going to talk about today. And there are 25 selenoproteins in humans, and then there are 24 selenoproteins in mice. And they can be grouped into broadly based on their functions. The classical functions of many of the selenoproteins is a redox regulation. So one of the classical proteins is the glutathione peroxidase, and then the new glutathione peroxidase 4, which is a hot shot for ferroptosis. And then there are other selenoproteins which are involved in thyroid functions, and then also thyroid hormone synthesis. And there are multiple selenoproteins whose functions are yet to be discovered. So my talk is divided into two. In the first part, I'm going to talk about, show the data where we show that for the first time, selenoproteins regulate peripheral B cell homeostasis and B cell development. And in the second part, I'm going to show about the mechanism behind the involvement of selenoproteins in regulation of B cell functions. So to address the role of selenoproteins in B cells, we developed a mouse model where we knocked out the TRSP gene. Remember, I told you that's a unique tRNA which encodes for the selenocysteine. If you knock it out, you knock out all selenoproteins. So, however, this is embryonic lethal, so we generated a tissue-specific knockout of selenoproteins, or the selenocysteine. So in this case, we primarily use a B cell specific knockout, so understand the role of selenoproteins specifically in B cells. So right off the bat, one thing we immediately noticed was that these mice had significantly smaller spleen. And then when we look at the total splenocyte number, they were reduced. And then if you look at the frequency of B cells or the total number of B cells, they're significantly reduced compared to your control mice. And then when we start looking at the specific subsets of B cells, and in the spleen, we have multiple subsets of B cells. For example, it includes transitional 1, 2, and 3, or the marginal zone B cells, or the follicular B cells. All of them are significantly reduced in the absence of selenoproteins. And then when we looked at the spleen and then did an immunofluorescence staining to look at the germinal center, what we saw was that the number of germinal centers were significantly lower in the absence of selenoproteins. However, the area was not different. And then if you look at the number of GC-specific B cells, they were significantly, again, lower in the absence of selenoproteins. And then as a point of interest, we also saw a significant decrease in T follicular helper cells. And that story is a different story. We'll probably, potentially, we'll talk about it in the next conference. So, and then we wanted to look at the consequence of this reduction in the germinal center numbers and the reduction in various B cell population. And then we measured the homeostatic immunoglobulin level. This is not in response to any antigen, but it's just a normal level of antibodies. We see a significant reduction in all classes of antibodies, IgM or various subclass of IgGs, and then all of them are significantly reduced in the knockout mice. Now, we wanted to see what happens if you give them a vaccine, right, or provided them an antigen. So we used Tdap, a pertussis vaccine. But our lab is historically interested in pertussis. So we used a pertussis vaccine, and then the only available was Tdap, but we are looking at the pertussis-specific responses in this case. When we vaccinate these mice and then look at the germinal centers, we see after 28 days, and we see a significant reduction in the number of germinal centers in the knockout mice compared to the wild-type mice. And that includes both area of the germinal, size of the germinal centers, as well as the number of germinal centers. And then when we look at the antibody level, this is now pertussis, Bordetella pertussis specific antibody levels. Again, we see a significant reduction over a period of time, either after primary vaccination or the booster. We have significant reduction in the antibody levels in both IgM and an IgG. And then what is the consequence of this? That if you challenge them with a virulent form of Bordetella pertussis and then look at the antibody level, again, the antibody level is reduced. And then after 3 days post-challenge, if you look at the bacterial numbers in the lung, it's almost a 2-log reduction in wild-type mice, whereas in the knockout mice, you don't see that reduction. So basically, bacteria keeps growing, and then eventually these mice succumb to the infection. So what it shows is that selenoproteins are somehow important in the B cell maturation, as well as the function of these B cells. Now, we wanted to take a step back and then see, is this B cell maturation in the spleen is the only thing that is affected, or something happened also in the bone marrow in terms of the B cell development? And I don't think I need to introduce this slide, but all of you know the B cells undergo multiple steps in developmental stage, starting from the hematopoietic stem cells all the way to immature B cells. And from the immature B cells, they exit the bone marrow and then enter the peripheral bloodstream. So one important aspect here we want to look at is, we wanted to look at is various stages of these B cell development in the absence of selenoproteins. And then we see that the first 3 stages of the B cell development, which includes pre, pro, and then the large pre cells, there's not much difference between the wild type and the knockout mice. However, something flips in the small pre-B cell stage. You suddenly see a drastic reduction in the small pre-B cells, and then that actually gets carried over to immature and then the recirculating mature B cells. So the main stage where the selenoprotein seems to be important is in between the transition from large pre-B cells to the small pre-B cells. So then we wanted to see what is happening in these cells, what is important in the large to small pre-B cell transition. Of course, we did the RNA-seq, and then there are multiple pathways, but the only one thing that I'm going to be showing here today is specific to the selenoproteins itself. So we see a differential regulation of 2 different selenoproteins here. I told you, right, there are 24 selenoproteins in mice, and then among them, the seleno-H and seleno-W are actually differentially regulated in the large to pre-B cells. So in the small pre-B cells, it looks like the seleno-W is highly expressed, and then that is not only at the RNA-seq, but at the protein level, we established that. And then we wanted to see what happens if you specifically knock out the seleno-W in these mice. So we generated this seleno-W knockout mice and then looked at the bone marrow various stages of development. And then again, as expected, the first 3 stages, nothing happened. There was not a significant difference. But to our surprise, in the knockout mice, you started seeing increasing number of B cells, particularly the small pre-B cells, and then the subsequent stages in the knockout mice. So what it tells us is that the seleno-W is a negative regulator of B cell development. If you knock it out, you start seeing increasing number of B cells. So, and then one thing and then if you look at the spleen at the periphery, then for some reason we don't see a significant difference in the follicular B cells. However, we do see a significant increase in marginal zone B cells. So that shows that somehow the seleno-W is important in the transition, particularly for the marginal zone B cell lineage. So all of you must be thinking now, what is the mechanism? What is this selenoproteins are doing? So in the B cell biology, all of you know that the B cells gets activated following BCR activation, and it is mediated through reactive oxygen species. And then if you look at the B cell development also, the pre-B cells, small to pre-B cells, small B cell is actually dependent on pre-BCR expressions. Therefore, we looked at the BCR signaling. So right off the bat, when we look at the ROS level following the BCR stimulations in the B cells, in the absence of selenoprotein, you see increased ROS level. And that is actually reflected both at the cytosolic level as well as the mitochondrial ROS level. And then consequently, you see increased cell death in the absence of selenoproteins in these B cells. Then the question is, can you reverse it? If it's the ROS mediated, so we treated with these B cells with N-acetylcysteine, which is a cysteine donor. And then in the absence of selenoprotein, now you can actually reverse these ROS levels, so reduce the ROS levels in the knockout mice, and the cell death is significantly decreased in the knockout B cells. Then can we do this in vivo? So in vivo, when we treat these mice with N-acetylcysteine, you can completely reverse the phenotype, and same way with both in spleen as well as the bone marrow. That's the end of it. So in summary, what we have shown is that the selenoproteins are critical for B cell functions as well as development, And it appears that there are two important selenoproteins. One is seleno W and another is seleno H. And there's both ROS dependent and ROS independent mechanism. And with that, I will have to acknowledge all the people who worked on this and then also the funding sources. And I'll be happy to take questions.
Thank you, Dr. Girish. And this is an interesting paper. I think it is open for one or two questioning from the audience.
It's a B cell. Nobody wants to ask questions.
If there are no questions from the audience, I am a chemist by training, and it is a very interesting chemistry problem. I would like to know from you, when you replace sulfur with selenium, cysteine to selenium, has the structural properties of selenium substituted amino acid has been understood properly, how it does all these changes, because it is very interesting that it is affecting the B cell properties in terms of immunoglobulin secretion and circulation. And hopefully it doesn't affect T cells.
In our model, it doesn't affect T cells because selenoproteins are specifically knocked out in the B cells. So there are a couple of papers where if you knock it out in the T cells, the CD4 T cells are perfectly fine in the development and everything. The function of CD8 T cells are affected. And then going back to your, I think what you're referring to is how is the sulfur and then selenium replacement, cysteine to selenocysteine, how is it changed? So you cannot, in the mammals, you cannot replace selenocysteine with cysteine. Because these proteins cannot be, because of the nonsense mediated decay, so they won't be transcribed at all. But in a bacteria, you can actually change the selenocysteine to cysteine, and the catalytic activity is almost like 1,000-fold reduced. So evolutionarily, the selenocysteine is, for whatever reason, it's turned out to be important. So you cannot change it back to sulfur. Yeah.
Just I want to control the questioning. Who is that asking? You did not ask.
Is there a codon exist for selenocysteine?
It is a stop codon. As I said, it's biochemistry is beautiful. It is recognized by a stop codon, but in case of the selenoprotein messenger RNA, there is a 3' C-KIS element, which is a structural way, and then that will be recognized by this tRNA, and then only it could be replaced with selenocysteine. And there's in the C-KIS element, there's an additional stop codon.
So is it a post-translation modification?
This is a post-translationally, selenocysteine is synthesized at translational
Then
level.
it is incorporated.
Yeah.
Okay, thank you.
So in order to control time, let us not have a free for everybody. There is a restricted time. I just want to ask you last question. What is the source of selenoprotein in nature?
So the selenium is primarily obtained through multiple green leaves and then meat and then also seafood. And many of the nuts are rich in selenium. So in the nature, it is comes as a selenomethionine. And in the mammalian cells can convert selenomethionine to free form of selenium, and that gets incorporated as selenocysteine. So particularly the vegetarians have a deficiency in selenium if the soil is selenium deficient.
So is there any pathological condition in which the absence of this micronutrient is going to be helpful? Because you see the immunoglobulin production is regulated. Is there any pathological condition in human where antibody production should be controlled, and by micronutrient manipulation you can control the immunoglobulin production?
So that's a whole new talk. So we have shown that what is considered as the adequate level of selenium is not adequate for the B cell. We can make the B cell function much better. So if I'm invited back, I'm going to show that data next year.
Thank you, Dr. Girish.
Sure. Thank you.
I'm sorry, because there are 7 speakers, and if we don't control the time, then we'll be well past lunch time. I do not want to do that.
One important announcement. Sessions have already commenced in Seminar hall, LHC 3 and LHC 4 as well. Those who are interested can move there also. Thank you.
Professor Taylor from NII is the next speaker. Deciphering the specificity within interferon regulatory factors leading to dendritic cell differentiation and innate immunity. Again, a very, very important, interesting topic. Praful, you have the job of presenting your thing within 13 minutes. I'm sorry. I would give you much more time, but 13 minutes, and then I will press.
I've made a very specific one. So yeah, great. Great. So at the onset, thanks to the organizer for giving me opportunity to present our work here. Okay, great. Great. So I think today my lab is mainly, it doesn't work, but my lab mainly works on dendritic cell development. And today we are going to talk about deciphering the specificity within, there is something has come up here, so I don't know how to close this. Okay, I can see here and then I can talk. This is blocked. So we are working on dendritic cell development. I'll be talking about deciphering the specificity within interferon regulatory factors. So I'll give a brief introduction about dendritic cell, brief introduction about IRFs, and then I'll go on to my work. And my work today is going to be more of molecular biology than of a cellular biology, and then you'll see why that is so. Great. So yeah, okay. This works. This doesn't work. So I'll have to work from here. Okay. Okay. So I think Dr. Pradeep Sen introduced dendritic cell very nicely, so audience knows about the importance of dendritic cell. I mainly work on mouse dendritic cells. We know dendritic cells are prime or professional antigen presenting cells. In mouse system, they all express CD11C on the surface, so CD11C becomes a pan-DC marker in mouse system, and then there can be loosely classified as plasmacytoid dendritic cells, which produce lots of type 1 interferon, or a classical dendritic cells, which were discovered way back in 1973 by Zhenwil Cohen and Ralph Steinman, which also comes in the form of CD4 positive, CD8 alpha positive, or CD4, CD8 double negative. And all these dendritic cells, this is very loose classification. As we go ahead, we'll see much more specificity in them. And all these different dendritic cell types are developed from dendritic cell progenitors or from a common lymphoid progenitor or common myeloid progenitor. But what we know today is there are two classical culture systems. That's GM-CSF-based culture system that depends on IRF4 or directs through IRF4 and gives rise to CD4 or then double negative dendritic cells, and IRF4 dependent dendritic cells, which is directed through FLT3 ligand signaling, gives rise to PDCs and CD8 alpha dendritic cells. And since there are different markers are used by mouse and human system, there was a unified classification system was developed by in 2016, which now classifies CD8 alpha positive cells as CDC1, and then these two combine as a CDC2. So now my next talk will be CDC1, CDC2, and PDCs. Great. So now we know that there are different subtypes of dendritic cell. Why one should be interested in looking at what is the differentiation of these dendritic cell subtypes? Because of the properties that these three different DC subtypes possess. Okay. So CDC1, I'll start with CDC1. They express very high IL-12 at very early upon stimulation that helps them to define the T cell polarity. And then studies have shown that tumors that have very high CDC1, that is called CD103 positive, CCR7, CDC1 positive subtypes, they have better tumor regression. And these are the cell types that naturally cross-present antigen, suggesting that they can take an exogenous antigen and then present it with MHC class 1 so that they can stimulate CD8 T cell responses. PDCs, as I mentioned, they produce very high type 1 interferon, so they becomes very important for antiviral responses. And since they produce very high type 1 interferon, they get activated, they can activate autoimmune responses, and they have been implicated in loss of autoimmune diseases like SLE and arthritis. And CDC2, which has very MHC class 2 expression, they are into classical MHC presentation and classical T-cell responses that you see, okay? And there are many other factors like IRF2, RELB, and BETF3-ID2 that works with IRFs to help the differentiation. So since the dendritic cell subtypes are very small in number, most of the studies have been come from studying the spleens or various tissues from a knockout mice, and then many molecules have been identified. And here is the lineages that have been identified based on these molecules. This is in 2016, but then after that, there have been some molecular mechanism have been understood in terms of how they direct these different disease subtypes. And we have been interested in how does this IRF molecule direct dendritic cells. So IRF8 that my laboratory has been working on directs dendritic cells, and they are the one that induce BETF3 and ID2, which are very important for CD8 alpha DC differentiation. What we showed is IRF8 is master regulator, and then BETF3 ID2 has a synergistic effect, as you can see here. ID2, which is a negative beta helix loop helix transcription factor, it blocks E2.2, which is an E protein, and when it binds to E protein, ID2 does not have DNA binding protein, it blocks E protein for binding to DNA, which has a promoter which has E boxes, and it blocks PDC development, and it helps in CD8 alpha DC development. And BETF3, which interacts with IRF8, yesterday Dr. Pali Bulandran suggested that BETF3, which is a member of AP1, does not have a synergy with IRF molecule, but here myself as well as Dr. Ken Murphy's laboratory has shown that BETF3 interacts with IRF8, and this interaction is very important for CDC1 development. So I think what Pali Bulandran sir suggested was, I think they have a divergence of these pathways upon stimulation rather than at this development stage. Then also there is the importance of type 1 interferon. We had identified that IRF8 regulates, this IRF8 beta-3 interaction regulates PFKF beta-3 regulation, which directs the formation of fructose 2,6 biphosphate, which also in turn is an allosteric activator. So I'm not going into detail into my past work. I'll just sharply go into what I'm going to talk today. This is IRF. There are 9 members of IRF. They have very conserved DNA binding domain, as you can see, which has tryptophan, 5 tryptophan conserved residues. They have a variable domain that is IAD domain, which defines the interaction with the partner molecule, and based on this interaction, they can either activate or repress the promoters. And what is here, I can see GAAANN is a classical motif on which this DNA binding motif can bind, DNA binding domain can bind, and it can activate the gene transcription, okay? And this is, when it's repeated, it is called ISRE, which is called interferon stimulated response element, which is found in interferon gene as well as interferon stimulated genes. I will come to the next domains also, but this domain can change based on the partners. Okay, so I'm coming to my talk today. The IRF4 and IRF8, I know, as I've told you, it forms a central axis of DC differentiation, right? And we looked at various IRFs, and when I see the homology between them, IRF4 and IRF8 are very similar, and I'll show you in my next few slides that they are even more similar when you look at them very closely, okay? But still, they are, even though very close, but they define the differences in terms of the differentiation of dendritic cell types and the cellular responses. Great, but if you look at the similarity in terms of the surface marker, they induce almost the similar kind of markers when you express them on the surface. But when you look at the molecular-based basis, they direct different transcription status or different transcription profile that you can see from here. These are the genes that we already knew that is regulated by IRF4 and IRF8. So we went ahead and we looked at this global gene regulation by the IRF4 and IRF8. So we expressed IRF4 and IRF8 in double knockout dendritic cells, and then we looked at the pattern. You can clearly see IRF8 and IRF4 are directing different kind of transcription programs. And you can see that they have very common genes that are downregulated, common genes that are upregulated, but indeed they have a distinct pattern that they are differentiating. Okay, so from this study, we picked up some genes and we validated those genes that indeed there is a specificity in terms of IRF4 and IRF8 that is induced. Okay? You can see green and red bars. Then we validated them by using, this is the IRF8 knockout mice, which lacks PDCs and CD8 alpha DCs. And you can clearly see here that there are some of the genes that are regulated by IRF8 are missing from these knockout dendritic cells. So we validated them by RT-PCR and also looking at the mouse model. We are now have got IRF4 knockout model, and we are validating IRF4 genes too. But what we were important is looking at the structure or sequence of these. We can see clearly that DBD is highly homologous, and people suggested, there was a previous report that suggested that C-terminal region of IRF4 and IRF8 have a negative role in terms of they control the DNA binding. So we first swapped these regions by using antibiotic complementary PCRs. So you generate 2 PCR products and then they combine both of them to generate the chimeric proteins here. Okay? So by using this technology or this technique, we exchange the C-terminal region between them, and then we looked at the gene transcription pattern. What you can see here, the gene that was induced by red color in IRF8 is still induced by IRF8 even if you change the C-terminal region with IRF4. So specificity, you can clearly see the red and green, yellow is work similar. So specificity does not change if you change the C-terminal region. Now, next we change the IAD domain here. IAD domain is the one that controls the interaction with other partner molecule, and you can clearly see here the red protein, which is a red line, which is IRF8. When you exchange them with IRF4, now IRF4 is able to induce the same gene that was induced by IRF4. IRF4 can induce the same gene that was induced by IRF8. You can see here everything. Right? So it suggests that IAD domain is the one that controls the specificity. And here, just to control, as a control, we wanted to look at some DNA binding domain because that was highly similar. But when we change DNA binding domain here, you can clearly see DNA binding domain exchange can also suggest the similar response as that of the IAD domain, that's suggesting that it's not only the interacting protein, but even a DNA binding domain also can direct the specificity despite them being very similar. As you can see here, I already shown you how similar they are, but most of the differences occur in the first few amino acids. As you can see, there is some deletion in IRF8 and there are some missing amino acids here and there. So we exchange these first 48 amino acids to confirm the specificity within IRF8 and IRF4, and what you can see here, just focus on the last 2 lanes here, that even if you exchange the N-terminal 34 amino acids and 48 amino acids, there is no change in specificity, suggesting that despite having so much of change here, this is not the region that controls the specificity within IRF4 and IRF8. So we went ahead and we looked at, this is complete DNA binding domain. As you can see, many amino acids are conserved. You can see so many stars there, suggesting they are very highly similar and same amino acids. But we found there are 2 amino acids which is different, that is glutamic acid to valine and asparagine to proline in IRF8. So we exchanged them, both of them, and what we see here, when we change this E to V or V to E here, we don't see much difference in terms of the specificity, but when you change this asparagine to proline in IRF8, now IRF4, 2 minutes, I'll complete. So in IRF4, you can clearly see that IRF4 is now able to induce the genes that were induced by IRF8. This is the GLEC9A, BACF3, and ID2, right? So there was a crystal structure available about IRF4 binding to DNA. We took the cues from here. Then we analyzed with Krishna Kishore Imanpuri from AIMS, and this is a structure that clearly shows the overlapping structure between IRF4 and IRF8. This is the common mutant that interacts with the DNA. This is K79. We validated our structure by looking at this, which clearly suggests that lysine is a positively charged, which interacts with the negatively charged DNA. When you change this lysine here with the glutamic acid, which is negatively charged, it doesn't allow it to bind DNA binding domain, and it clearly can see that it does not interact with the DNA. Okay, so this is the one, but then you can look at the P015N and N90P, which is the one that we looked at the construct, and we find that it is not directly interacting with the DNA, but there is an allosteric interaction between DNA and this domain, and that is what we are rightly studying by the MD stimulation studies. But even if you looked at, if you remember my graphs, you could see that it is the P2N transition did not completely reverse the change. It could do what IRF8 could do, but it was not complete. So we thought that there must be some specificity left in DNA binding domain. So we created a lot of structure here, as you can see, but I'm not going to specificity and I'm not going to discuss everything because there's not much time. But what I'll try to say here is that the deletion of those 8 amino acids there plays a very key role. This deletion, as well as the 8 amino acids, that is the region, along with this NP, is the one that controls the transcription factor. Here you can see the black bar, the last one. Okay, so we have been able to decipher the specificity here. I'll just show you this last slide. This is my last slide here. This is GLEC9A gene that I've been showing you repeatedly. This is the gene that is expressed by CDC1 here. You can clearly see not in CDC2. And we looked at the binding pattern by ChIP-seq data analysis. You can clearly see here that there are IRF8 peaks, and now CDC2 does not have IRF4. That one peak is missing, and that is where the X-IRF binding site is there, PTCC as well as GAAA here. So now we are simulating this. We are working more on it, and then we'll be defining more in the incoming presentations. And currently we are performing RNA-seq experiments to validate this. We are trying to identify more genes and trying to see how this global transcriptomic pattern changes when IRF4 and IRF8 is expressed in the different dendritic cell types. This was the work that was started with Hemant, Monica worked on it, and then Bhupendra, who is in center, is the one who is working, and currently my current students are taking it forward. So these are my funders and collaborators. Thank you so much. Thank you.
Thank you.
Sorry, I had to go a little bit fast. And
it is open for questioning. 2 questions, please. One up. We have to.
Yeah.
Hi, myself Shubham from PGH Chandigarh. So very wonderful talk in terms of like you provided that basically created a database for the IRF4 and the IRF8 in terms of the mutation changes in a different domain and how they are basically playing an important role. So I mean this is now my question is with respect to the immunodeficiency. Like we know that IRF4, I know like there are examples like in IRF4 deficiency in human leads to the combined immunodeficiency having a low T and B cells, whereas the immunodeficiency of the IRF8 leads to the NK cell activity, not the NK cell, I would say the dendritic cells defective and the defective development of the cells of the dendritic cell. So I'm just trying to understand whether you looked into the role of the IRF4 in the B and T cell development.
So my laboratory mainly works on dendritic cells, but I'll just tell you one thing here is that if you look at the deficiency pattern, that will give you a clue for further thinking. So IRF8 knockout mice, okay, IRF4 and IRF8 have a very important role in B cell development, 2 T cells, 2 as well as dendritic cell. We work only on dendritic cell, but to give you an idea, IRF4 knockout mice develops a CML-like syndrome. Okay, so they have more of neutrophils and granulocyte rather than macrophage monocyte lineage, right? Whereas IRF4 knockout mice have a B cell proliferation there. Okay, suggesting that there may be some finer differences between them. And I think also it's not about IRF4 and IRF8. I also showed you there are many other factors. Okay, by which the cell interacts with the stromal for it to get differentiated. And what we find now is IRF4 and IRF8 can direct because they are very similar, but also what kind of programs are going on and what are the factors that it gets to interact with, that depends and that directs a different differentiation. And I think you already know and you are already cited, if you look at the various human studies have already shown that IRF8, there are specific mutations, lacks certain type of dendritic cells and certain type of that kind of studies. And that's what we are also interested in.
Yeah, thank you very much. Just appreciate that your work is really helpful for the clinician, where you have created all this mutation database and showing the differences in terms of activity, and that would basically help in the patient data as well.
Yeah, we are already working with Dr. Sudhir Gupta with his group, and then we are trying to understand immunodeficiencies in each other.
Yes. Thank you very much.
Thank you, Professor, for a wonderful talk. Very important from the development vaccines, antigen presentation, and understanding how dendritic cells control immune response. Thank you very much.
Thank you so much.
Next speaker is Dr. Shravan Swarat. He's an associate professor at the Indian Institute of Science, Education and Research, Mohali. His talk is on RABA8A mediated improved functioning of the dendritic cells.
Cursor.
Pointer.
Cursor.
Cursor.
Can I start?
Yes.
Yeah. Okay. Thank you for the presentation opportunity.
The same thing, 13 minutes, 2 minutes before I will.
Yeah, okay, fine. So I'm going to talk about the role of this small GTPase, RAB8A, in regulating DC functions. Particularly, I'll be talking about, or I'll be showing data on role of these cells reconstituted of RAB8A in antiviral immunity. And I will end up my talk by showing some data on probably it also has relevance in anti-tumor immunity. As you know, the aged population actually is highly susceptible to viral infection. And we know that the immunosenescence essentially means the ability of aged population to respond poorly to vaccines or any infection. Yeah, so and the age-related aberrations are evident both in innate immune cells as well as in adaptive immune cells. A restricted repertoire of T cells, inefficient TCR signaling, all these things are observed in aged individuals. And as a result, so if these cells respond and are recruited in the response, so there is an oligoclonal expansion of these cells, and which lead to very poor memory generation. While in adults, so this actually is far better than what you get in aged population. Similarly, the responses of innate immune cells, such as dendritic cells, are also compromised as you become older. And these are reflected in terms of production of cytokines, the ability of these cells to stimulate T cells and eventually expand adaptive immune responses. So what we were interested in knowing was what are the molecular details or insights into the functioning of the dendritic cells. So what we did was we performed an RNA-seq analysis on splenic DCs from young animals and aged animals. Young animals essentially were 8 weeks. That if you actually translate to humans, so this will be approximately 20-25 years of age. And in aged, so this is close to 80 or 85 years. So as you can see from this plot, so a lot of inhibitory molecules are expressed by dendritic cells. But a lot of molecules which are actually feeding to interferon pathway were downregulated. And these things are actually shown in this cartoon here. So starting from interferon ligand and then JAK-STAT pathway, so all the transcription factor and the interferon stimulating genes all were downregulated in splenic DCs isolated from aged animals. And this is at basal level. So unmanipulated mice, they are inherently deficient in responding to probably viruses because of the compromised interferon response. However, if you isolate splenic DCs and do functional analysis, this is very frustrating because you get very few number of cells. So we resorted to bone marrow-derived dendritic cells from aged and young animals. The first experiment that we did was compared the transcriptome of splenic DCs and bone marrow-derived dendritic cells. And we find that of the downregulated genes, so you have approximately 81% similarity. And on the upregulated genes, so this similarity is close to 62%. So we consider that these cells can be used as a surrogate for analyzing responses in the aged individual. This next experiment that we did was, so we isolated these cells from young bone marrow cells, differentiated them into DCs, and stimulated these cells with influenza A virus with 0.5 multiplicity of infection, and analyzed interferon response. Clearly, you can see that the interferons, alpha, beta, gamma, transcription factor, the effector molecules, they were all highly downregulated in the cells isolated or differentiated from aged mice. And exactly similar results we also obtained for human PBMCs, which were enriched for dendritic cells. We also found that the growth factor receptors, IGF-1R and small GTPase, RAB8A is downregulated quite significantly in these aged individuals or aged animals. Okay, so as of now, what we know, if we isolate DCs from aged individuals, so they are very poor in mounting interferon response. So they also downregulate RAB8A. So we showed a lot of data on comparing the wild type and interferon receptor knockout animals, and we find that these cells from young animals, which are deficient in interferon response, they are very poor in co-stimulation and antigen presentation. However, this is fine, but our idea was to identify a molecular switch that you can pull on to jack up the immunity. So, and then we resorted to RAB8A. RAB8A is a small GTPase responsible for cellular differentiation, cell movement, and vascular transport. So whether this RAB8A can serve as a molecular target for enhancing functioning of dendritic cells. The experiment that we did was, so we basically isolate bone marrow cells from young, aged animals, and the aged animal DCs were also reconstituted of RAB8A, and then you let them differentiate into BMDCs, and after that you analyze their ability to produce cytokine as well as their ability to stimulate antigen-specific T cells. And you can clearly see from here, so following reconstitution, you get very high expression level of RAB8A. And alongside, so you also see a very high expression of interferon alpha, beta, and gamma. So I'm not showing you the data, but all other effectors were also highly upregulated as a result of RAB8A reconstitution. Then we also did this co-culture experiments of these DCs pulsed with the antigen, and the model antigen we are using is OVA, and stimulating these cells with OVA and co-culturing them with OT1 cells. So essentially we are looking at cross-priming of these cells. As you can clearly see, the aged BMDC pulsed with OVA actually respond very poorly in terms of stimulating these antigen-specific cells. You don't see much proliferation here. Young cells are fine, so they induce very rapid proliferation, but the RAB8A reconstituted aged cells actually almost recover their phenotype, and they stimulate these OT1 cells almost to the similar level that you obtain for young animals. After showing this in vitro analysis, then we did some in vivo experiments, essentially in form of a DC vaccine. So you have DCs, you pulse them with the antigen, and then you put them back in a congenic animal, alongside some antigen-specific T cells, which can respond and generate effector cells, and then some of these cells also make a transition into memory. So this is the experiment. So, and once the memory actually is established, then you recall these animals with influenza virus encoding this epitope, synfacal, which can stimulate these OT1 cells that we transferred to begin with. In the acute phase, if you look at the proliferation of these cells, so the young DCs actually, they stimulate OT1 cells pretty nicely. Aged cells were highly compromised, and following RAB8A reconstitution, so you get almost similar level of expansion of these cells as you find in young cells. And when you do a recall with this virus, so in all the organs, whatever the organ that you look for, so you have a very rapid expansion of the antigen-specific cells. Essentially what we are looking at is the donor cells that we transferred in these animals. So they are expanded in mediastinal lymph node, in spleen, in lungs. And as a result of this expansion, these animals are better able to control this virus. So we get almost 2 log-fold viral titers in these animals. And the gross lesion, if you actually isolate lungs and look at the morphology, so that actually is also quite striking, that the RAB8A reconstituted aged DCs, because of their ability to produce cytokine and more efficiently by inducing antigen-specific T cells, are able to control this virus, and the lung pathology is not evident that much. So then we also looked at some of the molecular details performing confocal microscopy. So, and what we are looking here is, sorry, this is not very clear here, but essentially you are pulsing these DCs from young animals, aged animals, or the aged DCs which were reconstituted of RAB8A, pulse them with OVA, and over a period of time you look at the expression of RAB8A, RAB11, and RAB11 actually, you know, is an endolysosomal marker. And you also look at these peptide MHC complexes, which can be detected using this CD25D1.6 antibody. So as you can clearly see, the young DCs are fine in terms of expression of all these GTPases, the expression of RAB11, and also production of the complexes over a period of time. And aged DCs actually do not do well. Rather, they actually very poorly generate those peptide MSC complexes. But following RAB8A reconstitution of these aged DCs, so they regain almost all the molecular signatures, which are good for presenting these peptide on surface of these MSC molecules. This is just a blown up image at 24 hours of one of these cells. So if you look at the co-localization of RAB8A, RAB11, and these peptide MSC complexes, you find very efficient co-localization in young. not so well in AIDS cells, but if you have the AIDS cells reconstituted of Rab8, so they do even better than your young cells. Okay, finally, we also compared the transcriptome of the cells which are isolated from young animals, aged animals, and the aged animals which were reconstituted of Rab8. And you can clearly see that the genes which are responsible for immune responses, so they were actually reconstituted almost to a similar level that you get for young animals. So which actually reaffirms that Rab8 actually is changing the transcriptional profile of these cells, and as a result, these cells become better at their innate immune functions. So what I'm saying here, to summarize the talk, if you have these young cells, so they, when you differentiate in presence of the cytokines, they express high level of co-stimulatory molecules. They respond to antigen, produce a lot of cytokines, particularly type 1 interferons, and they also expand antigen-specific T cells and generate very nice memory response. Although I have not shown data, but we also have actually obtained this data. When you have young cells, young DCs from young animals, and then you deplete Rab8 by knockdown approaches, so these cells actually lose function to almost the level that you obtain for aged animals. On the reverse, if you have aged bone marrow cells, if you reconstitute them of Rab8, so they almost become similar to what you obtain for the young cells. Okay, so this is about the antiviral thing, and last slide, if I have some time, I can show you that the same thing actually is also happening in a tumor microenvironment. And this is with the B16, B16F10 melanoma condition, so where basically you inject these cells ectopically under the skin, and you basically let a tumor develop. And this is actually shown that the tumor developed nicely in these animals. If you isolate tumor and then isolate DCs from tumor microenvironment and from splenic tissues or lymphoid organs from these animals, and you compare the transcriptome of these cells, particularly using RT-PCR, so they exhibit exactly the same phenotype that you also obtain for the aged individuals. So their ability to mount interferon response is compromised, and the Rab8 expression is significantly downregulated. So we think that these cells can also be reconstituted of Rab8 to recover or to restore anti-tumor immunity. And with this, I thank you. And if there are any questions, so I would be able to take. The work that I have shown actually was done by Sudhakar, Arihant. Funding is from these agencies. And obviously, I would like to thank Animal Facility, because if you have to maintain animals for almost 2 years, so you have to have a very clean animal facility. So, and we have an excellent animal facility at ISER. Thank you.
Thank you, Dr. Sarawat. Questions?
Great, that's excellent talk. I think I saw that you are working on CD11C positive, CD11B positive, CD103 negative. Basically, you are working on CDC2, and CDC2 are the dendritic cells that depends mostly on RELP expression and IRF2 expression, which are mostly suppressing type 1 interferon. And you don't generally expect a type 1 interferon to be produced by them, but the data that you showed is really commendable. That indeed in old animals you can get those. The question is about role of this factor, the Rab8 that you mentioned. How much of it is directly interacting with the machinery that works with type 1 interferon induction versus that of machinery that works in vesicular transport, and then it helps in secretion?
There are several questions that I need to answer here. One obviously is because the Rab8 is working upstream following this signaling through growth factor receptors, right? Upstream to PI3K mTOR pathway. So if you reconstitute Rab8, obviously, so you are actually rewiring these cells so they become better at their differentiation. And we know that Rab molecules are very critical for vesicular transport, trafficking of proteins within the cells. And that precisely is happening here. So if you produce cytokines, so those cytokines can very efficiently be released if you reconstitute these cells of Rab8. And in terms of their ability to expand T cells, obviously this is through generation of very efficient generation of peptide MSC complexes. Those are then displayed on surface of these cells and engage with T cells. And as a result, so you get very efficient expansion of these T cells, right? It's a very generalized effect in terms of cytokine secretion, in terms of trafficking of these proteins on the surface of these cells. And that is why, and even the viability of these cells is also enhanced. So if you look at the BCL2 expression on these cells, so that actually is pretty nicely upregulated. Yeah.
Any more questions? If there is no more question, I would like to ask you one question. Sure. It's very interesting that you can reverse the aging process in dendritic cells. So we are back to square one now again. People were talking some time ago about aging and reversing the aging, but now you have a molecule which is showing that it's very clear that dendritic cells, young, has particular phenotype, aged, that phenotype is lost, but as you add your molecule, the partially phenotype is reversed. Can you now say that you can reverse aging?
Well, probably not aging as such, but the immune functioning in aged population could be enhanced, probably using a DC vaccine kind of approach, where you have these DCs, isolate these DCs and then reconstitute these DCs of RAB8A, and then stimulate with whatever the antigen you are interested in mounting a good immune response. But as such, whether this molecule can reverse aging, I am not sure. Okay, but yes, it can for sure try to reverse immunosenescence. Right, so immunosenescence can be reversed, at least for DC function. And DC, are very critical for inducing all kind of responses. So you are hitting where actually it hurts the most.
Thank you for that clarification. This is very important. Thank you, Dr. Sharma. Next speaker, Dr. Meghang Srivastav. Dr. Meghang Srivastav is a senior principal scientist in the molecular microbiology and immunology division of CSIR CDRI Lucknow. He is going to speak about dissecting the role of innate immune effector cells during filarial manifestation of tropical pulmonary eosinophilia. Dr. Meghang, your time 13 minutes plus 2 minutes.
Yes, thank you. So I thank Dr. Ambakra and the entire team for giving this opportunity, and thank you, Professor Ka, for chairing the session. So I'll take you all to entirely new domain of parasite biology and talk about the role of innate immune effector cells during filarial manifestation of tropical pulmonary eosinophilia. So those who are not familiar, it is a manifestation for lymphatic filariasis, and we all know that it is lymphatic filariasis is an old neglected tropical disease prevalent in the Indian subcontinent and of course parts of Africa as well. So the earliest evidence is around 1500 BC from Egypt, and in India the first record is from Shushruta Samhita, which was published around 6th century BC. So interesting is that there are only 3 major parasites that cause human lymphatic filariasis. Among them, Wucheria branchofti, Brugia malai, and Brugia timori, and the vectors are mosquitoes. So most of us actually recognize this disease by its manifestation, which is elephantiasis and of course lymphedema. So elephantiasis, lymphedema, and of course, in females, you have lots of breast enlargement, and of course, hydrocele as well. So if you look at the disease that, India shares 40% of this burden, and the current elimination target for LF is 2030, that is declared by the WHO through the use of 3 drugs that are used in combination, diethylcarnosine citrate, albendazole, and ivermectin. However, there are significant challenges to this elimination therapy. The first is that we do not have drugs that can kill these adult worms. There are no vaccines either that can support the elimination process, and we do not yet understand the complete immunobiology of this disease. So the parasite size is a major challenge as compared to the 2 other prevalent parasitic diseases in India, namely malaria and leishmania. This is an extracellular parasite and it is pretty huge in size. So there's the adult worm, and there's an infective larvae, okay, that is transmitted by the infected mosquito, and that leads to the manifestation of the disease manifestation in the infected host. The offsprings of these adult worms are microfilary, which are pretty big in size. As you can see, the microfilary from Brugia malae is approximately 200 micron in size, and Wucheria branchofta is even bigger. The infected larvae is of the order of several millimeters in size, about 2 millimeter in case of Wucheria branchofta that you can see. And if you see in the bubble graph, this is how the adult worm would be in comparison to the size of MF and the infective larvae. And just for comparison, this is the size in the bubble graph of an RBC that is about just 7 microns, and the average size of human macrophages is 20 micron in size. So you would appreciate the fact that it is beyond the capacity of our best known phagocytic cells to engulf such big worms, okay? Not even the babies or the offsprings, which are order of 10 times the size of the best phagocytic cell known in the humans. So what we did, this is the scenario of the adult worm that has been pricked with a needle. It is releasing its offspring. So this is a gravid female, completely fertilized by the male inside the host, and this is releasing its babies. This is the offspring. They are actually sheathed. This is also sheath microfilary, slightly bit more advanced in the developmental stage as compared to these ones. And you can see that these have a certain movement, which is known as the filarial dance sign, and all the different stages of these parasites, the infective larvae, the microfilary, and the adult worms, continue to show high amount of motility throughout their life cycle. This is the infected larvae that you can see here, and this is the adult worms actually that you can just see with the naked eye, and they just entangle themselves and of course form nests that blocks your lymphatic drainage system, leading to lymphedema and in the chronic stage elephantiasis. So what is interesting is some of these microfilary actually that circulate in the blood of the infected host would end up into the lungs and lead to a manifestation that is known as tropical pulmonary eosinophilia, because you have high numbers of eosinophil count accumulating in the lungs of such patients. The prominent clinical correlates are that you have elevated IgE, IgG, and of course diffuse reticular nodular opacities, and among other symptoms. And important is you have huge numbers of eosinophils coming into in the lungs of the host, leading to alveolar eosinophilic alveolitis. Okay, there is no standard therapy to manage TP, and patients that are treated with DEC, which is a potent microfilaricidal drug, at least 20 to 40% of them relapse within 5 years. The immunological underpinnings is absolutely complicated. So what we did is, these parasites, this parasitic stages, when it gets entrapped into the lungs, they degenerate, secrete their antigenic composition, and of course among them is of course EVs, which is the extracellular vesicles that also down-modulate the host immune response. So you have different sizes that you can capture using zeta sizer. This is from the adult worm and the microfilary, and you have the you can see from the TEM analysis also showing the laminar structure of these extracellular vesicles. Interestingly, the lung sentinel cells can phagocytose such EVs, and you can see this is the trajectory of a green labeled, FITC labeled extracellular vesicles and the PE labeled or the red labeled macrophages, and you can see that these EVs are phagocytosed by macrophages over a period of time. What is interesting is you can confirm these data using confocal microscopy, and you can see that these are macrophages that have phagocytosed the EVs that have been label with a dye. What makes eosinophils an interesting cell is it has a number of organelles that are usually found in all the immune cells. You have the mitochondria, ER, the nucleus, and the Golgi bodies. But what makes it different is you have a lot of lipid bodies. You have adhesion receptors, cytokine, chemokine receptors, Fc receptors that can really activate these cells by sensing what is present in the microenvironment. And what makes them unique is such structures that are really unique to the eosinophil physiology, like the eosinophil vesicles, the secondary granule, which is the MVP, eosinophil cationic protein, eosinophil-derived neurotoxin, and the eosinophil peroxidase, that makes these cells highly active because they degranulate and release them into the microenvironment and really cause a lot of damage to the tissues. So what we did is, a couple of years back, we developed a mouse model of tropical pulmonary eosinophilia that mimics the phylar manifestation as seen in humans. So there is a sensitization phase and a challenge phase, and then you assess these mice after a gap of 10 days, and they show immunopathological assessments that are closely linked to the manifestations that you see in humans. So this is the lung histology, histopathology of these mice over a period of time, and you can see there's a continuous buildup of the leukocytic infiltrates in the lung at day 10, okay? If you see the inflammation score, it starts to increase around day 4 and day 6. That is related to the upregulation of a protein in eosinophil that is known as AIPLA2 that we have worked out in the lab. And over a period of time, we have found that actually eosinophils do not exist as single cells. Actually, they are subsets. These are actually inflammatory eosinophils and the resident eosinophils, hereafter named as IEOS and RES-EOS. So these eosinophils in these mice actually accumulate over a period of time in the bile fluid, as you can see from the data from the control mice, and as you can see, they build up to 28% by day 10 in the bile fluid. They increase in the blood also, and they increase in the lung also. And they are morphologically distinct, actually. You can see the serrated margins of these highly activated inflammatory subset and the smooth margins of the resident eosinophils. These cells actually are highly activated, as you can see from the upregulation of CD62L, CD69, and of course CD101 as compared to control EOS. The TP-IEOS upregulate all these markers. They also release a lot of ROS, increased calcium influx, and increased affecting polymerization, suggesting that these are highly activated cell subsets that can really cause damage to the lungs. So we also explored the kinetics of other cells inside the lungs over a period of time. This was a gating strategy that we used to identify migratory dendritic cells, not going into the details of that. But very close to this, just last year there was a paper in Nature Reviews that talked about the role of innate lymphoid cells, which are regulators in helminthic inflammation. So what we did is, we studied the kinetics of recruitment of these innate lymphoid cells in the lung, and as you can see, these cells, both in the lung and in the bile fluid, accumulate over a period of time in these TP mice. So you can see the increase around day 6 and then fall over a period of time. We explored these in different organs. The data from the bile fluid in the lung is shown here. Those from the spleen and peripheral blood is shown here. As you can see, the increase in numbers as compared to the control. If you see the ILC2 subset and the natural ILC2 subsets, actually they increase at day 6 and then go down in numbers. This increase in the number of both eosinophils and the innate lymphoid cells is actually correlated with increased fibrotic scores in the lungs of these mice. As you can see, the images on the left-hand side are from the control mice, and over a period of time, both the HE stain and the Mason's trichome stain, you show these increased presence of collagen in these animals, which of course contributes to the increased fibrosis score. So what we thought is, whether they can attract eosinophils, have some kind of crosstalk with ILC2 cells, or the other way around, whether ILC2 cells crosstalk with eosinophils. So interestingly, using an air pouch assay, we came to the conclusion that they did exhibit chemoattractant properties, and if you inhibit this ILC2 axis using a neutralizing antibody against IL-33 and ST2 axis, you can see that the total number of cells substantially comes down when you have the neutralizing antibody treatment, as in case of, which is shown in the case of lungs on the left, and on the right in case of the accumulation of eosinophils was also affected. So blocking IL-33 ST2 axis, of course, prevents damage to the lungs. As you can see, it leads to decreased ROS generation, decrease to which Th2 cytokines, IL-4, IL-5, and IL-3, and results in improvement of the overall lung histopathology, as is evident from the anti-IL-33 treatment, where it restores the alveoli or the lung structure to almost normal condition, and there are no signs of fibrosis. So schematically, just to sum up what I have talked about, so this is the hematopoietic stem cells that actually gives rise to different kinds of leukocytes that circulate in the blood and of course enter into tissues. You have this epithelial barrier, actually where you have these cells circulating, all of them circulating here. In case of lung, if you see, you have some constitutive levels of all the different cells type, the resident eosinophils, the inflammatory eosinophils, ILC2 cells, monocytes and neutrophils, dendritic cells, all different cells circulate in the lungs. But whenever you have an entrapment of MF or the microfilaria, which leads to the onset of TPE, some of these cells are actually recruited in response to the antigenic composition that is released by these MF. So you have recruitment of all different cell types that accumulate in the lungs. They form loose eosinophil granuloma, which is not as distinct as you see in tuberculosis. Some of these cells would migrate to the mediastinal lymph nodes and of course give rise to antiparasitic Th2 lung immunity. What also happens is, if you zoom out this area, what leads to the ILC2 activation is the following. The entrapped MF causes damage to the lung epithelium. There are release of several alarmins, among them IL-33 that I talked about today. The IL-33 has the receptor for IL-33 is ST2 that is present on only ILC2 cells and not on eosinophils. They sense them, release Th2 cytokine that leads to several processes like plasma cell activation, recruitment of eosinophils, and bronchial hyperreactivity. There are some repair processes also that are active in the lung that is guided by amphiregulin, and that leads to eosinophil activation. A lot of IgG cross-linking actually activates downstream pathway that leads to activation of eosinophil, that is JNK phosphorylation, AIPLA2, the AIPLA2 translocates and release then inflammatory intermediates and causes degranulation of these cells. So overall, it suggests that it is the ILC2 cells that act as very pivotal effectors in the management of TP and of course have a lot of crosstalk with the eosinophil subsets in the lung. So overall, the work that was discussed, the mouse model was discussed, was developed by Pankaj, who is now a faculty at NCCS. He was my first PhD student. It was further carried by Lakshmi, and the ILC stories being carried by Rahul, and the EV stories by Shweta. I thank my director and of course the former directors for bringing the flow cytometry facility to CDRI, my collaborators, financial support, and of course all the students who have been part of my lab that are listed on the right. With this, I leave you with dancing worms, part of the screening protocol that we do at screen antiviral drugs against. Thank you so much.
If there are no questions,
yeah,
I have some questions.
Yeah, sure.
For the first time after a long time, I am listening to a presentation on filariasis.
Yeah.
Because I work in RMRC Bhubaneswar, which the main objective was to control filariasis, develop methods of detection and control. And we thought that working hard, using drugs, there is no vaccine for filariasis, we have been able to control transmission of filaria parasite. But recently, the director of RMRC told me that the filariasis is back. And now you listen to this tropical pulmonary eosinophilia, poor lungs is open to the environment. It is not only getting the polluted air, but it is also getting the antigen from the parasite, infiltrating into the lungs and inducing all this. So what is your, I'm really going to come to CDRI and visit you sometime.
It would be a pleasure, sir. Most welcome.
I would definitely like to go to see your lab and meet your people.
Yeah. My pleasure.
But the main question is that how are you, like, there is no new drug. The diethyl carbamazine was there long ago, albendazole and ivermectin. Ivermectin is toxic. Albendazole is also equally toxic, but that is the only way that we could control by giving drugs to people.
Yeah.
But now, what is the strategy?
So the
Because
strategy.
are you going to focus on eosinophilia, tropical pulmonary eosinophilia, or control of IDA?
No, this is part of my fundamental basic biology work that we do, but we do a lot of screening. But as you said, you are absolutely right. So the current therapy that is used to control LF in India, and of course that is approved by the WHO the world over, is combination of all the three drugs: dithylcarbamazine, ivermectin, and albendazole. Unfortunately, all these three drugs, even when used in combination, individually they have only microfilaricidal effect. They do not actually have effect on the adult worm, just a modest about 33 to 35% effect. So the adult worms, once they are formed inside the host, they would continue to release microfilary in the host, and because of their long lifespan, 6 to 8 years in the host, they continue to release MF. These MF are actually taken up by the infected mosquito, and therefore, as a vector, they would transmit the infection to another individual. But since there are no adulticidal drugs, it is really very, very difficult to control the pathology of the disease, because that is the reason the pathology is known to be caused by adult worms. So that is one of the strategy. Over the years, there have been a lot of effort in repositioning of compounds and repurposing of drugs. And one recent therapy is to target Wolbachia that is present in this worm. It is a bacterium, but you cannot use an antibiotic to target them. But that is one of the strategies that is coming up, and a lot of groups in the US and in Germany are working on to target and position anti-Wolbachial drugs for eliminating the adult worms.
So we will discuss this, Dr. Srivastava, which is of my great interest.
Yeah, thank you so much, sir.
For a wonderful presentation.
Yeah, thank you so much.
Next speaker is Pranita P. Sarangi from Indian Institute of Technology, Roorkee. Pranita is going to talk about integrin receptors as central drivers of monocyte and macrophage cell function during inflammation. Pranita, you have 13 minutes plus 2 minutes. And
pointer isn't working.
Ladies and gentlemen, I request those who are representing their poster must reach the poster presentation area by 2 p.m. as judges has already arrived there. Thank you.
Hello everybody. First of all, I thank the organizers for giving me this opportunity to share my group's work. So, my group at IIT Roorkee, we look into various aspects of integrin and extracellular matrix signaling, and how it influences various aspects of innate immune cell function. And today I am going to talk more about macrophages. So, as we know, inflammation, it is a, what is this? It's a biological response to any infectious or non-infectious stimuli, and when it is locally contained and cells are able to clear and bring in homeostasis. Otherwise, if it is in some occasions when there is if the inflammation spreads to different systems, and it can lead to hyperactivation of innate immune cells, leading to abnormal migration, trafficking, and multi-organ failure. So, basically, cell migration or trafficking from bone marrow to blood vessel and to different site of inflammation and visceral organ is a central process in any kind of inflammatory response. Maybe it is a tiny mosquito bite or it can be as severe as a sepsis. And when cells are migrating in the blood vessel, they interact with various cell adhesion molecules like selectins and ICAMs and beta-2 integrins, and when they come out of the blood vessel, they interact with various extracellular matrix protein, which has a chance to refine and retune their immunological responses. So integrins are the heterodimeric proteins which are the receptor for extracellular matrix protein, and they are usually present in a closed conformation, but when they receive signal from the cell, from the chemokines and chemokine receptor, they come to a activated conformation, and which can then bind to ligand and cause the outside in signaling. So these receptors, they form varieties of, in combination of alpha and beta subunit, they form 24 varieties of integrins which participate under various circumstances, and leukocytes and innate immune cells, they mostly use their beta-2 group of integrins in intravascular migration, which are very well studied like LFA1 and MAC1, and whereas they are interacting with matrices, they use their beta-1 and beta-3 group of integrins. Macrophages, we all know these are like professional phagocytes. They participate in tissue homeostasis as well as in host defense, and the unique characteristics of these cells are their ability to change their flavor from inflammatory to an anti-inflammatory depending on the what kind of signals they are sensing from the environment. So my lab works in both inflammation and cancer system to look into macrophage biology, and today I am going to talk a little bit about how these cells migrate and what we can we know about how this, while migrating, their functionality is being modulated by integrin receptors. So I'll be showing data from two models of systemic inflammation. One is endotoxemia and CLP. So Sivaprasad Das, he looked into a very extensive experimentation by taking the cells from bone marrow, blood, peritoneum and lungs, and which integrin is actually mediating their trafficking and from different tissue compartments. And what he found was alpha 4, these are monocytes, and alpha 4 integrin, alpha 4 are expressed highly on monocytes in different compartments, both in CLP and endotoxemia model, with little bit variation in alpha 5. And CCR2, which is a chemokine receptor for monocyte, it also was expressed highly on alpha 4 expressing cells. When he looked into macrophages, which are F480 positive cells, especially in peritoneum, we call them the inflammatory cells which are generating from the inflammatory monocyte as small peritoneal macrophages, because large ones which are CD11B moderate and F480 high, they usually leave once the inflammation starts. So he found that these monocytes, which are like inflammatory monocyte becoming F480 cells and macrophages, they express alpha 5 integrin. So these are now we have two candidates, alpha 4 and alpha 5, and then towards the end, later in a little later time of inflammation, and uniquely in lungs, mostly in lungs, alpha V integrin was also coming up on these macrophages. So when he did little bioinformatics, and he found that alpha 4 and alpha 5 were mostly associated with SRC molecules which are like inflammatory, and then alpha V was more into TGA beta receptor. And in fact, it was the case, and when he looked into phosphorylated SRC, it was high on alpha 4 and alpha 5 expressing monocytes and macrophages, whereas the alpha V expressors had high amount of CD206, which is a molecule for anti-inflammatory macrophages and also TGF beta receptor. So from here we kind of concluded that inflammatory monocyte, when they are exiting bone marrow to blood vessel and into various tissues, they are utilizing their alpha 4 integrin, and the macrophages are during their inflammatory stage they are more of alpha 5 positive, whereas when they are entering into a anti-inflammatory stage they are upregulating their alpha V integrin. So with this knowledge we looked like in the parallel we were like looking how this integrin and other functionality can be modulated by any other approach, and at that time we are studying fibrillin 7, one protein which was initially found in tooth matrix, but no function of it was known, and we started looking at from immunoregulatory point of view. Why? Because it is expressed in different immune privileged areas like placenta, eye and even choroid, and then initially it was demonstrated that it is binding to alpha 5 beta 1 and it can inhibit angiogenesis. So interestingly what during our experimentation we found that one of the C-terminal fragment of fibrillin 7 is able to reduce the inflammatory function of monocyte and macrophages, and it has the characteristics of like it is bringing in a phenotype characteristics which is almost similar to using one alpha 5 beta 1 blocking antibody. I will come to the reason later. We did some more studies, and in fact it was able to regulate their functions negatively, and in septic model it was able to improve survival. And later on one more student Saloni, she looked into one of the smaller peptide, because peptides are more therapeutically more like we can use them more with advantage. So this peptide was able to regulate negatively the monocyte and macrophage function, and even in the cells which are isolated from the blood of septic patient, when we cultured them in presence of this FC10 peptide, there was reduction in their activation phenotype, and again it was able to administration of FC10 was able to improve survival. So coming to why it is happening, the question that we had initially asked, that what is so unique, how it is binding that it is bringing in this blocking antibody like phenotype. With structural bioinformatics that is done by Puneet, who is a biomedical engineer from IIIT Allahabad. So he found that this fibrillin 7C is binding to the hinge area. So if you remember the video that I had showed in the beginning, so they have to open, these integrins they have to open in order to get activated and initiate all the signal. But fibrillin 7C, it is binding to the hinge area and it is not allowing the integrins to get activated. And then FC10, although it is not binding to the hinge, but it is binding to the side domains on the side, and they are also needed for the activation because they are the metal ion binding site, and that's how they are able to inhibit all the inflammatory functions of monocyte and macrophages, which are mediated through the integrin signaling. So Saloni also looked into another aspect of monocyte macrophage function, that is the metabolism. And because at that time, like in few years back, this integrin signaling was correlated with metabolism in cancer model. So when we looked at macrophages and monocyte, where we are already finding that integrin signaling is associated with their different kind of functions. And recently we also published one paper about more into integrin function, and where FAC was very closely associated with macrophage polarization and differentiation. So Saloni looked into how FC10 may be regulating metabolic processes, and what she found that glycolytic pathway, which is a dominant pathway in M1 macrophages, was down regulated, which this is the phosphofructokinase and pyruvate kinase. So these were down regulated in presence of FC10, and blocking alpha 5 beta 1 kind of restores it back, and it increased the citrate synthase and succinate dehydrogenase, which are part of TCA cycle, predominantly active in M2 type of macrophages. She showed it both in monocyte blood, blood from the isolated from the human blood, and also the macrophages which is derived from the monocyte. Similarly, she also has looked into different other like a THP1 derived monocyte and macrophages, and there also she showed that FC10 by binding to integrin alpha 5 beta 1 is reducing the glycolytic cycle and helping in the activating the TCA cycle, and that's how it is bringing the cell monocyte and macrophages into a anti-inflammatory phenotype by reducing the inflammatory pathways. Glutamine is another pathway which also feeds to TCA cycle through alpha ketoglutarate, and she found that when she is culturing them like in TH1 like interferon gamma in presence of LPS, and she found that glutamate concentration, intracellular concentration is increasing, and also the glutamine receptor is upregulated in case of when the cells are cultured in presence of FC10, here also FC10, and when the blocking antibody is used, it is reducing. So basically she found that FC10, it can enhance M2 type markers like CT206 and reduce the genes which are associated with glycolysis or M1 type phenotype, and it is also promoting TCA cycle and glutamine metabolism, which can help the monocyte and macrophages to get into an anti-inflammatory phenotype. So to summarize, our data shows that inflammatory monocyte to exit from bone marrow to blood vessel, they are using their alpha 4 integrin, and then later on macrophages in the inflammatory stage they are more of a alpha 5 dependent, and when they are switching into into an anti-inflammatory phenotype, they are using their alpha V, and also it correlates with CD206 and TGA beta receptor, and fibrillin 7C and FC10, which is again a peptide derived from this C-terminal fragment, fibrillin 7C, they can bind to alpha 5 beta 1 integrin, and it can negatively regulate the inflammatory properties of monocyte and macrophages by influencing their inflammatory function as well as metabolic activities. Thank you so much again for listening, and I thank my group, my student who have worked and generated this data, and my collaborator from IIT Roorkee and AIIMS Rishikesh, and funding from different sources for undertaking this study. Thank you, and I can take some.
Thank you, Pratima. It is already late. We are at 2 o'clock now, and 2 more lectures, so I will just allow one question. If there is no question, thank you, Purnita. I'm not also asking any question. I have some questions, but I will not ask. I'll clarify it from you.
Yeah.
Thank you very much.
Yeah. Thank you again. Thank you.
Next speaker is Dr. Azam Gupta, as an associate professor in the Department of Rheumatology at AIIMS, New Delhi. Dr. Gupta, you have 13 minutes and 10 plus 2 minutes. Unfortunately, I'm sorry that we are started late, but I controlled the timing very well. I think not allowing anybody to waste your time, but lunch will be about 30 minutes from now.
At the outset, I would like to thank Dr. Ambak in particular, and then the Indian Immunology Society for giving me this opportunity to talk on a clinical topic. And I think I am only the odd man out, at least at present in the hall, as I do not see any clinician, but I'll try to prove my immunocompetency over next 13 minutes. I'll try to show some of our work. So I'm going to speak about autoantibodies in lupus, and lupus is a disease that affects ladies of childbearing age, and it can affect virtually any organ in the body. And again, if you have not noticed, the problem is that it affects ladies in their prime of age, that is childbearing age, and that is why it is important. So let's see, if we look at the celebrities, because that's how probably we recognize the disease. And probably if you do not know anyone, they are like novelists, they are journalists, they are filmmakers, they are actress, and then there are some sportsmen, and the most important one then probably you will remember is the last two, Selena Gomez and Mrs. Sunanda Pushkar, who had lupus. And if you do not remember any one of them, then probably this is the face that you'll remember, who had lupus and they have suffered it. So I'll just, with that background, I'll come to what lupus is. It's a disease that affects primarily ladies, and the most important and treatment defining the manifestation is lupus nephritis. And there is ample evidence by experiments that it is an autoantibody mediated disease. That's why B cells are important, and the latest proof is the phenomenal success of CAR T cells in treating the patients, and who have remained drug-free remission over last at least 1 to 2 years. So what are autoantibodies doing in lupus? So it's a disease that cannot be diagnosed unless we prove that patient has got an anti-nuclear antibody done by indirect immunofluorescence on Hep2 cells in at least 1:80 dilution. And this is how the patterns look. On your left you have anti-nuclear pattern, you have centromeric pattern, you can see the centromeres getting stained, and on the right hand side you have nuclei which are not stained, but you have got cytoplasm which has taken various pattern. And why it is important is because these correspond to different antibodies. Among the antibodies that I have shown on this panel, these 4 are lupus specific. They do not happen in any other disease, whereas the other 6 or 7 are lupus non-specific, but they can happen in lupus. So specific ones are antibodies to dsDNA, nucleosome, histone, and anti-Smith, and this point will come later in my presentation, so I'm just showing that. Now this elegant study published almost 2 decades back on healthy people in US who were army soldiers, collected their sera every year for last 10 years, and sera collected from 5 million soldiers. Only 130 developed lupus. Now they took their sera back and tested for these various antibodies, and you can see that antibodies start developing almost 5 years prior to actually having the disease. That means that antibodies are pathogenic and they start developing earlier than actually the disease manifestation. Now the earlier antibodies are non-specific ones, and as the patient goes closer to the diagnosis or first manifestation, you have specific antibodies appearing in the serum. So that's the first proof that this is the antibodies are pathogenic. So we have done this work in this multi-institutional cohort which was funded by DBT for 6 years, and the coordinator person was Dr. Amita Agarwal at SGPGI. And the lessons that we have generated on autoantibodies, I'm showing you that. So as I told, lupus has some specific antibodies, dsDNA, nucleosome, histone, and Smith, which produce this homogeneous pattern. Non-specific ones like Rho52, La or U1 RNP antibodies produce this kind of speckled pattern on the nucleus. And if you take, what we did is that we looked at their patterns at the baseline. So what are the patterns? If we test them in different dilutions, we saw that maximum nuclear patterns happen at 1:640 dilutions, because they are probably suppressed earlier because the antibodies are in prozone phenomena. So it may be important to test the sera in different dilutions if we want to look at the other patterns which are suppressed at earlier dilution because of the higher concentration of different antibodies. Now conventionally anti-dsDNA antibodies are considered to be a biomarker of the disease. That means we start the patient on treatment, and as the treatment progresses, patients become better, the antibody levels start going down. But what we did in our cohort is that we tested for all those specific and non-specific antibodies at baseline and after 3 years of treatment. And what we saw that all the lupus specific antibodies, that means dsDNA, histone, nucleosome, and Smith, all decrease significantly at 3 years, which is a novel finding which has not been shown earlier. So lupus-specific antibodies can prove to be a biomarker of the disease. Next, we look at a hard outcome, that is mortality. Nothing affects a patient more than death. So we looked at our patients, the cohort of almost 2,500 patients across 9 centers in India, and we took their clinical features, how they have presented, whether they had nephritis, whether they had hair affection, whether they had oral ulcers, and we looked at the antibodies. We took 25 characters and made 4 clusters of these patients. And as we make 4 clusters, we see that cluster 4 particularly doing bad as compared to other clusters in terms of survival. At 50 years, at 50 months, the survival is only 50% in these patients. So why is that so? And you see that cluster 4 has higher concentration of lupus specific antibodies as compared to lupus non-specific antibodies. Now the cluster 1, which is particularly doing better, we look at the antibodies. The lupus specific antibodies are much lesser as compared to lupus non-specific antibodies, which shows that lupus specific antibodies particularly have a poorer prognosis. Then we looked at the various features that our patients have, and we saw that our patients particularly have higher ulcers, oral ulcers, higher fever, and higher disease activity, which is because of lupus nephritis as compared to other cohorts in USA and other areas. We looked at why it should happen. We have looked at the various concentration of prevalence of antibodies, like we see dsDNA nucleosome histones, which are higher in as compared to other cohorts. Similarly, Smith antibodies also higher. Now we just discard these antibodies and concentrate on the first 3, which are dsDNA associated proteins, and the lower 6, which are RNA associated proteins. Now we test them in various combinations, and we did another cluster analysis to see how they are associated, how they have different features. And in our analysis, we saw that all dsDNA associated proteins, they clustered together. Similarly, all RNA associated protein antibodies, they clustered together. Then we looked at what clinical features they have. Cluster 1, which is dsDNA associated proteins, have higher arthritis, having higher proteinuria and lupus nephritis. Cluster 2, which has got higher RNA associated proteins, have higher alopecia, hair fall, oral ulcers, fever, etc. So clear demarcation is there among these 2 clusters, and that is only based on autoantibodies. Then obviously the next natural question was, are these all 3 antibodies are important, or the most important one, that is dsDNA antibody, is more important? We looked at the patients and we saw that dsDNA was having an association with nephritis. Then we tried to omit the co-presence of anti-nucleosome and anti-histone antibodies, and what we saw that dsDNA lost its association with nephritis. That means dsDNA alone is not sufficient. Then we reintroduced the positivity one by one for anti-nucleosome antibody. It again did not have any significant association. Either an alone anti-histone antibody, it again did not have any significant association, but only when all the 3 are present, it has a significant association with lupus nephritis, which is again a novel finding, because so far what is conceptualized, what is known is that only dsDNA is associated with lupus nephritis. The next question was whether other antibodies, RNA associated protein antibodies, also have any impact on lupus nephritis. So we divided the patients into 2 parts basically, and this study was done only on the AIMS cohort. We did not have the cohort, the other institutional cohorts. In that we had 363 patients. We divided them into 2 groups: patients who had dsDNA with RNA positive antibody, patients who had dsDNA with RNA negative antibodies. And so what we see that when RNA antibody is negative, association disappears. For this further analysis, we have collaborated with University of Tokyo cohort, and there what we saw is that the hypothesis was same, that autoantibodies affect the genome transcription and thereby the clinical phenotype or the lupus nephritis manifestation. So with these 2 as the objectives, first is to test the same association in their cohort, and then to test whether RNA associated protein antibodies affect the gene transcription. We have looked at their cohort and the transcriptomic data, which was on 28 individual immune cell types. Now, As we saw in our cohort, in Japanese cohort also, anti-RNA associated protein antibody, if it is negative, patients do not have any association with lupus nephritis, which was akin or almost similar to what we had seen. So the first hypothesis was proven. We went on to the proof test second objective, where we have looked at the immunological features of these patients and tried to associate it with the transcriptome in the presence of only anti-RNA, only anti-dsDNA, and obviously the interaction term. We have looked at the differentially expressed genes, and what we found that anti-RNA associated protein, when antibody is present, the differentially expressed genes are present across all the tested cell types. But when we looked at only dsDNA associated protein antibodies and the cell differentially expressed gene, it was present only in the B cells. And as I told in the beginning, lupus is considered to be a B cell, primarily a B cell mediated disease. Then we looked at the interaction term, and we again saw that interaction term associated differentially expressed genes are present again only in B cells, and to our surprise it was highest in the double negative B cells. Then we have looked at the interferon signaling pathway enrichment. It was important because lupus is considered to be an interferonopathy. It is basically a disease which is mediated by interferon alpha, and we saw that whether it is anti-RNA antibody positive or dsDNA antibody positive cells, the interferon alpha gene, so interferon alpha induced pathways are enriched in both of them. But when we take the interaction term, and we saw that there was enrichment of cytosolic ribosome pathways or rapamycin sensitive pathways in double negative B cells. And why is it important is that past studies have shown that double negative B cells are considerably higher in other autoimmune diseases as well as in lupus. Lupus, it is even higher in than other diseases. And what we hypothesize is that hyperactivation of mTOR happens in atypical memory B cells because of the co-positivity of both the antibodies, which induces ribosomal genes, and these are because of mTORC activation. And as we have a therapeutic proof from our clinical observation, rapamycin inhibits this differentiation of atypical memory B cells, and thereby the response comes. So all this suggests that autoantibodies in lupus are not just a marker of disease, they are also a marker of disease activity as well as prognosis. There are multiple autoantibodies in lupus, and as the number of autoantibodies increase, they are associated with more severe disease. We have lupus specific autoantibodies which are particularly markers of disease activity, and anti-dsDNA in isolation is not associated with lupus nephritis. Whenever there is an interaction of anti-RNA antibodies and anti-dsDNA antibodies, it induces particular changes in B cells, mainly double negative B cells, by enrichment of cytosolic ribosome and rapamycin sensitive genes in these cells. With that, I would like to thank my institution who has given me opportunity to do all this work, and my collaborators at AIIMS Delhi and at IIT Delhi as well as University of Tokyo. Thank you very much, and I'll be happy to take the questions.
I know everybody is hungry, but I just had one. This is an interesting presentation, very important presentation, so I will just allow one question. If no question, I would just like to ask you one question.
Sir, please.
That Dr.
Ranjan Gupta.
Is the incidence of autoimmune disorders, and particularly in women, is increasing now?
Sir, there are studies which suggest that yes, it is increasing. There could be multiple factors, but I would, with that, I would also like to tell that probably we are diagnosing them more because our tools have become better. We have better facilities to diagnose them early, so it could be that. But overall consensus is that yes, there is an increase in overall incidence of rheumatic diseases or autoimmune diseases.
That's very distressful. Thank you very much.
Thank you,
Dr.
sir.
Gupta.
Thank you.
Thank you. The last talk, just pardon with me for another 10-15 minutes, is by Mahmood Khan
Sir, I'll come
Rasool.
down. I think that will be better. Thank you.
Dr. Mahmood Khan Rasool is going to talk about leveraging interleukin-17A signaling blockade as a powerful therapeutic modality to combat rheumatoid arthritis and psoriasis. Dr. Rasool, you have 13 minutes again, plus 2 minutes, because I know everybody is waiting for lunch.
So good afternoon to one and all. It is very difficult to play as a last batsman. Whatever it is, we have to restrict the time. Here I am going to present our research findings, okay, especially on leveraging the IL-17 role in rheumatoid arthritis. All of you know that rheumatoid arthritis is a chronic inflammatory disorder characterized by the inflammation of synovial cells in the joints. Once the disease got affected, it will make you to immovable, and then even though it is not a life-threatening disorder, but it won't kill you, it will take your life. So that as a basic researchers, I request all of you to give importance to this disease so that we can prevent that immobility in our later stages of our life. Here when you are seeing this, around 4.2 million Indians suffer from this disease, and then around 1 lakh to 2 lakh Indian rupees is found to be cost for the patients per year. And then when you are seeing this pathogenesis here, even though a lot of cytokines has been contributed in the pathogenesis of RA, it has been well accepted that IL-17A plays a very important role in the pathogenesis of RA. So here we thought to check this role. Here we thought to check this role on this IL-17 on the pathogenesis of RA. Here we had seen that it has been well reported that IL-17A promotes the activation of FLS, that is the synovial cells, which is an important compound of the synovial joints. When the synovial cells get hyperactivated, it will release one protein called rankle. That rankle will go and bind to the rank in the osteoclast, which will convert that macrophage to osteoclast, will cause the release of the matrix metalloproteinase collagenase, which will cause the cartilage degradation. And then apart from that, this IL-17A is a major inflammatory cytokine which will enhance the immune cell infiltration in the synovial joints, which will maintain that inflammation. So that we thought to target this IL-17A so that it can downregulate or it can control the pathogenesis of RA. Here in our lab we have seen that IL-17A augments that osteoclast mediated bone erosion, and then we found that IL-17A controls the secretion of inflammatory mediators and the rankle mediated osteoclastogenesis by FLS in rheumatoid arthritis. And as all of you know that we are having several IL-17 inhibiting drugs, but all these drugs are not used by, it can't be used by the common person as Ahmadmi. So all these drugs is only applicable to those who are having a lot of money. So that we thought of whether we can able to find some drug, some small molecule inhibitor which can inhibit the IL-17A and then IL-17 receptor interaction. So in that search, we came across one compound called cyanidin, which was first reported by Cleveland Clinic. They found that this cyanidin, which is the source of flavonoid, anthocyanin, which is found rich in blueberries, they found to act as a competitive inhibitor against that IL-17A, so that it will compete to bind with the IL-17 receptor, and then it will block that IL-17 receptor signaling. After seeing this work, we thought why we should not try with this rheumatoid arthritis. And then here with who is working in Harvard, she investigated that cyanidin is able to prevent that IL-17 mediated hyperproliferative potential of FLS in rheumatoid arthritis. And then she found that the cyanidin able to inhibit the IL-17 cytokine signaling mediated monocyte migration and differentiation into mature osteoclast in rheumatoid arthritis animal models. And then the cyanidin is also able to restore the Th17 Treg imbalance and inhibits the T helper follicular cell differentiation. By having all these promising results, we went to a new investigation. Here we thought whether this IL-17 plays a very important role in nephrocitosis. All of you know that the nephrocitosis is a very important process, very essential one, which is necessary to clear the apoptotic bodies in our system. Usually during the inflammatory condition, lot of immune cells will get infiltrate, and then it will undergo death. Once it is, after its function is over, it has to be cleared by the phagocytic cells by the process called nephrocitosis. When that nephrocitosis get disturbed by these inflammatory cytokines like IL-17, then that apoptotic bodies, apoptotic cells which is getting accumulated, especially in the synovial joints, it will maintain the inflammation, and then it will cause, it will provoke the pathogenesis. So we thought whether the IL-17 plays a very important role in promoting this nephrocitosis. So we had this question whether IL-17 drives the accumulation of apoptotic bodies, or this the JAK-STAT pathway activation will impair the nephrocitosis via macrophage polarization, and then whether the cyanidin restores the nephrocitosis by acting as an IL-17A inhibitor, and then whether cyanidin improves the PDL1 expression so that it can terminate that T cell signaling. Okay, so this is the hypothesis what we have proposed. So when this IL-17 receptor signaling is IL-17 signaling is blocked by this small molecule inhibitor, this JAK-STAT pathway can be inhibited, so that it will promote the activation of upregulation of STAT5. The STAT5, what it will do, it will stabilize the MERTK, which will go and promote the activation of, promote the process of nephrocitosis. So with that hypothesis, we started our experiment. Here we found that, we did this experiment with, we took that synovial macrophages from the arthritic rats, and then we co-cultured this synovial macrophages with IL-17 stimulated synovial macrophages with apoptotic cells, which is that is the apoptotic T cells. And then after that, we checked that nephrocitosis. This nephrocitosis was quantified by measuring the pH rhodo positive synovial macrophages. Here, when you can see this IL-17 stimulated synovial macrophages, the nephrocitosis is found to be impaired by that. We can see that which has been evidenced by that lowest percentage of pH rhodo positive synovial macrophages. Whereas this cyanidin treated IL-17 stimulated synovial macrophages, that the pH rhodo sensitive positive synovial macrophages are found to be, the percentage found to be increased. This was further confirmed by that IL-17 receptor knockdown group, where you can see that highest percentage of pH rhodo positive synovial macrophages, which shows that this IL-17 is able to impair the nephrocitosis, whereas this cyanidin, this IL-17 inhibitor, it can able to restore the nephrocitosis by clearing this apoptotic cells. And then followed by this, this results was confirmed by this fluorescence analysis, where you can see that the cyanidin increases that percentage of pH rhodo positive synovial macrophages. And then we checked whether this IL-17, which activates that STAT3, whether it plays a role in the impairing the nephrocitosis. As we expected, this IL-17 upregulates that JAK-STAT3 activation, and then the cyanidin treatment downregulates that STAT3 activation in association with the upregulation of the STAT5. That upregulation of the STAT5, we found that the cyanidin is able to increase the percentage of pH rhodo positive synovial macrophages, as like with the STAT3 inhibitor. So we came to the conclusion that the restoration of nephrocitosis is possible through by the IL-17A inhibition, is dependent on the reciprocal activation of STAT3 and STAT5. And then we all of us, we know that this inflammation is associated with the macrophage M1 phenotype, whereas this nephrocitosis needs that macrophage anti-inflammatory M2 phenotype. Here, when we are saying this, we know that IL-17 induces the macrophage to shift to M1 phenotype, so that the nephrocitosis won't happen properly. This when we are checked with that cyanidin, the cyanidin is able to disturb that IL-17 signaling, so that it shifts that macrophage to M2 phenotype, so that when that macrophage reaches the M2 phenotype, it will efficiently it will carry out that nephrocitosis. And then we checked that ADAM17. ADAM17 is the major protein which cleaves that MERTK, the MERTK which is essential for that nephrocitosis. Whereas this cyanidin treatment, which acts as an IL-17 inhibitor, it downregulates the expression of ADAM17, and then it upregulates the MERTK, so that the nephrocitosis, that nephrocity process is happening properly. And then we checked whether this mitochondrial fission, which is mediated with the presence of DRP1, is necessary for the uptake of apoptotic cells by that the phagocytic cells, the macrophages. Here we saw that this IL-17 stimulated synovial macrophages, this DRP1 protein was found to be downregulated, so that which evidenced by that mitochondrial fission is got inhibited. whereas this cyanidin, which acts as an IL-17 inhibitor, there's a disruption of this IL-17 signaling, upregulates the DRP1 protein, where we found that the mitochondrial fission is found to be increased, which shows that this disruption of IL-17 signaling triggers that DRP1 mediated mitochondrial fission, which helps to restore the efferocytosis in synovial macrophages. So all of us, we know that increased number of Treg cells and sustained activation of PD-L1 in macrophages is essential to terminate that autoimmune response. In previous experiments, we saw that cyanidin is able to restore the Th17 Treg imbalance, but we don't know what is the mechanism. We thought whether the cyanidin is able to upregulate that PD-L1 expression in macrophages, and then we checked whether it can help in the efferocytosis. Here, as the first time we saw that the IL-17 inhibited that PD-L1 expression in the macrophages, whereas the cyanidin the treatment, it upregulates the PD-L1 expression, which indicates that that cyanidin is able to increase that Treg cell concentration, as well as it terminates the autoimmune response. Okay, this studies was further supported by that fluorescence staining and the flow cytometry analysis, where you can see that the cyanidin is able to increase that PD-L1's expression in synovial macrophages, so that it will help in the efferocytosis process. In summary, our findings demonstrate a novel pathological mechanism of IL-17A that impairs efferocytosis in synovial macrophages and aggravates the inflammation in RA. Our study highlights the cyanidin, a natural anti-IL-17A IL-17 receptor interaction inhibitor, to reinforce efferocytosis, which maintains homeostasis of the joints and stops RA disease progression. These are all scholars who worked on this work. And then this is our IL-17 journey. We are working for the past 10 years. And then recently our patent was granted for what we formulated one topical cream which can able to constituted with IL-17 inhibitor, the cyanidin, which is able to curtail that psoriasis pathogenesis. I want to thank my group and then my funding agency who is helping me to do some meaningful research. Thank you.
Thank you, Dr. Rasool. It is already late, but is there any question? Yes.
What, Professor? You are asking about safe.
Safety.
Yeah, we checked that renal toxicity also and the liver toxicity. Okay, it is found to be safe on both the organs. And it is found to be rich in blueberries. So our intention is to, instead of going with the monoclonal antibodies, which is very costly, why we should not try that natural inhibitors, which what we are taking in our diet.
So thank you, Dr. Rasool. I have a question. Is interleukin 17A anything to do with psoriasis?
Sure, Professor. IL-17A plays a very important role in the pathogenesis of psoriasis by increasing the proliferation of keratinocytes.
So blocking 17A will improve psoriatic condition?
Yeah, by increasing the proliferation of keratinocytes and the infiltration of the immune cells in that epidermal region, it increases that the psoriasis.
Thank you. Thank you very much, Dr. Rasool. The organizers are there now? Oh, yes, we are over.
Thank you all.
We extend our heartfelt thanks to our respected chairperson, Professor Santosh Kar, for their valuable time, insightful comments, and for so gracefully conducting this session. Your guidance and encouragement have truly enriched our discussions today. Now I respect our chairperson to kindly come forward and present mementos to our distinguished speaker. We will now request all of our speakers to kindly come and receive their mementos. I warmly invite our speaker, Professor Mehboob Khan Rasool, Dr. Girish S. Kiri Manjeshwar, Dr. Praful Taylor, Dr. Shravan Sehrawat, Dr. Mrigang Srivastav, Professor Pranita P. Sarangi, Dr. Ranjan Gupta. And a very humble request, please stay with us on the stage as we will proceed with the group photographs.
Whatever is there in front of me. Sorry, sorry. He can become hungry and he has gone for lunch. We'll keep it. We'll give it to him when he comes back. They gone?
I request Dr. Mrigang Srivastav to please come up on the stage if he is present here. Okay. Oh, sir, we'll proceed with that. Okay. We have completed. Sir, please for it. Sir, sir.
Oh, sorry. I will be keeping this photograph. I need this photograph. I would like to say that after such a brilliant group of people. This one photo now on each. Please. Fantastic array of presentation.
Thank you, professors. Now I request Professor Shivesh Sharma to kindly to kindly present token of appreciation to our respected chairperson, Professor Santosh Kar, as a gesture of gratitude and respect. Sir, wait.
So I have to receive also something. Yes, yes.
Like who was you scores 12 runs like that. Right.
We have now reached the scheduled lunch break. As we are running late, we request everyone to have a short lunch break. Kindly ensure that you return to your respective halls after lunch for the commencement of next round of parallel sessions. Thank you everyone. Enjoy your lunch break.
parasite into host macrophages. So this is the Leishmania, this is the macrophages, and with the macrophages, what is the pointer? Okay. So this is the macrophage, and this is the Leishmania parasite gaining access into the macrophages. And the protein which is important for attachment of these parasites into the macrophages, it is called kinetoplastid membrane protein, and roughly about 2 million copies of protein per parasite. And this protein is essentially present on the cell surface, and when we stain these parasites with the anti-KMP11 antibody, we found they are actually everywhere in the Leishmania parasite. And we cloned this protein later on and used as a vaccine candidate, and that was many years ago. And it is a kind of important vaccine candidate for Leishmania, for developing vaccine against Leishmania. Now this KMP11 protein has high affinity for the membrane cholesterol, and at a monomeric form, it is kind of, for many years we are trying to find this monomeric protein, or rather this protein, in serum of the Kala-Azar patients, but we are never successful, and it turned out that it can form oligomers, and this oligomeric protein presumably can form a pore in the macrophage, and that is the way probably it helps in the entry of the parasites into the host cells. And we also did show that this oligomeric form by AFM images, that the KMP11 forms oligomer, and that attaches to the cell surface, and that gives the access of the parasites into the host macrophages. And we found that the look for the binding affinity of this purified protein with membrane cholesterol or other cholesterol. So we took fluorescent level cholesterol, that is NBD cholesterol, and it has excitation and emission at 469 nanometer versus 537 nanometer. And as a function of cholesterol concentration, what we see that the fluorescence intensity progressively increases, indicating that it is binding with cholesterol with not that fairly, but some degree of affinity with membrane cholesterol. So then we ask the question that what actually does, after taking away cholesterol from the membrane, what happens to the macrophages? So we try to determine the membrane fluidity, that whether membrane becomes fluid or not. So therefore we measured the fluorescence anisotropy, which is a kind of measure of the membrane fluidity, and it is inversely proportional. That is, if the fluorescence anisotropy is increased, that means the fluorescence, the fluidity is decreased, and vice versa. And we took a probe that is one of the probes that is DPH, and it gets into the membrane, and therefore if we excite it, and there is excitation and emission at 350 and 420 nanometer, and DPH gets into the membrane, and if it is membrane is more fluid, it will gain access more into this bilayer membrane. And we studied that if we take these macrophages from the normal animal, normal bulb C mouse, these macrophages are kind of, they have all the processes, etc., etc. But if we take these macrophages from the infected animal, and what we see that lot of blebs and fissure, and indicating that it has membrane fluidity must have been altered. And if we infect these macrophages with the Leishmania parasite, and as a function of time, if we want to see the membrane cholesterol level, and we can see that as a function of time, the membrane cholesterol is significantly decreased. And if when we try to do some fluorescence anisotropy study with the normal versus infected macrophages, and we could see that there is a significant decrease in the fluorescence anisotropy or FA value, indicating that increase in the membrane fluidity because cholesterol has been taken away from the membrane. Then when cholesterol is taken away from the membrane, the lateral fluidity of the membrane proteins also increased. We took the one protein, that is PLC delta 1, that is coupled with GFP, and we tried to monitor the GFP expression. So we bleached the cell with some laser, and then it is bleached, and after certain period of time, this lateral, because of the lateral mobility, protein comes to that site. So this is the way one can see that we have bleached it here, then lateral mobility increased, and the GFP protein expression is restored. So that's the way we took pre-bleached, that is the normal macrophage, and we bleached it, and then after post-bleach, wanted to see that what is the status of membrane lateral mobility of the membrane protein, and we study the diffusion coefficient in the normal macrophages, infected macrophages, and if we treat the macrophages with the cholesterol, and we can see that the infected macrophages, the diffusion coefficient is significantly increased, suggesting that the lateral mobility of the membrane protein has been increased, and if we put back cholesterol into the membrane by liposomal delivery, then it is coming back to near normal, suggesting that macrophages during infection process takes up membrane cholesterol, and that causes increase in the membrane fluidity and also increase in the lateral mobility of the membrane proteins. How actually Leishmania takes up membrane cholesterol? So we studied what actually it does. So we came to one microRNA, that is microRNA 122, and indirectly promotes the HMG CoA reductase activity, and inhibiting microRNA 122, HMG CoA expression is reduced and leading to lower plasma cholesterol level, and this is the GP63, which is a surface protease, metalloprotease, and this cleaves the dicer and which prevents the maturation of pre-microRNA to microRNA 122, and therefore leading to decrease in the membrane cholesterol level. And even in the clinical scenario, even in Kala-Azar patients, we see serum cholesterol is significantly decreased as a function of parasite load in the spleen as well as in the liver. Now, then we took purified KMP11 protein and asked the question whether KMP11 alone can increase the membrane fluidity or not. So this is the macrophage, this is the fluorescence anisotropy, which is roughly about 0.3. And if we infect with the Leishmania parasite, the fluorescence anisotropy is decreased. That means the fluidity of the cell actually increased. Then LD infected macrophages, if we treat them with the liposomal cholesterol, that is putting back cholesterol into the membrane, then it is significantly increased. And then if we take, instead of parasite, if we take KMP11 protein, and then also see the fluorescence anisotropy is decreased. And if we take KMP11, cholesterol, and the macrophages, it is also significantly increased. This tells us that KMP11 protein is important for interacting with the cholesterol, and actually that takes up cholesterol from the membrane, and KMP11 actually secreted from the parasite and from oligomer, and then it really binds to the membrane and makes the pore. So another study we wanted to see that which terminal of this KMP11 protein is important. It is roughly about 92, 93 amino acid protein. So we we make all this tryptophan mutant at the position 5 and position 48, position 62 and position 89, and wanted to see that the which way it is interacting with the membrane. And we take the freight, that means you excite the donor, and if it is within the faster radii, that is the 10 nanometer, then it will stimulate the acceptor and you get a fluorescence at a different level. So that indicates that these two molecules are in the close proximity. So all this tryptophan mutation that has been done in the in this KMP11 protein And tryptophan one can excite at 295 nanometer and emission at 328 to 350 nanometer. And the liposome that was prepared using the, this was prepared using this dihydroergosterol, and which one can excite with 325 nanometer to 350 nanometer, and emission one can study at 375 nanometer. So using this kind of system, we did some Freight analysis and asked the question that how KMP11 is interacting with the membrane, and we found that the donor acceptor distance is less when this mutation is done at the position 5, and again this distance is less at the position 89. So we tend to believe that this part of the protein is somehow interacting with the membrane, and also this part is interacting with the membrane, and other parts somehow are actually floating around above the membrane. So probably this KMP11 is interacting with the membrane, and which is taking up cholesterol and ultimately responsible for increase in the membrane fluidity. So we did some functional analysis with the peptide sequence 1 to 19, the KMP11 protein. And if we treat these macrophages with Leishmania parasite alone, this is the number of intracellular parasites per 100 macrophages. But if we treat it with the KMP11 protein, and there is a significant increase of the parasite number in the macrophages, suggesting that KMP11 can act as a virulence factor for entry of the Leishmania parasite into the host cells. And also you wanted to study whether KMP11 peptide can favor the parasite entry into the host cell. So we also see that the peptide alone is capable of doing the job by increasing the membrane fluidity and allow the parasites to enter into the host cells. So what Budhaditya did actually, he prepared KMP11 knockout parasite and asked the question whether its infectivity is gone or not. So this is the wild type parasite. This is a kind of normal shape and size. But when KMP11 was removed, the morphology of the cell significantly altered, and after complementation it is restored its morphology, suggesting that the KMP11 probably important for maintaining the morphology of the parasites. And taking this wild type parasite, what are the number of cells actually, parasites are actually binding to this or interacting with the macrophages. So about 250 parasites per 100 macrophages, but this knockout parasite, it is completely essentially gone, and after complementation it is restored. And if you take one parasite which is cultured for many years, about 200 days, then also it loses its virulence, and also a parasite which has been cultured for many days, it also lost its virulence. So therefore KMP11 probably as an important protein and needed for interacting with the host cells. Now this is the kind of model that we are trying to propose, that this is the membrane and where KMP11 is interacting with the membrane and causing the membrane fluidity to increase, and so distortion of the bilayer membrane. And if we can restore this kind of thing by putting liposomal cholesterol, and this kind of macrophages or dendritic cells where cholesterol is lacking, they are defecting in the T cell activation. That is another way of looking into the problem of the defective cellular immunity in Leishmania infection. So these are kind of my students and collaborators. Rajiv joined, then Achinto Shanigrahi. Rajiv is still a graduate student. Achinto Shanigrahi now at abroad. Budhaditya Mukherjee is sitting in the audience. Moumita Bhumik must be somewhere here. And Junaid Jibal, he's a professor, assistant professor at Presidency College. And my collaborator is Krishnanando Chattopadhyay and Shubhendranath Bhattacharya. And with this and our funding agencies, CSIR, IICB, and part of the fund we have received from European Commission. With this, I stop here. Thank you very much.
Thank you, Dr. Roy. for highlighting the significance of this membrane protein 11 favors navigation in leishmania. Any questions, please? We can take 2 questions.
Okay,
Yeah.
I think, no, nobody is much excited.
Well, I have one clarification.
Yes.
If you can block this receptor, how it will be? Whether the leishmania would fail
Which
to
receptor? KMP11?
Yeah.
KMP11, when in knockout parasites, they are not infective at all. They cannot attach with the macrophages.
And they die after
They,
inoculation?
they live, but the replication is very slow.
Okay.
The membrane morphology is completely distorted.
So this again reconfirms that
Yeah.
through this receptor only the entry is there.
Yeah, this is, we tend to believe that this is another virulence factor of the Leishmania parasite and causes spore in the membrane, and that favors the entry of the parasites into the host cells.
So now may I call upon Dr. Gayatri Mukherjee from IIT Kharagpur. Dr. Gayatri Mukherjee is an associate professor, School of Medical Sciences and Technology, Indian Institute of Technology, Kharagpur. Her research focuses on autoreactive CD8 T cells, dendritic cell biology, antigen processing, and siRNA-based immune modulation. With PhD from NICD Jadavpur University and postdoctoral training at Albert Einstein's College, she investigates immune mechanism underlying autoimmunity, infection, cancer, advancing translation immunology through innovative antigen targeting strategies and interdisciplinary collaboration. Dr. Gayatri.
So the pointer, this works or no?
No, ma'am, it doesn't work that.
No? Okay.
There's a pointer in the PowerPoint which can be used.
Okay, good afternoon everyone. So first of all, I would like to take the opportunity to thank the organizers for giving me the chance to present my work here today. So today I will be talking about a novel immunoregulatory axis that we have discovered in our lab, in our research group. So this is basically two molecules that we all know of. One is PD-1, one of the big bosses of immune regulation, and the other is E-cadherin, which is very much involved in adhesion and cell migration. So E-cadherin, as you know, does homophilic interactions with other E-cadherins through there are 5 ectodomains, and they are involved in cell adhesion and cell migration. But what is lesser known is that E-cadherin can actually have heterophilic interactions between other immune molecules like KLRG1 as well as an integrin called CD103. So the interaction with CD103 leads to homing of the cells into the gut epithelium and homing of the T cells into the gut, and the interaction with KLRG1, which is expressed on NK cells mostly and some subsets of T cells, actually leads to inhibition of NK cell cytotoxicity. So the question is that arose in our minds is whether E-cadherin can interact with any other immune receptors like the big boss, the PD-1. So to answer this question, we first cloned, expressed and purified both the IgV domain of PD-1 as well as the EC1 domain of E-cadherin and did a surface plasmon resonance study. So as you can see here, so in the surface SPR study, we used both human PD1 and humans E-cadherin EC1, or mouse PD1 and mouse EC1. And we using the analyte as the EC1, we had the mouse PD1 as the ligand, and we used the EC1 as the analyte. And we found that in the SPR studies, there was a concentration dependent binding that was happening in both human as well as in mouse. So this is just one experiment that I've shown here, but we have also flipped the experiment where the PD1 is the analyte and the ligand is the E-cadherin. And this has also been done with both EC1, EC2 construct of the domains of E-cadherin. And it has given a similar KD value which is comparable to the PD1, PDL1 KD. So obviously this indicated that there is some interaction going on, but we wanted to check whether it was actually happening on cells or not. So what we did next is we wanted to make a probe out of the EC1, and to increase the avidity of the EC1 probe, we made it into a tetramer. So what you do is basically have the E-cadherin EC1 linked with an AVI tag, which is a 15 amino acid long sequence on which there is this lysine that interacts with biotin and ends up in presence of a very important component called the B-ray ligase, and it ends up adding the biotin moiety on the E-cadherin EC1. So this was then conjugated with streptavidin that was P conjugated, and what we got is a P-labeled EC1 tetramer. So using this as a probe, and we took cells from the murine spleen, the CD4 T cells. First we wanted to check whether at what time point upon activation the PD1 expression gets higher. So first we did this. So when you activate the CD4 T cells with anti-CD3 and anti-CD28, we found that the PD1 expression progressively increased over a period of 72 hours. Also, simultaneously, when we use this P-labeled E-cadherin tetramer as the probe, we found that if you look at the y-axis in the upper right quadrant, you see that there is a, I think the numbers are not very clear here, but so what we find is the higher binding, the higher percentage of cells were binding to the E-cadherin tetramer. So that actually shows that the E-cadherin tetramer is binding to something on the T cells. So obviously there was a question whether it was KLRG1. So we checked the expression of KLRG1 and it was not present on those T cells upon this particular treatment. So we could confidently say that the E-cadherin EC1 is binding to PD1 on the T cells, which are PD1 positive but KLRG1 negative. Now all is very well, but does it actually produce anything? So we went for the most obvious and easiest thing to look at, that is the IL-2 secretion, and we did a dose response curve, and we found that there was a concentration dependent significant decrease of IL-2 secretion by these CD4 T cells. So then, we were very happy that it was actually a productive engagement. So we looked at different modalities in both CD4 and CD8 T cells. First of all, we found that there was a decrease in the percentage of CD25 positive, CD69 positive activated T cells in both CD4 and CD8. Here what we did is we have the cells, we have the CD4 T cells activated with anti-CD3, anti-CD28, in presence or absence of E-cadherin EC1 or PDL1, which was kept as a positive control. So you can see here that the maroon colored bar is actually the PDL1 treated cells, which show a highly decreased activation as well. Similarly, we found that this EC1 treatment was able to decrease the proliferative ability of the CD4 and CD8 T cells, as well as the effector cytokines. IL-2 we have already seen in CD4s, it was found the same in CD8, and interferon gamma was also decreased. One important thing to note here is that although in the CD8 T cells the effect was similar, like it was a significant decrease of all the effector cytokines or even activation, it was definitely much less dramatic in comparison to the CD4 T cells. So this is one part that needs to be considered in a maybe later project. So obviously, since this is a very new interaction and nobody has reported it, so we went for a bulk RNA-seq data, RNA sequencing. So again, we sent the T cells that were activated by CD4, CD20, anti-CD3, anti-CD28, or in presence of EC1, and we found, and we did the transcriptomic analysis. And this volcanoplot shows the top 10 upregulated and top 10 downregulated genes here. The IL-2 shows up as a confirmation that this transcriptomic, at least it makes sense. So if we look at these upregulated data, so if we first started looking on these, and we found that if we look at the protein-protein interaction network, we found that there was an enrichment in genes that were related to T cell migration and leading to also T cell survival. So when we looked at these genes, so there were the most important ones that stood out were KLF2 cell, which is basically L-selectin, S1PR1, and BCL2. So a literature survey shows that KLF2 actually controls T cell trafficking by activating the L-selectin and SPR1 transcription. Importantly, we also see S1PR1 higher expression in the transcriptomic study. So and also S1PR1 leads to the T cell survival by involving BCL2. So S1PR1 is a very important molecule because it has dual role. In physiological condition, it actually inhibits Treg differentiation through the mTOR pathway, but in the tumor microenvironment, it actually leads to accumulation of regulatory T cells. So we also wanted to check whether these cells were being driven to a regulatory phenotype, and upon further investigation of the transcriptomic data, we found that FOXP3 was indeed significantly upregulated. So when we cultured the CD4 T cells in presence of the activation signal as well as PDL1 or E-cadherin, we found over a time period of a bigger time period of 96 hours, we found that both FOXP3 and TGF-beta as well as CD25. So CD25, FOXP3, CD25, TGF-beta double positive cells were upregulated, which shows that the cells were being were being harnessed to a regulatory phenotype. So, well, that was interesting, but we wanted to also check what is the binding interface, because we are talking about PD1 here, which is interacting with PDL1. So what we did, so remember that PD1 as well as E-cadherin both have their three-dimensional crystal structure solved, but we use the AlphaFold 3 platform where we use the sequences of mouse PD1 IgV domain and the E-cadherin EC1 as inputs for complex prediction. So when the top 10 predicted models were selected and then visualized using PyMol, and based on this visualization, we found that there are 4 residues in E-cadherin, which is the W2, I4, P5, and P6, which seem to be interacting with in the interface. And similarly, on the E-cadherin side, there were 4 residues, the K45, L95, V57 and I101 that were involved in the interface that was given by the AlphaFold prediction model. So what we did is we mutated these residues by site-directed mutagenesis, and we found that these mutated proteins were still functional, they were still folding properly, and they could be purified as a monodispersed peak just like the wild type, although the yield was low. So using these mutant proteins, we again ran the SPR analysis, and what we did find is that upon so these are the E-cadherin, so the ligand here is the mouse PD1, and the analyte here is the mouse E-cadherin, the wild type, as well as the 4 mutants. And what we see here is a decrease in the SPR response with the E-cadherin EC1 mutants, and simultaneously we also saw that the mutants, when in the similar assay setup where we looked at the IL-2 release, where the mutants were found to be able to reverse the inhibition, although at different levels, not at the similar level. So these 2 data together shows that these 4 residues from EC1 side must be involved in the PD1 EC1 interface. Now, what about the other one? So we had 4 residues. Actually, we have 6, but I'll just talk about the 4 first and then go to the rest of the 2. So those 4 residues that were mutated for PD-1 also were found to show less SPR response, showing that they are involved. Now, there are 2 more residues basically that we also saw. These 2 residues are I93 and E103. All these 6 residues are actually involved in the PD-1, PD-L1 binding interface as well. And importantly, what we find here is that I93A and E103A, the site-directed, the mutant proteins, actually did not show a decrease in the SPR response. So that shows that although out of these 6, these 4 were involved in the EC1, ecadherin EC1 and PD-1 interaction, these 2 which are involved in the PD-L1 were not involved in this binding interface. So we can suggest that an overlapping but dissimilar binding interface of PD-1 ecadherin is present, which is then dissimilar to the PD-1 PD-L1. Now, all is very well, but why the point is that PD-L1 is also highly expressed in various tissues. Ecadherin is very ubiquitously expressed in epithelial tissue. So what is actually happening? So there were several things that are coming in our mind. So one thing is we would like to say that this could be a very important regulation in the tumor microenvironment, especially in tumors where PD-L1 expression is low. So what can happen here is basically so there 3 things can happen actually. In the tumor, due to so in the tumor microenvironment, there are the tumor-associated macrophages, which are in the periphery of the tumor TME. And these stamps have been found to be expressing high levels of ecadherin also. So the T cells that have been activated in the draining lymph node can home into the tumor, interact with the TAMs in the tumor microenvironment, which leads to the expression of KLF2, and that drives the expression of FOXP3, also drives S1PR1. So upon FOXP3 expression, the T cells are driven to a Treg phenotype. Additionally, in the tumor microenvironment, there is a lot of ecadherin expression. But during EMT transition, this ecadherin is proteolytically degraded into different fragments of EC1, EC1, EC2, EC1, EC2, EC3, and so on and so forth. So these soluble fragments of ecadherin can go and bind to the PD-1 on the T cells and again drive them to the regulatory phenotype. Now, these interactions will lead to the, obviously lead to a pro-tumorigenic effect. Also, the KLF2, which drives S1PR1, remember S1PR1 in the tumor microenvironment actually gets through the JAK-STAT pathway and leads to the survival as well as Treg migration into tumors, further fueling the tumor microenvironment to becoming immunosuppressive. So to check that, for starters, we have, so this is an ongoing work actually. So we took the 41 breast cancer cells, which are inherently capable of EMT transition, and these are also PD-L1 low and ecadherin high. So these we grew in for different time points, and we collected the cell supernatant and did an ELISA to check the soluble ecadherin. So this basically binds to the EC1 of the ecadherin EC1 domain. And we found that upon 24 to 48 hours, there was a highly increased expression or secretion of soluble ecadherin in the supernatant. So next what we did is we co-cultured the 41 cells, which we grew into a monolayer, and then cultured them with the activated CD4 T cells, which have been activated for 72 hours, and then co-cultured in different concentrations for a period of 24 to 48 hours, looked whether they also lead to the regulatory phenotype. So what we found here is that upon co-culture of till 48 hours, we actually find a higher expression of CD25, FOXP3 as well as TGF-beta, somewhat confirming our hypothesis, also although not totally there. So what we are, ongoing investigations are going on as we speak. So there, we obviously want to look at the soluble E-cadherin enriched tumor supernatant instead of the co-culture, and we are looking at the similar phenotype change if we see that the activated cells can become regulatory there in presence or absence of PD-1, PD-L1 or E-cadherin blocking antibodies. Also, we are developing a tumor model where 41 cells were injected in the flank and they are treated with anti-PD-1 antibody as a control, and PD-L1 or E-cadherin wild type or mutants to see what's the effect of this interaction on tumor growth in vivo. And also we are investigating the role of the E-cadherin positive TAMs in modulating T cell response via this EC1, E-cadherin and PD-1 interaction. So this work, I would like to thank my collaborators, Ibbendu and Riddhiman, who have been working with us very closely on this project, and Partho, Pooja and Shaumadeep has been working for different aspects of the project. Pooja has her poster which will give more details on the biophysical interactions that she has seen, which is tomorrow, poster number 74. So I would request you to kindly visit her poster. And I also thank STARS for the funding, and also IIT Kharagpur for giving us the opportunity to do this research. So thank you, and I would love to take any questions.
Thanks, Dr. Gayatri. Any questions? We can take one. Yes, sir.
When you stimulated your T cells with ECAT, so theoretically you would expect IL-10 in this body. Yes. You did do it.
We did look at IL-10, but we didn't see a very high change there as with the IL-2.
But you are getting...
Yes, we are getting the regulatory phenotype, yes. But I mean, that was done once actually, so we will need to definitely repeat that.
Okay, thank you.
Any other question? Yes, please.
This is just out of query, although I can ask you later. What happens if we compete PD-L1, PD-1 and E-cadherin?
So actually we tried to do that competitive binding once with PD-L1 and E-cadherin. So that time we actually had not figured out whether it was it was an overlapping but not entirely similar interface. So at that one experiment that we did, we actually found that we expected that competing PD-L1 and E-cadherin would stop the E-cadherin mediated decrease, but we didn't see that. So now, upon analysis of the binding interface, it actually makes sense that there is a decrease, but not to the extent that you would expect. So that kind of agrees with our AlphaFold data as well.
Okay, thanks Dr. Gayatri for the presentation.
Thank you very much.
Thank you, Gayatri. I missed almost 3/4 of your talk. It is my bad luck. So now Dr. Arun Tribedi. Dr. Tribedi is from CDRI Lucknow. I think he is from Cancer Biology Division of CSI. These things. I think that's fine.
You can make a no, so with the person you can do it.
Okay, good afternoon, everyone. At the outset, I would like to thank chairpersons and also the organizing committee, particularly Dr. Ambak Rai, for inviting me to this exciting conference and giving me opportunity to talk about my research work. So today I shall be talking about DART32, which we identified as a novel regulator of M2 macrophage polarization, and we showed that it sustains STAT6 phosphorylation by directly binding and inhibiting tyrosine phosphatase SHP1, which is known to dephosphorylate STAT as well as JAK signaling pathways. So the genesis of this project stems from our previous paper that we published in Molecular Cancer Research in 2021, where we showed that an E3 ubiquitin ligase FBW7 inhibits myelin differentiation by negatively regulating master regulator PU1, where we showed that FBW7 ubiquitinates and degrades PU1 in a GSK3 dependent manner. During that study, what we observed, wherever we were depleting FBW7 in monocytes, these monocytes were spontaneously differentiating to become macrophage. They were adhering to the cell surface. And this was happening not only in the THP1, rather also in the myelomonocytic cell line U937, which is an AML cell line, and they can also be differentiated to the macrophages because they are monocytic lineage. So during that study, when we observed that cells were becoming macrophage-like, we observed that some of the cells were taking very elongated shape, and that gave us an idea that it is likely that when we are depleting FBW7, the monocytes that are becoming macrophages, they are taking an M2 polarized state. So to really evaluate if macrophages are becoming M2-like, what we did the CRISPR-Cas knockout of FBW7 from the THP1 cells, and we observed that these cells were becoming indeed, macrophagic-like, and they were taking very elongated morphology, and we did found that they were expressing higher levels of CD163. We confirmed the depletion knockout of the FBW7, and even at protein level there was complete abrogation of FBW7 protein. So after confirming, what we did, we make the whole cell extracts of the mock condition and the FBW7 knockout condition. We simply prepared the lysates, ran on the 1D-SDS page, stained with the Kumasi, and we tried to figure out what are the proteins that are differentially appearing in these conditions. So the bands which are boxed here, we excised them, we trypsin diested them, and we did multi-top-top analysis to identify what these proteins are. And we identified few number of proteins, and we were really interested in A5 because it was really potently induced in FBXW7 knockout condition. What we found that this protein was PPR1B, which we did not know what it was and what its function was. So we looked into the literature and we found that it is a phosphatase inhibitor, and it is known as DARP32. So I will come to it, what is DARP32, but before going that, we just validate it if it is really induced when FBXW7 is depleted, and we found that when we probe with the DARP32 antibody in the depleted condition, DARP32 was indeed induced. Now what is DARP32? Well, DARP32 stands for dopamine and cyclic AMP regulated phosphoprotein, and it actually is a phosphoprotein phosphatase regulatory subunit 1. It is a potent inhibitor of protein phosphatase 1. It is basically reported to have very elaborate function and activity in neuron cells, where it has been shown to integrate information from variety of neurotransmitters including dopamine, which produces a coordinated response involving numerous downstream physiological consequences such as calcium signaling, also the glutamate signaling, etc. And what it has already been shown in neurological cells, that this DARP32 may have a dual function. On one hand, it can act as a phosphatase inhibitor, while on the other hand, it can also act as a kinase inhibitor. And how it happens? Usually when dopamine signaling is activated, the PK gets activated, and this kinase phosphorylates DARP32 at threonine 34, and once it is phosphorylated at threonine 34, it serves as a phosphatase inhibitor. So it inhibits the activity of PP1, which is very well established in them. And it was also shown that when it is phosphorylated at threonine 75 by CDK5 here, this phosphorylated DARP32 acts as a kinase inhibitor, and it rather inhibits the activity of pKa. So it is really diverse protein, it has dual function, and the phosphorylation status of this protein dictates what kind of function it will attain. Well, it is very well established in neurological roles. However, recently there are some papers where they have shown that DARP32 also have some kind of oncogenic roles. There are very few papers though. For example, in gastric cancer, it has been shown to induce tumorigenesis, in breast cancer, drug resistance, and in one of the lung cancer paper, they showed that it is also involved in the cell migration. However, so far no role of DARP32 is reported in immune cells, particularly in the macrophages. There was nothing known, and because we identified it in the THP1 that were differentiating and becoming a macrophage-like, we thought it may have some function or activity in macrophage biology. So we proceeded to really explore if it has any role in that. But before that, we wanted to really evaluate when we were depleting FBXW7, it was getting upregulated. So is it regulated by FBW7 at all or not? So we first checked what happens if we overexpress these two. What we found, when we gave increasing amount of FBXW7, DARP32 protein levels went down. This is the ectopic expression in heterologous system HEK293T. Whereas endogenous system also we found that in raw macrophages as well as in BV2, the microglial macrophages, the endogenous DAP32 decreased when we increased the FBXW7 levels. Now, because FBXW7 is an E3 ubiquitin ligase, we surmise that obviously it will be down-regulating it via promoting its ubiquitination, leading to proteasome degradation, and we did found it indeed was happening so, because when we gave the proteasome inhibitor, the DAP32 levels restored, and this was further confirmed when we used a ligase inactive mutant of FBXW7. You know, the FBXW7 wild type protein has a delta F box domain which keeps the whole machinery intact. If you deplete that region, it loses the ligase activity. So when we overexpressed wild type, DAP32 was inhibited. When we used this ligase inactive mutant, DAP32 remained. We further showed, like, you know, when we depleted FBXW7, endogenous DAP32 was restored. And very interestingly, when we gave IL-4 alone, DAP32 was induced. And this DAP32 induction was even prominent when we depleted FBXW7. This suggested that DAP32 indeed has a role in M2 macrophage biology because IL-4 induces M2 polarization. Now, because IL-4 was independently inducing DAP32, it suggested us that indeed DAP32 may have a role in IL-4 mediated M2 polarization. So after this, what we thought we wanted to look, because we could have worked on it, that how FBXW7 is regulating DAP32, but we did not go on that path because DAP32 is phosphorylated at several sites, and also FBXW7 in order to bind with the substrate needs to bind with the phosphorylated moiety. And we did not know which kinase exactly is involved here, so we did not proceed with that. Rather, we wanted to check if DAP32 has really role in promoting macrophage differentiation and M2 polarization. If so, how it does that. So we went on to look. What we did, we differentiated different macrophage cells to different states. For example, we used murine cells, also the THP1 derived macrophages, to different states like M0, M1 and M2. And what we found that only in M2 conditions also it was always very much induced, indicating that during M2 polarized state, DAP32 levels are indeed induced. We further showed even in the bone marrow derived macrophages from the biopsy mice that DAP32 was induced, and even through microscopy we saw that there was huge expression of DAP32. This we also corroborated in human CD14 derived macrophages. Very interestingly, the monocytes, either CD14 negative cells as well as CD14 positive monocytes, do not express DAP32. As you can see here, they start to express only when they differentiate either to NAV, M1 or M2, and particularly in the M2 condition, its expression is very high. This we also confirm through microscopy. So far we established that these proteins, DAP32 particularly, is induced in M2 condition, which was not known earlier. And then we thought to really understand what happens if we overexpress DAP32 in THP-derived macrophages. Because so far what we showed, whenever we are inducing through some stimulant like IL-4, DAP32 was getting upregulated. Now we wanted to test, can DAP32 alone, even without any stimulant, can drive these cells to become M2 state or not. So we overexpressed this in the THP1, and then we probed for the different markers. As you can see, when we overexpressed mRNA of DAP32 was highly up, so was M2 marker CD163 and CD206, whereas the M1 markers TNF-alpha and CXCL10 downregulated. Alternatively, when we depleted DAP32, it inhibited even the IL-4 mediated induction of the M2 polarization. As you can see here, the markers for M2, CD206 and CD163, they were not induced even in the presence of IL-4 because we had depleted DAP32. Similar study we also did in the raw macrophages. When we overexpressed DAP32, mere overexpression was sufficient then to make M2-like state because very elongated macrophages were there. And we also confirmed through real-time PCR for the certain marker genes like CD206. In the rat macrophages, arginase 1 is a key marker, it was also up, whereas the M1 markers were downregulated. This again we did in the bone marrow derived macrophages, and we showed the similar results. And then because DAP32 was promoting M2 state, which is anti-inflammatory, we surmised that it will be inhibiting the pro-inflammatory functions of the macrophages. And we indeed showed that the phagocytic ability of the macrophages was inhibited when we overexpressed DAP32. Not only the phagocytic ability, even the ROS generation was inhibited in presence of DAP32. Even LPS-induced ROS generation was potently inhibited. Now, as you know, phospho-NF-kappa B is one of the key markers for the pro-inflammation. When we overexpress DAP32, it potently inhibited even at the basal level. And when we gave LPS and IFN gamma, the induced NF-kappa B was further inhibited, indicating that DAP32 is a potent anti-inflammatory factor. We also showed in THP1-derived macrophages and human CD14-derived macrophages that when we overexpress DAP32, it potently inhibits TNF-alpha secretion, which is a key pro-inflammatory cytokine. Whereas when we depleted it in the human CD14-derived macrophages, TNF-alpha secretion was enhanced. Now, because mere DAP32 overexpression was sufficient to drive them towards M2 state, we really wanted to know how it is doing that. And because IL-4 was inducing DAP32, which activates this JAK state signaling pathway, we focused on STAT6 and we first checked what happens to STAT6 and DAP32 during IL-4 mediated M2 polarization. We found that STAT6 was induced, so was DAP32. And in this condition, when we overexpressed DAP32 and gave IL-4, this STAT6 phosphorylation was further. It was very prominently induced. Now, here we showed that when we overexpressed DAP32, STAT6 phosphorylation was increased, whereas when we depleted DAP32 and gave IL-4, this induced phosphorylation was mitigated, suggesting that STAT6 pathway is getting regulated by DAP32. Now that DAP32, if you recall my previous slides where I showed that DAP32 when phosphorylated at threonine 34 acts as a phosphatase inhibitor, we surmise that STAT6 phosphorylation is getting regulated by inhibition of the sum of the phosphatases. So to really confirm that, we first checked what happens to the threonine phosphorylation of DAP32 in different states of polarization. And we found that in M2 state, phosphothreonine 34 was highly induced. And very interestingly, what we found when we generated a threonine 34 point mutant to alanine, which loses the phosphorylation ability, only wild type was able to induce phosphorylation, whereas mutant failed. And also the phosphate release was reduced when we overexpressed FBW7 in THP1 macrophages. Even at M0, M1, M2 state, the inorganic phosphate release was very less because this acting as a phosphatase inhibitor will inhibit phosphatases and therefore the phosphate release will be reduced. Now, which phosphate is getting inhibited by this phosphorylated DAP32? And we checked into the literature and we found that SHP1 is the tyrosine phosphatase which actually removes the phosphate group from the STAT6 and thereby it checks the phosphorylation of STAT6. Now, we went back to the literature and we found that when DAP32 inhibits PP1, it binds to it in its active site and thereby inhibits its phosphatase activity. So we thought maybe, although we could not confirm, we did some experiment to confirm, but we did not have structure, so we could not really check like this. But what we thought, it is likely that DAP32 is also binding with the SHP1 and inhibiting its phosphatase activity. So we did the immunoprecipitation, and what we did, we checked what is the interaction status of SHP1 and DAP32 in different states, and we found that although there was no interaction in M1, in M2 condition, DAP32 and SHP1 were very strongly interacting. And very interestingly, when we used a point mutant, this interaction was lost. It really strengthened the fact that the DAP32, when it is phosphorylated at threonine 34, it is binding with SHP1. Once it is binding to it, the SHP1 activity is inhibited and thereby the STAT6 remains phosphorylated. We further confirmed, we did other experiments to show that macrophages that are expressing DAP32, when they were co-cultured with tumor cells, for example 4T1 TNBC, murine TNBC cell line, there was induction of DAP32, also the M2 marker arginase 1, whereas M1 marker was reduced. This shows that tumor cells may exploit macrophages and they may induce expression of DAP32 to make them to the M2 polarized state. We further confirmed through Western blotting that when we co-cultured raw macrophages with the tumor conditioned media, there was induction of DAP32 in these cells. And further we showed that PDL1 was also induced when we overexpressed DAP32. Very interestingly, When DAP32 is expressed in macrophages, and if it is not in the tumor microenvironment condition, like there was no 41, almost there was no expression of PDL1. However, when they were placed with 41 tumor condition medium, there was robust increase in PDL1, and this increase was very high in DAP32 overexpressed condition. Alternatively, when we depleted DAP32 and placed them in the tumor condition media, PDL1 was not induced. It really says that DAP32, if it is present in macrophages and if it is placed in the tumor microenvironment, it may induce PDL1 expression. We further showed, that these THP cells did not, induce the expression of PDL1 or VGF1 when we depleted DAP32. This we further confirmed with the human CD14 positive derived macrophages. So in these macrophages we depleted DAP32 and then we co-cultured them with the tumor condition medium from a TNBC patient sample. So what we did, we isolated the single cells from the tumors, we cultured them in the media, and we took those condition medium and then we co-cultured these these human derived macrophages. And what we found, in SI control these factors were heavily induced. However, when we depleted DAP32, their expression was potently mitigated, suggesting that if DAP32 is there in the macrophages within the tumor microenvironment, it may promote tumor progression. This we further confirmed in the tumor sections from different different breast cancer primary tumor samples, and we showed that within these tumor samples, the infiltrated macrophages have very high amount of DAP32, and particularly in the HER2 positive breast cancer, it was very high. We further showed that the macrophages which express DAP32, if we co-inject those macrophages along with the tumor cells in the mice, they help the tumor progression. So what we did here, in the mice, we depleted the endogenous macrophages using cloturinate liposomes, which can be phagocytized by the macrophages, and then those macrophages are killed. So there will be depletion of the macrophages, and in these macrophages we co-implanted THP1 murine cells, either just with raw or the raw cells that were overexpressed with the DAP32. So we confirmed that there was depletion of the macrophages, and we found that the cells that were co-injected with the raw that was overexpressing DAP32, the tumor size were really high, and the ones that were only with the 41, they had little reduced sizes. Here this is the tumor volume which is given, and this is the confirmation that the raw that we co-injected, they did express DAP32. This study confirms that the macrophages which are expressing very high levels of DAP32, they actually represent tumor-associated macrophages, and they help in the tumor growth of the cancers. So if you look overall finding here, what we are showing here, that in monocytes there is little amount of DAP32. However, when they become macrophages, the DAP32 is induced, and its expression is very high in M2 polarized macrophages. In M1 macrophages, its expression is less. And this DAP32 actually at phosphorylated threonine 34 binds with SHP1 and inhibits dephosphorylation of STAT6, maintains hyperphosphorylation of JAK-STAT signaling pathway, which sustains the M2 polarized state. This finding we recently, in fact last month we published in Oncogene, and this work was actually done by Sangeeta Chaudhary. She is sitting here. She also has a poster tomorrow at poster number 60. So if you want to see more, because there are many more data that are there in the paper, but due to lack of time, I did not include them. So I will request you to go to the poster tomorrow. And my collaborators are Dr. MLV Bhatt and Dr. Anand Mishra from KGMU. And funding from the Lady Tata Memorial Trust is acknowledged. Thank you very much. And I'm really sorry for exceeding the time. I tried to keep it tight, but I'm sorry. So I'm ready to take any more questions.
Thank you, Dr. Arun.
Thank you, sir.
I think you did not take much time, only 2 minutes extra. That's permissible.
Okay.
So, one question, only one question. Yeah, please.
Hello, sir.
Hello.
So I wanted to know, so this molecule, DAP322, is, although you've shown it in tumor cells, that if the expression is high in macrophages, it goes to M2 polarization and it progresses, the tumor can progress. But is there any information about its expression in infectious diseases also? I mean, the expression level, because a lot of diseases
I agree with you. If you recall my initial slide, I showed you that the most of the function of DARP32 are known in neuron cells, and when we started to work on it, even we did not know if any other known functions are there in cancer cells. So I really don't think that it might be known in infectious disease, and I think it really wide opens the area. You can really check it, because when I looked into the literature, I did not found it to be reported in any of the immune cells. And what we are seeing here is it.
This is macrophage.
Yeah, this we reported. That's what I'm saying. When we started, it was not known. That's why we picked it, because it was very exclusively upregulated in that knockout condition when we did the mass spec, right? So it is really important protein, and because it is a phosphatase inhibitor, it may have very wider implication if you look into the infectious diseases.
Yeah, because infections like TB.
Yeah, exactly. Absolutely true, because macrophage is involved there. Very, very. I agree with. Yes, yes. Right.
Okay, thank you. If there are more questions, I think tea time.
Yeah, okay. Thank you very much.
Thank
sir.
you, thank you.
Thank you.
So next, it's my pleasure to call on that Professor Kalpana Luthra, whom I know for more than 2 decades. She is the chair of Department of Biochemistry in All India Institute of Medical Sciences, where I have been till 30th June. His work, she has persistently focused on HIV research, I think more than 2 decades. So Kalpana, for you.
Okay. Just 1 minute, let me start the timer, please.
Laser pointer होना चाहिए ना?
This is laser pointer. This is not working.
Not working. A very good afternoon to all of you. At the outset, I wish to thank Dr. Ambak, the organizers and the chairs for having me here. So today I would like to talk about our journey for more than almost 1.5 decades to work on recombinant antibodies as potential anti-HIV-1 therapeutic reagents. So all of you would be aware that with the current NACO guidelines of test and treat, the HIV infection and disease progression has slowed down. However, the antiretroviral drugs cannot affect the latent cells and also cannot cure the disease. Therefore, the need for monoclonal antibodies, and recent human clinical trials have shown promise that when used in combination with ART or the monoclonals alone in combination, they have the ability to reduce the viremia substantially for at least 2 months or so, and there is still more work going on in trying to see which is the best suitable cocktail. So as you I don't think the pointer is working. So if you can see the areas marked in red, so in India it's primarily the clade C viruses which are predominant, and in most of South Africa, while in the West it is predominantly clade B viruses, and there is a lot of genetic differences between these different clades. So therefore, it warrants that we try to characterize the circulating viruses in our population and also see which are the antibodies that elicited by the HIV infected patients in these populations, and which is best in neutralizing the viruses. Yeah, okay. Is that okay? So we see that often, although every HIV infected individual is able to elicit antibodies, thank you,
So every HIV infected individual is able to elicit antibodies. However, because this virus is highly mutating, there are a number of epitope mutations leading to viral escape. And you can see, as a result of which, the extent of genetic diversity between the circulating strains in the various populations. So the only way that we can combat this is to would be an attempt to screen the viral reservoir. So which are the currently circulating viruses in the population? So if we try to understand those viruses and what are their epitopes predominant, and then generate antibodies, such as recently an attempt is being made to make multi-specific antibodies, because especially for a HIV like virus, where the viral spikes on the envelope are at a distance. So when you have a IgG which is bivalent, it can bind to one arm. With one arm it binds to the epitope, while the other arm is not able to access the other spike on the envelope, as a result of which the antibody falls off and is not very effective. So if you have bispecific antibodies or multispecific antibodies, then they can sort of latch on to the different epitopes even if they are distant and try to neutralize the viruses. So the current goal in HIV research is towards generating antibody combinations which are most effective in curbing the viruses. We primarily focused on pediatric HIV infection, since the disease progression is very different in children as compared to adults, because most of the children get infected with HIV at birth, perinatally or at birth, and then it is seen that children have a higher CD4 counts, and therefore they are measured as CD4 percentage. More so, they have an immature immune system, and therefore if for more than 2 years, if they are not initiated on ERT, there is more than 50% mortality in children seen, and only less than 5% of the children become long-term non-progressors, that is individuals who do not progress to AIDS irrespective of not giving antiretroviral therapy. So our focus mainly lead on pediatric HIV infection. So way back in 2016, Muzammil generated primary viruses, primary isolates from a number of HIV infected children which were recruited from the pediatric OPD at AIIMS then, and we found that whatever broadly neutralizing antibodies were there, we tested a large panel against these primary isolates which are as close to the the natural virus circulating in the individual, and found that just 4 of these antibodies were able to neutralize 6 out of the 8 viruses tested, whereas all the others, for example the anti-CD8 antibodies or V1, V2 antibodies, were not very effective. So therefore the need to generate antibodies which are effective in neutralizing Indian clade C viruses which are currently circulating in our population. So using the reverse vaccinology strategy proposed by Dennis Button, wherein what we do is we screen the HIV infected children blood plasma for the presence of circulating antibodies, and then identify which amongst those plasma antibodies are neutralizing, and go on to isolate recombinant antibodies from their PBMCs. So either you can generate short fragments such as SCFVs, which are the single chain variable fragments, and one of my students, Rajesh, he is here in the audience, and currently now he is associate professor at IIT Roorkee. So he went on to generate a recombinant antibody library from the PBMCs of some such children. So what he did was, he adopted a unique strategy that earlier we tried to generate hybridoma technology antibodies, but that was very cumbersome. At the end of it, out of maybe 10 to the power 6 clones, you landed up with one effective antibody. So therefore what he did was, he took already the antigen specific B cells, and then isolated the cDNA and went on to generate the SCFV using the Phage display library and doing a number of rounds of biopanning and identified antibodies. So this unique technique, modified technique, was published in BMC Biotechnology. So my next student Sanjeev then went on to generate anti-CD8 antibodies, which are again based on the PBMC based Phage display library technology. So very recently it has been shown that ibalumab, which is a CD4 binding site antibody, can be used in HIV infected individuals who have failed second line of therapy. So therefore we wanted to see whether our library had these CD4 specific antibodies, and we did find 2 of the clones, 2B10 and 254, which were having CD4 binding site activity, where we went on to perform competitive ELISAs and also competed it with the known CD4 binding site antibody, that is VRCO1. So as we increase the concentration of the competing antibody, we found that this particular SCFV clone showed reduced binding, suggesting that it had CD4 binding site like activity. Then we went on to map the epitope specificities using wild type viruses and also mutated viruses against the N332 region, which is basically a V3 epitope, and found no dependence here, further confirming that this is a CD4 binding site antibody. So these identified SCFP clones 2B10 and 2E4 showed predominantly CD4 binding site activity. Next, we tested the neutralization potential of these two clones, and you can just see the color codes. More darker the color, the red, shows higher neutralizing activity, and we found that one of the clones showed very good breadth and was able to neutralize 38 out of the 49 viruses, which included different strains of the viruses, clades A, B, C, and so on. And we found that many of the Indian viruses were also neutralized by this particular clone. So the main advantage of these kind of bispecific antibodies, which I said SCFVs can be used to generate, is that they can bind to various epitopes, and therefore the chances of falling off and not binding to two different spikes reduces and shows better efficacy. And it was also shown in infected humanized mice that when you take a combination of 3BNC117 and PGT135, which is a combination of CD4 binding site and a V3 directed antibody, this showed synergistic enhanced in vivo protective activity in these mice. So that is the advantage of these basically these kind of bispecific antibodies. And I just mentioned to you about ibalizumab, which is already being used as a second line of therapy once the treatment fails in HIV infected individuals. So recently Sanjeev went on to identify from this cohort of children two identical pediatric twins, and we also HLA typed the different haplotypes of these children in Dr. Mitra's department. Dr. Uma Kanga helped us in HLA typing, and we found that they were genetically identical twins, and despite that both of them showed difference in their immune responses over a period of time. So when we had recruited these children and followed them up for 5 years, the guidelines then were that till the time the CD4 counts do not fall below 500 cells per microliter of blood plasma, you do not initiate them on ART, since they do not have any symptoms. So therefore we recruited this one particular very good elite neutralizer donor 330 and went on to do single B cell sorting. So we sorted for the CD19 positive cells and the IgG positive cells, and of these HIV specific IgG positive cells, that is BG505 is the HIV antigen native like trimer, and we found about 20 cells from 2 million cells of PBMCs, which were HIV specific B cells, and these were the various colors, fluorochromes, the antibodies were tagged to. So when we went on to express this particular antibody from this donor and extensively characterize the epitope mapping, which I haven't shown due to paucity of time, we found that this antibody showed very broad neutralizing activity. It was a fully human IgG because it was isolated from human B cells and showed neutralizing activity of 67% with a potency of 0.5 microgram per ml. Then we identified that this particular antibody was mainly targeted against the V3 region, and we also showed nanomolar concentrations of binding reactivity. So this was a good antibody that we took ahead for further studying, and from this same particular donor, when we found a very good neutralizing antibody, what we did was we took 3 different time points to see how there is affinity maturation of this particular antibody clone. So what we did was we did bulk B cell receptor sorting of 3 time points of the PBMCs of this particular donor, and we found that this particular, initially this parent antibody neutralized not only the contemporaneous viruses, but also the evolving viruses, as you can see. So these were the viruses circulating at the time point at which we isolated the antibody, and then we also generated the evolving viruses at a later time point. And if you see here, Ames PO1 was able to neutralize not only the contemporaneous but also the evolving viruses. So, as I told you, we did bulk B cell receptor sorting of 3 different evolving time points of the B cells, and we identified matured Ames PO1 heavy chain lineage members of the Ames PO1 family, and we went on to express the matured antibody along with the light chain of the earlier Ames PO1, since the light chain did not show much variation. So, we then got it structurally characterized by cryo-EM with our collaborator Dr. Somnath Datta at the Indian Institute of Science, Bangalore, and we found that this affinity matured antibody, if you see here on the y-axis you have the percentage ability or breadth of neutralization against the various viruses, and on the x-axis you have the neutralization potency. So we found that while having similar potency, the 44M much increased its breadth of neutralization. So, in order to see, so the current aim is that whenever you have a vaccine or an antibody, a vaccine which can elicit a protective antibody, you need to know whether the currently circulating viruses are able to be neutralized by that antibody. So therefore, the student Nitesh went on to characterize the viruses circulating in these 329 and 330 elite neutralizers. You can see the various time points, and even at the end of 5 years, their CD4 counts were above 500, that is 743 cells per microliter. So we then went on to extensively do single genome amplification of the viruses from each time point, and then generate the viruses, sequence the envelope region, and we found that in some of the regions which are really eliciting antibodies, such as the V2 region, N160 was mutated in one of the donor, that is 329, while in the other donor it remained as N, that is the asparagine. So this shows that in the 329 donor, although they are identical twins, the neutralizing activity dropped over a period of time as compared to the 330 due to the number of mutations, V1, V2 loop lengthening and evolving functional pseudoviruses in the 329. So this led to escape of the 329 donor from V2 antibodies. So overall we found that both these donors, although they had differences due to mutations in the virus, they were both able to elicit V2 and V3 directed BNAbs despite the diversity of the circulating viruses that were evolving in them. So this also gave us a clue that if there is a vaccine in a particular population with these kind of circulating viruses, then that vaccine could be effective if the viruses have shared epitopes. So this is showing you the neutralizing activity that we checked in 49 infected infants who were less than 2 years of age, and out of a cohort of 50 children, we found that 4 of them showed very elite neutralizing activity. And because in children the antibodies are less mature and the B cells are less mature, so the potency and breadth are low. So we went on to develop a new neutralization breadth potency matrix known as a neutralization score for these infants. Then it was comparable to that seen for adults and adolescents. So I am not going into this. We just found that those top 4 which you saw were elite neutralizers. In them, the elite neutralizing activity was due to the multivariant infection in them. They were not infected with a single strain of virus. So this suggested that if you give a polyvalent vaccine, maybe it will be able to elicit a more robust response. So of course, I am not going to go into detail of this. We generated a number of pseudoviruses from these infants and found that ultimately polyvalent vaccine approaches may be effective for pediatric infection. So currently in the lab, Sanjeev earlier had generated a native-like trimer. Earlier we used to use the one generated by Roger Sanders from Amsterdam, which is BG505, derived from a clade A virus. So in our lab, we have generated two native-like trimers, that is a 329 and 330. Again, structural characterization was done, and the trimers were expressed and purified and extensively characterized. And also in different forms, we immunize them in rabbits. So either we immunize as the SOSEP version or in the form of multimeric presentation in the form of nanoparticles, ferritin nanoparticles. And we found that definitely when you multimerize the immunogen, you get a higher immune response here as compared to the one which is in the SOSEP version. So this trimer is now further being characterized in the laboratory, and we found that from the 330 donor, where the neutralizing activity persisted over a period of time, we found that this particular trimer, when we expressed as a ferritin nanoparticle and also in the form of SOSEP trimers and immunized and did cryo-EM analysis of the structures, this work is still ongoing, and we are finding that there is some unique interaction with the clade C specific trimer as compared to the other trimers. So this is yet ongoing work, and here again we found that multimerization of the ferritin nanoparticle yielded a better immunogenicity as compared to its monomeric version. So,
Dr. Kalpana,
I'm just concluding in a minute.
Unpleasant duty I have.
Yes, just a minute, please. So, currently what our aim in the lab is that we are doing single B cell sorting so that we get from each single B cell a functional human monoclonal antibody, and this student Harsh Bakri is involved in single B cell receptor sorting and sequencing, and from here we identify 40 top hits which are now being expressed, purified and characterized in the lab in order to identify the best combination of antibodies that can be tested for their safety and efficacy in preclinical models such as the macaques, and this work is in collaboration with Dr. Jayanta. So he is working on adult HIV infection, and we are trying to identify these pediatric antibodies, and from these same donors also we are having the circulating viruses. So eventually the goal of this project funded by DBT is that at the end of it we have 100 pseudovirus panel well standardized from the pediatric cohort and 100 pseudoviruses from the adult cohort, so that all those antibodies that can be taken for preclinical trials will be tested whether they are effective against these circulating viruses in the patients. So the importance of the BNAbs in HIV therapy is already shown in human clinical trials, that they are able to protect although not for long period of time, and therefore different combinations are being tried out. So without these different students, PhD, MD and MSc students and all our collaborators, this work would not have been possible. Thank you.
Thank you, Dr. Kalpana. I think you put lot of data, there would be a lot of questions. Only for the paucity of time, only one question if anybody has any question. If not, then thank you.
Thank you very much. Thank you.
May I now call upon the next speaker, Mansoor Ahmed. He is Associate Professor and Head Department of Biosciences, University of Kashmir, holds a PhD in Immunology from IMTech CSIR JNU with 15 years of research and teaching experience. His research focuses on cancer metabolism, macrophage functional plasticity in tumor, microenvironments and molecular mechanisms of drug and multi-drug resistance in pathogens like mycobacteria and salmonella. Mansoor.
It's not coming.
good afternoon to all of you, and very thankful to the organizers for giving me this opportunity to present the data which we had been doing since last 3-4 years. So I will be talking here today on a different topic, that drug repurposing. Repurposing of adapalene and cryptolipine to enhance the sensitivity and overcome resistance in triple-negative breast cancer. We know that the drug repurposing is a upcoming solution to that instead of development of the new drugs, usually which is very laborious and time-taking and consumes billions of dollars. So here I would first like to introduce, this is my university where from I came. This is the lab which we had been able to develop through RUSA in last 4 years, Animal Cell and Tissue Technology Laboratory, and this is the department from which I come, and the Kashmir Valley cladded with the snow. As we know that the breast cancer has taken a center stage because it has enhanced over the period of time. If we talk about 1980s, when the breast cancer used to be at the number 3 serial, but in last year when we compared the data of 2020 to 2023 of Globocon, we found that breast cancer has taken the lead and is now the number 1 breast cancers among the women in terms of the incidence as well as the mortality, with a 24% incidence rate and almost 16% of the mortality rates. Further, when we go down, we find that this cancer burden across the globe, this is going to increase, and it has been increasing over the period of last 10 years. When the projections for the next 20 years are there, by 2040, the number of newly diagnosed breast cancers is projected to grow over by 40% to about 3 million cases every year, which is very huge. So we need to find out the things to how to tackle with this breast cancer and what are the different variants of it. So if we talk about the Indian scenario, it's not very different from the world average. Otherwise, it's worse than the world average. You see the breast cancer incidences and the breast cancer mortality. Incidence is in the blue bars and the mortality is in the red bars. You can see that in Indian scenario, it is very worst as compared to the rest of the world. Breast cancer is not killing the Indians, but it's killing the India too. You see the breast cancer burden on the health sector is almost $8 billion annually in 2021, and it's expected to grow by almost $14 billion by the 2030, which is very huge for a country like us as in India. When we talk about the breast cancer, this is a projected score. When we talk 10-20 years before, the breast cancer used to be in the older women because of hormonal change and many other things after the age of 40 or 45. That's why the breast cancer programs or screening programs in the West were started for the women above the age of 45 or 40s. And that has now gone reduced to 35 years in Western countries, but in our country, there is also a problem that if we take the data of 25 years back, we will find at present the younger women or younger girls are getting the breast cancer more as compared to that of the older ones. So this is a cause of concern and worrisome for all of us. So molecular subtypes of breast cancer is there. There are a lot of breast cancers, and one among the worst breast cancers is the triple-negative breast cancer, which has the worst prognosis, and it is metastasizing rapidly, and then its therapy is not available, the targeted therapy, because it lacks the 3 receptors like estrogen, progesterone, and HER2. So this brings us to this slide, that among the TNBC, if we talk about the average, it's 6.7% to 27% in different countries. But in this case, this triple-negative breast cancer, which is the worst, with worst prognosis and diagnosis, India is the topping all of the world with almost 43% is in the Nagpur, which is the highest city in terms of the incidence. It is followed by the Srinagar, where from I belong to, and Northeast and the Mumbai with 31%. So when we talk about the survival curves and the relapse-free survival in case of this very aggressive type of breast cancer, that is the triple-negative breast cancer, we see the overall survival as well as the relapse-free survival time is more worst in case of the TNBC groups as compared to that of the non-TNBC breast cancers. So this brings us to the question, our study we had been doing, that treatment options for the TNBC, they are very less because it lacks the targeted therapy, the receptors are not there. So the chemotherapy remains the chief systematic treatment for triple-negative breast cancer, despite multiple efforts to discover targetable therapeutic abnormalities. So why we are looking for this, why chemotherapy is used, lack of the targetable receptors, targeted therapies are ineffective about it, systematic treatment is needed. That's why there is dearth or there is urgent need to come up with the new molecules, small molecules as otherwise, or combination therapies to tackle this menace of breast cancer. So as we know that the natural compounds are as a source of drug, and overall 50% of all medications used in the clinical trials come from the natural molecules. And at least 25% of the total comes from the higher plants, and nearly 60% of cancer treatments approved by the FDA are coming from the natural sources. So we also tried to use some of the molecules which are from natural sources from different classes of drugs which are used as anti-cancer drugs. Some of them are like alkylating agents, antimetabolites, and mitotic spindle fiber inhibitors or topoisomerase inhibitors. So we are also using representative from all of these, either the 4th generation or the 5th generation drugs from all of these classes in our lab. So we had used adapalene, paclitaxel, doxorubicin, zirambun, cryptolipine, and many more. So I am here focusing on the results of the 2 of the drugs which we had worked with, adapalene and cryptolipine. So this is the preliminary data of one of the drugs that when we are treating the drug in terms of the in vitro studies, we are going for the MTT assays, then SRB assays, then the neural red assays, and then do the computation software analysis of that through Chautale method. We found that all of these drugs, adapalene and GDC, work through in a synergistic manner, which is desired. So this brings us to the studies of impact of adapalene on the proliferation, colony formation, and migration ability of the breast cancer cells. We found that all the 2 drugs, both the drugs, adapalene as well as cryptolipine, causes the inhibition in the proliferation of different types of cell lines. We had used 4 different cell lines in our lab, and it also inhibits the colony formation assay as well as the migration assay, because the cancer cells have the ability to migrate very rapidly. So once we did those assays, we find that there is inhibition in the migration assay as well. This was followed by the mechanistic elucidation of this adapalene and GDC combination, and we found that there is inhibition in the mammosphere assays also. ROS was increased, the reactive oxygen species in those cells, and we found that the apoptosis got enhanced. And finally, we found that adapalene causes CDK2 inhibition, which in turn causes cell cycle inhibition. We took this along with another drug, the adapalene, in combination with the doxorubicin, which is a normal drug used for, because it works on the principle of just enhancing the ROS. In normal cells, there is also RS, but rapidly dividing cancer cells have more ROS. So we are giving the doxorubicin, it causes the enhancement in the threshold of that and causes the death of the cells. So when we used with our molecule, that is adapalene, we found at all the levels from mammosphere assay formation to this colony formation assay or migration assay, apoptotic levels as well as the protein levels, we find there is increased expression of the ERK1 and ERK2 mechanisms, which causes the death of the cells. We then, this is the mechanistic elucidation of that adapalene, doxorubicin adapalene, that it causes ERK1, ERK2 upon treatment with these. So this is some of the papers what we published out of this study. One was in the Frontiers in Pharmacology and Oncology, and then another was in the results in the chemistry. Next, we checked that there is another molecule, which is the P53. Usually when it is not mutated, it is in the cells in normal phase, it causes the, there is mutation of this protein P53 in most of the cancers. And in TNBC, almost 90% of the TNBC cases are having P53 mutated. So we took this, worked against this protein by using our drug cryptolipine and adapalene, and we found that we know that this is the mechanism of P53, how it works through endogenous stress and exogenous stresses as well. We took this and downloaded some of the data sets from the TCGA and other portals and find that 90% of the TNBC patients have an overall enhanced expression of this protein P53, mutated P53. Then we further checked through all other means and found that there are major missense mutations in the TNBC in those data sets, and we found that there are almost 7 hotspot mutations in that. Next, we checked that whether the, we docked the wild type with the mutant P53 protein with our drug, that is the cryptolipine. Protein, P53 protein and drug, both wild type and mutant, are shown in this figure. And we found that on checking, we find wild type and mutant P53 shows comparable doc scores in both of them. The next we checked with the drug DNA as well as the wild type and mutant P53 proteins. And we found that cryptolipine, when the cells were treated with the cryptolipine, that showed enhancement in the DNA binding ability of mutant P53 as it enhanced the doc scores computationally. We checked through this with MD simulation, and then we looked for the pro-apoptotic molecules as well as the anti-apoptotic molecules on and their expression in terms of treatment with cryptolipine and without the treatment of cryptolipine. We found that there is significant enhancement in the pro-apoptotic molecules like BAX and PUMA, while as the anti-apoptotic molecule there was downregulation of BCL2 expression. This way, further comparison of the expression levels of the P53 and P21 on treatment with the FDA-approved APR2, we found that APR is showing no change in expression levels of the P53 and P21, while as the cryptolipine is enhancing both the expression and the activity of the P53, and we asked the question why it is so. Hence, we went to do the QRT-PCR, and we did the primers were designed and their sequences. This is, then we did the DNA pull down assay, and we found that there is enhancement upon the increase in the concentration of the P53, which shows that CRP, that is the cryptolipine drug, enhances the DNA binding ability of the mutated P53. And we use two cell lines in which the one is MDMB231, which is having the mutated P53, while as the other cell line which is having the MCF7, which is having non-mutated P53. So this is the overall scenario of this, and conclusion of the study is that it induces the cell cycle arrest, promotes apoptosis and inhibits the ROS and promotes MOMP, while as inhibits the migration, colony formation as well as the proliferation of the cells. We published these two studies in three different papers. One was in the Biomedicine and Pharmacology, another was in the Frontiers in Pharmacology, and then also one in the Discover Oncology. So we also published some of the related research with this, and also published a book with the Elsevier CRC Press, P53 in breast cancer. We had filed 2 US patents for these 2 molecules, adapalene and their analogs. This I showed the data of just these 2 drugs, but we had created along with our team from the chemistry analogs of these adapalene and cryptolipine, which we had submitted and claimed in these 2 patents. Next, I will show you a wrap of this repurposing paclitaxel, the resistant cancer cell lines, because the problem in the drug repurposing is one that if you have a patient who is resistant, one of the standard therapy is the paclitaxel. So these patients are showing resistance to paclitaxel. So what we did in our lab, we developed the paclitaxel resistant cell lines as well as the cross resistant cell lines. And we confirmed that these cell lines are there. They are showing the 2 markers of P53, CD44 and BCL2. There is enhanced expression of these 3 these anti-apoptotic markers. CD44 is also a memory marker, but all of the expression in these 3 in these resistant cell lines was enhanced. That in that way we also did the sequencing of that, and we found that these resistant cell lines are showing this enhancement in the anti-apoptotic molecules. So when we treated these cells with our drug of interest here, that's with the retinoid adapalene, we found that this is the first one is the parental cell line, another one is the resistant cell line, and third is with the cells which are treated with the adapalene drug. And we found that CD4 expression is getting diminished on treatment with the adapalene. And this is the DAPI staining in the second one, and third one is the co-localized or the merged staining. So we found that there is inhibition in the CD44 expression on treatment with the adapalene as a drug, as a repurposed drug.
Mansur, can you conclude? So
this is the overview of the study. Adapalene reverses the drug resistance in the triple-negative breast cancer. Conclusion from this is some of the papers which we published from this study in the last 2 years. These are some of the publications what we had been doing in our lab since last 2-3 years. This is the international conference which we organized last year, and this year we are organizing on 18th to 22nd to 24th of December, the second international conference on breast cancer in the University of Kashmir. So anybody who is most welcome to come out there. Thank you. I would be happy to take any questions.
Thank you, Manzoor. Any question? One question can be permitted. If not, then I have one clarification.
Yes.
You showed very striking difference in some of the places about the breast cancer. You showed very high percentage, 43% of them means whatever the issue was there in Nagpur. Any
Yes.
specific reason?
The specific reasons, there are lifestyle changes are there, the genetic variations are there. Then the specific reasons had not been attributed. It's common vivid reasons like lifestyle change and dietary mode and other things. There are no specific reasons.
Okay, thank you so much.
On the behalf of the organizing committee of IMMUNOCON 2025, I extend our sincere gratitude to our respected chairpersons, Professor Dipendra Kumar Mitra and Dr. Shripad Patil, for their valuable time, guidance, and for conducting this session with such grace and depth. As a token of appreciation, we would now like to present mementos to our distinguished speakers. I request our chairperson, Professor Dipendra Kumar Mitra and Dr. Shripad Patil, to kindly come forward and present memento to our esteemed speakers. All the speakers are requested to please kindly come forward on the stage. Dr. Mazur Ahmed Mir, Professor Syamal Roy, Dr. Gayatri Mukherjee, Dr. Arun Trivedi, Dr. Kalpana Luthra, and Dr. Masood Ahmed Mir. I request everyone to please be on the stage. Sir, excuse me, sir. Sir, I request you please be on the stage for the group photograph. Now I request Dr. Ambak Rai to please kindly present memento to our respected chairperson, Professor Dipendra Kumar Mitra and Dr. Shripad Patil as a token of heartfelt gratitude. Sir, after that, a group photograph will be there. Now I request everyone to please be there on the stage for a group photograph. Thank you, everyone.
Unpublished data from clinical trials. I've been told by the sponsor that we cannot share. Okay. There are some regulatory. Yeah, we have. Please do so. So if you need any connector, you tell me. I have everything. Then you know how to switch it on.
With, with that, we come to the formal conclusion of our session, cell signaling and checkpoint inhibitors. We thank all our chairpersons, speakers and delegates for their active participation and valuable contribution. We will now break for tea and the poster session will also get resumed. Kindly reassemble here in the MP hall for the next round of talks following the tea break and the poster session. Thank you for all your participation. Please enjoy.
लेके आओ कभी गेस्ट हाउस है वहाँ पर जैसा रुकता हूँ मेरे को बताना बहुत अच्छा है आपको
एक बार इसको बोलो भाई पे नकुल लहरा चाय नकुल लहरा बोलो यार नकुल लहरा चाय
going on in the MP hall itself. I request everyone to kindly assemble back to MP hall.
So may I start? I will start both as chairperson, ad hoc chairperson, as well as speaker. So I think if there is no objection, then I can start, because they are in paucity of time. It is already late. So I am D.K. Mitra. I used to work in All India Institute of Medical Sciences. I was now since June 30th, I don't work anymore, at least in the field of medicine. So I took my liberty to change the title, but essence is basically the same. So host immunity, how we can use that, how we can restore, how we can modulate in the setting of a human disease, tuberculosis. So it starts with the two basic tenets. One is that we know the TB immune response is very much present among patients. Basically, these are the T cells. Antibody cannot go inside the cells, macrophage. Therefore, T cells, they play a very significant role. And those T cells, either they polarized T cells, they either stimulate the macrophage to contain, to control the proliferation of the Mycobacterium tuberculosis, and therefore keep them under tight check, which we define as latency. And if there is a failure, then TB develops. And if that failure or immune suppression or immune deficit, specifically T cells, antigen specific T cell deficit is very severe, then we get, or relatively less severe, we get kind of disease which are localized disease. We get a good granuloma formation, which is nothing but a tissue reaction or immune tissue reaction to wall off the site of infection, so that infection cannot spread to the other parts of the body, which will be fatal for the patient. If that magnitude of immune response fails to develop and there is more immune suppression or immune deficit, then you don't have those efficient granuloma formation like the walling off reaction. So therefore macrophages which are infected, inside those infected macrophage, lots of proliferation, and at one time point they undergo necrosis, and necrosis means spread of bacilli to the bystander macrophages in the lung. That's the way disease spreads, and even hematogenous spread of the disease occurs, which we call miliary tuberculosis. And we showed that if it is the spectrum of T cell immune response against Mycobacterium tuberculosis which dictates or determines what form of tuberculosis a person after getting the active disease will develop, either a localized pleural effusion, which I had in my medical school days, or a disseminated disease, which is like disseminated tuberculosis. And this is proven by very many facts, because immunosuppression, whenever there is immunosuppression like post-transplant immunosuppressive therapies and all, the endogenous tuberculosis is reactivated. So therefore first tenet is TB cannot be removed in a sterilizing manner. Always it will be in one of your macrophages. Issue is that if you can control that with good deal of immune response, specifically cellular immune response, you don't develop active disease. And if you develop the disease, that means there is immune suppression. And it also suggests that immune containment by our in vivo immune system, T cell macrophage interaction, it is possible to contain the infection. If so, then question was, we actually started with this tenet. If so, then why not we can try to understand the human immune response among patients, because there is a lot of, we learned a lot from mice immunology of tuberculosis, but there is a fundamental host parasite interaction differences between mice and human. In mice, you infect with tuberculosis, almost 100% of mice, they will eventually develop active tuberculosis. So there is no latency. Latency is absent virtually. But on the other hand, all of us, most of us, at least in this part of the world, almost 70 to 80% of us, we have been inflicted with TB infection at one time point in our childhood, most possibly. but still we contend that we don't develop the disease. Only 1 to 2% of those infected PPD positive patients, they develop active tuberculosis, even though they harbor viable live bacilli in their body somewhere in some corner. So this again suggests that immune containment, which can restrict the development of active disease, is very much possible. So on these two questions, we tried to understand the immune response in human. We learned a lot from mice, but it is good to learn from human. So what we found, actually I will not present my 23 years data in 25 minutes. So what I will do is that I will focus very less on data or results in concreto, but I will try to focus on conclusions that we made in abstracto. So what we did is that we tried to understand the synergism between the drug therapy and immune response, drug-immune interface, that I learned from Dr. Shamul Roy long back. So we tried to look at that while we treat the patients, how the immune function, how there is gain of immune function. We know that there is suppression of immune response, and we have tried to dissect out the mechanisms active for this operational suppression in human patients. So what we found is that if we treat by chemotherapy, there is gain of containing immune response, which are primarily signatured by gamma interferon, and we also believe that TNF plays important role, because TNF actually helps those gamma interferon macrophage stimulating T cells to be recruited at the site of the real disease pathology. So that's the reason we believed, and now many people believe, now it is more or less accepted, that polyfunctional T cells play a very important role. So we showed that along the treatment course, and this is, I think, perhaps for the first time we showed that while treating, you get the gain of immune function and reduction of the regulatory T cells, which are antigen specific in our cases in human patients, and there is reduction of IL-10, TGF-beta, and there is gain of immune function, this containing inflammatory immune functions. So these are some of the representative, because I could not show you everything. So what we showed that what form of disease in the first, this one, what form of disease will be developed, either localized or content, that tightly depends on what is the magnitude of effector immune response in terms of T cells. We also showed that the more suppressive environment is there, if there is more suppression, there is disseminated tuberculosis, and we showed that those suppression at the local site is mediated by FOXP3, and later on we showed that those are very dominantly mediated by set of immune checkpoint inhibitors, primarily PD-1, and later we showed that TGIT and other molecules as well. And that suppression is more at the pathologic site when we studied the bronchoalveolar lavage derived T cell from the patients and compared them with the peripheral blood, because local gain of immune function was much more higher magnitude than the peripheral blood. And these are some basic slides, basically, where we show that if we block the PD-1 axis, especially on the FOXP3 positive enriched, I would not say sorted, but it is heavily enriched Treg cells, we can get gain of immune function, at least one immune signature function, that is a gamma interferon. And that is more so in the T cells derived from the bronchoalveolar lavage of the human patients. So suggesting that in the actual pathologic site, suppression is more, suggesting that there are good cells, Th1-like or polyfunctional T cells, they are going, nature is trying, body is trying to send them to restrict the disease, but those cells are actually functionally incapacitated by other set of lymphocytes, which are Treg cells. We have shown that tremendous enrichment of Treg cells in miliary tuberculosis, and they are actually suppressing the good cells. So it is a representational balance of good cells and bad cells at the site of the actual pathology in human patients. And we also dissected out the polyfunctional T cells, and indeed polyfunctional T cells are grossly compromised at the local site as well as in the peripheral blood of miliary tuberculosis patients. That's also we have found and reported. And if you see that when we block the PD-1 molecule from the tissue-derived, means lung-derived samples, we get maximum gain of function or gain of effector function with the double positive cells. And we also showed that these double positive cells, that gamma interferon and TNF positive cells, they express maximum density of FOXP3 PD-1 on their surface, and that clarifies why there is majorly compromised effector T cell functions in the lung of miliary tuberculosis. So we did some in vitro experiment to find out under this different cytokine milieu what kind of tissue response or tissue outcome we get in terms of apoptosis versus necrosis of the macrophage. Because if the infected macrophages undergo apoptosis, that is relatively better because you don't get dissemination of the bacteria, so bacteria can be contained by the in vivo immune system, whatever is left, effector and functionally active immune cells. On the other hand, if there are lots of if there is dominant necrosis, naturally they will spread the bacilli and they will infect freshly the bystander, the alveolar macrophages, and that's the way infection cycle goes on and infection disseminates and even through the blood. So what we found is that with the TNF, definitely there will be lot of necrosis. With gamma interferon, lot of apoptosis, but with the mixture, it is kind of balanced. Not that necrosis is completely gone, but that's what actually you need in the in vivo patient scenario, because you need also the TNF alpha to recruit these good cells, gamma interferon producing cells. So we believe that this kind of, and we did some titration experiment from human patients basically with different doses of recombinant cytokine combinations. With a spectrum of this dual cytokine range, you get actually gain of apoptotic death of the alveolar macrophages. So that is quite evident from this in vitro MDM model, that when you are blocking both the cytokines together, you get maximum reduction of the colony formation. So that suggests that polyfunctional T cells gives right milieu to contain the infection more efficiently, so that disease can be restricted at the least. And when that combines with the chemotherapy, then you get better clearance and more efficient clearance and quicker clearance as well. So these are some experiments. Here we show that this different cytokine milieu. Basically, if you block with the PD-1, naturally the maximum polyfunctional environment you are gaining. So there is more apoptosis and less necrosis, and you get, when you are adding both of these together, you can reverse those gain, suggesting that actually the polyfunctional environment plays a critical role. Of course, it is a spectrum, so that plays critical role how the bacilli will be contained, whether disease will be localized or whether there will be elimination of the disease process. So we wanted to go into the clinical trial with the PD-1 because PD-1 is used in human patients. It is just repurposing, so we don't have to do phase 1 and phase 2 trial. But we wanted to be cautious, so we killed some mice before taking a risky job. So what we did is that we treated the mice with chemotherapy, and then we added PD-1 at different anti-PD-1 PD-1 blockade. And what we found is that this is very gross, but what you found that indeed there is additive effect. When you treat with ATT, anti-tuberculosis therapy, and PD-1, your lung becomes almost near normal. These are my study. I mostly study human in my life, but this is the first time I studied the mice study, and it supported our thinking that probably there will be synergism and it can be exploited to use the host immune response, whatever is left in vivo, for the better clearance of the bacilli. That means the therapy. And these are the lung, these things we show that with ATT and PD1, CFU is low, gain of immune function and so on. So I will skip this one, but one vaccine we tried to boost therapeutically the immune response in TB infected mice, and what I find is that that boosting is very efficient, or it is augmented if we can simultaneously block the already suppressive element, which is PD1. And it sounds very reasonable and very simple, because if there is active suppression mechanism operational in TB patients, so it will be always good to suppress the suppressor element, so that you get adjuvant effect of immune restoration along with the drug, so that you get better and earlier bacillary clearance, which should be the goal of any therapeutic regimen development. So this is a paper that helped us because they showed that, they nicely showed that there is a lag time. By the time you get the gain of immune function, when you treat the patient, the bacillary, in drug resistant cases, the bacillary load actually increases, so that immune clearance cannot catch up with the bacillary load. So we had biased thinking that we can, based on definitely some experimental evidence, very strong evidence, that we can rescue the suppressive element which are operational in human patients, and it can give us a synergistic impact in bacillary clearance. And we went further, we thought it can prevent the relapse. So it is likely to potentiate the therapy as well as possibly the therapeutic vaccination, which we tried later on. So this, I will avoid this. This is another piece of work. Here we show that immune milieu actually controls the expression of the drug pump, and that makes bacteria more sensitive to lower doses of the drug. So this is another story, in fact. So based on that, we proposed two proposals. One is the therapeutic vaccine with Ruti vaccine along with the immunotherapy, and we were very fortunate to get more than sufficient fund from Horizon 2020 program. This is the first program we got, I think, in India. That year actually, this is the only trial or project it was approved by Horizon 2020. And we wanted to do these things, but there were some regulatory issues, so we could not do this part so efficiently. Now Ruti trial is finished, so we are just looking at when we find some degree of clearance, early clearance, and my presumptively biased thinking is that had we been able to add Ruti, adjunct PD1 based immunotherapy, probably that clearance would have been more drastic. But we were supported for another clinical trial of limited small scale phase 3 clinical trial with PD1 therapy along with the drug in human patients, no therapeutic vaccinations. So there we did lots of analysis and we found much better clinical parameter improvement in terms of body weight, fever, compared to the therapeutic regimen, including the bedaquiline. And that was very stimulating. I will show only one piece of data because this is still not published. I cannot show every very detailed data. It is inhibited by the regulatory agencies. But what we found, one interesting thing is that in drug sensitive cases, how much you can increase with therapy within 8 weeks you get almost, if therapy is taken properly, within 8 weeks you get around 87% sputum conversion. You can increase 1 or 2, but problem is with multi-drug resistant MDR. So there we found is that generally we found week 4, if you see the nivolumab in 0.5 milligram per kg of the body weight, there is a big difference. I can tell you the figure verbally. So at 8 weeks of time point, generally with therapy, standard therapy, 40 to 51%, that is the global meta-analysis data, they get sputum negativity. That means each patient, 50% of the patients are still spreading their viable bacilli. And it is estimated epidemiologically that each single patient of MDR cases, they generate at least 6 to 7 more patients during their positivity time. Therefore, if we can reduce the positivity time, if you can do the early conversion, actually we save a lot of new cases development. So that's what is suggested from this slide. And WCAT, it is much more pronounced. Basically, we followed these patients for almost 2 years, and what we observed is that if with the standard therapy, around 70% patients, 61 to 70% patients of MDR cases, their sputum become negative. But with our adjunct immunotherapy along with the bedaquiline based chemotherapy, it goes up to 91 or 92%. So that is a significant. That means 20 more patients with the standard therapy, they are still spreading the infection. So 20 into 7, means 140 patients you are actually preventing. So this is an intervention, I think, which can augment the clearance time, number one, and by doing that, we are actually able to influence the epidemiology of the disease transmission, especially the MDR. And I think that's very crucial. Even though it is a limited trial, we were allowed only in 60 patients, not more, but we got very good set of data. We are now writing the manuscript. And this is from the patients, in vivo PD-1 treated patients. If we take, this is the lymphocytes, and we do this tricky experiment of autologous MDM, then infecting them, and then co-mix those primed in vivo therapy-based primed, immunotherapy-based primed T cells. What we find is that with nivolumab primed T cells, actually clearance is much more evident, much more. So therefore, PD-1 blocking can help us, especially in bedaquiline. It's a very toxic drug, especially cardiotoxicity. We encountered so many cardiotoxic events. Fortunately, we had only one death in this clinical trial. So therefore, and bedaquiline has lots of side effects as well, in terms of clinical presentation, clinical recovery. So with immunotherapy, actually you don't get those things, and there are reasons for that. I'm not going into that. So with that, I think this is the piece of, this is our 20 years experiments. We developed some intervention which may not be very novel, but actually it shows a very good impact in human patients. I think that's what is the, I think, positive message I will try to convey to all of you. And we have been in this business for about 23 years. These are all work. So this present study, which clinical trial was very efficiently run by a member of Indian Army, Colonel Parikshit Sannal. And this is the work of all these people, Prabhat and all these. She did on some geriatric TV. This is Colonel Sannal. She actually performed, he actually, it was his thesis actually, PhD thesis. So it was excellent, and I am glad that I got some very time-bound, goal-directed person who takes reasonable task and performs that in time-bound manner. So I am indebted to my collaborator, Professor Anant Mohan Guleria and Mandira. And we are actually indebted for, we got a lot of fund from these agencies. They were very generous, and thank you all for your patient hearing.
Thank you very much, Dr. Mitra, for a very, very informative talk. If we have quick 1 or 2 questions we can take before we go on to the next speaker. Yes, please.
Yes. I cannot hear you.
So the cost, economics-wise, did you get questions like, is it viable in a country like India to, introduce these checkpoint inhibitors as a supportive therapy? Is it?
So you are
how is the feasibility?
Yeah, you are a medical doctor?
No.
Okay. So very relevant and right question. Each vial of PD-1 costs 37,000 rupees. So it is very expensive. Agreed. In fact, that was the question Randeep asked me. Mitra, ye kya hoga isme? But this is a proof of concept. We were fortunate that we got the fund. And number 2 is that we tried to reduce the cost basically. there are a lot of biological aspect in that, because with PD-1 therapy, you get a lot of immunitises, dermatitis, pancreatitis, pneumonitis, and so on, especially after 3 months. So we tried to be very clever, to be very balanced, so that at the initial phase, when you are treating, so that means your suppressed immune system is coming gradually gaining its function. It is in the recovery phase. So during that time, if we can fasten that recovery, probably that will help. So in human, actually, it is 3 to 5 mg per kg of the body weight is given in cancer, and that is very frequently until you see the clinical remission. So we have tried different doses. So we optimized 0.5 milligram, 10 times less, and I'm glad that we did that. We didn't get any complications, life-threatening or major complications. And that is a very valid question. But if I think this may be useful still with the government support program for MDR cases or XDR cases. And thirdly, understanding that actually Professor C.P. Thakur, who has been my mentor, who brought me actually to AIIMS long back. So we started looking at the plant-derived product for their immunomodulatory things. So I collaborated with Pulok from Jadavpur University. Incidentally or serendipitously, we found one single molecule fraction that is actually inhibiting, that is actually causing gain of immune function some way as from the TB patients. And we later, I don't understand much of finer crystallography and all those things. I think some people, some of my good biologist friends, they suggested me to collaborate. I collaborated with CDRI. I had some friends. So they actually did these docking studies and those. They found that is actually blocking the that is sitting on the same residues on the PDL1, so where PD1 binds. So if we can take that forward, or I cannot take that forward at this age, but if somebody takes that forward, I think Glenmark was very keen to take that up. So that will be, as opposed to 37,000, it will be per shot. I think maybe
Dr. Mitra, I think there's
700.
one more quick question we can take if we can.
So I don't have time, as you said,
yeah.
right? Yeah, please, go ahead.
Dr. Arora wishes to, if it is urgent, otherwise we can discuss over the evening.
It is not urgent, Sunil.
I would like to make a comment here.
Yeah.
So it's always pleasure.
But I want to make another comment. His comment will be always biased in my favor. I know that, because we are very good friends. Carry on. I give you freedom now.
Okay. But still, I will say that it's always a pleasure to listen to this talk, on human tuberculosis and using all that. What I want to say is that you have shown very good data on PD-1 and blockage and everything. But even in cancer, where the anti-PD-1 therapy was first brought with Nobel Prize and everything, now that the questions are rising that there are multiple checkpoint inhibitors which get expressed, and single therapy may not work.
Yes, Sunil. So may I stop you now? Yeah. I take that privilege.
So, Dr. Mitra, thank you very much.
No, I have to answer this. No, you cannot deprive me from this. In fact, that's the right question he asked. 2 of my graduate students, they finished their PhD with this multiple TGIT and other molecules, LAG3. So that data, indeed, it is true. TGIT and PD-1, they are the hierarchic molecule in this suppressive business, at least in human tuberculosis. That's what we found. Yeah. So it may be a combination therapy as well. Agreed, fully agreed.
Thank you. Thank you very much, and we can continue this interesting discussion the evening. So we go on to our next speaker, Dr. Samarjit Das. He's an assistant professor in the Department of Anesthesiology and Critical Care Medicine at Johns Hopkins University, USA. And his research mainly focuses on mitochondrial regulation, non-coding RNA signaling, and metabolic communications in cardiovascular diseases. Further, he also works on understanding how microRNA and these mitochondrial pathways influence cardiac injury, repair, and regeneration integrating molecular biology and stem cell-based approaches to develop novel therapeutic strategies. Supported by competitive research grants, including from Maryland Stem Cell Research Fund, his lab collaborates widely to advance translational research, bridging mitochondrial biology and cardiovascular medicine. Dr. Samarjit, please. So you have 25 minutes and I would request you to restrict your time, please. Thank you.
Thank you, Madam Chair, for the nice introduction, and thank you everybody for listening, as well as I would like to thank the organizers, especially Dr. Ambak Rai, Dr. Mitra, to bring me here. And today I'm going to talk about nothing about mitochondria, what you heard, but I'm going to talk about social media. And that's why my job here is very, very easy, because everybody in this room knows about social media, especially my friends at the back. They are doing it right now. So every time I talk about any disease, molecule, whatever, just think about your knowledge of social media and how would you react to whatever I'm showing. So remember that. Keep using social media. And so here are the social media platform. If I'm missing your favorite one, I apologize. These are my knowledge. This is my disclosure. Today I'm going to talk mostly about some of the discoveries we made in Hope Diagnostic. However, my disclosure today mainly will be, your thing is popping up. Mainly would be, this is my first talk after getting moksha this morning. So thank you, Prayagraj. So today, hopefully, I could convince you a new dimension in therapeutic world and biomarker world, which is extracellular vesicle, or EVs. And hopefully, at the end of my talk, you will go back and figure it out. That's maybe the social media our human body uses every time. So the server space, that's what Facebook, Instagram, Reddit, they use a server space. They rent it out from AWS or wherever. But in our body, they comes in 3 flavors. The bigger size is called apoptotic body. And apoptotic bodies are mainly released when a cell tries to kill itself. For example, lactating mother, when they finish lactation, they want to get rid of the power of generating milk. And instead of making small vesicle, they just use more than 1 micron size vesicle, put everything in it, and excretion process comes out. The next process, or next server space, I would say, is called microparticle. They are between 500 nanogram to 1 micron in size, and you can see most of the cellular organelles are present in this particular vesicle, lipid-bound vesicle. And one thing you will notice is the DNA, RNA, as well as mitochondrial fragment is also present in this particular vesicle, and they do not use any energy, ATP, for the cell. So they bleb out from the cell membrane. Therefore, mostly plasticity or cancer cell, they use this method to release it outside. But today I'm going to focus on that small particle. We call it small extracellular vesicle, or exosomes. Why? And that's the logic I'm going to give next couple of slides. This advantage of small extracellular vesicle or exosome, they have a specific marker on the surface, molecular marker, and that is tetraspanin protein, CD9, CD81, and CD63. So these proteins identified whether this is a small extracellular vesicle or microparticle. Apart from the size, these are less than 130 nanometer in size. And they also have some matrix protein, which is Alex and others. They also give a unique identity of this. And another advantage when you try to talk about translational medicine, you need a lot of tool to separate them and characterize them. So that's what we have currently to study small extracellular vesicle. So why a resting cell without any major problem or stress will secrete something like this? That means they're using their ATP. So here are the 4 reasons one resting cell try to maintain the regular homeostasis when they have protein quality control, cellular polarity, or ECM, extracellular matrix. So at that time, cell try to get rid of those stresses by releasing this small extracellular vesicle. It's like when you use a lot of groceries in your home, you take empty stuff, put it in a trash can and take it out. That's the cell's logic of secreting this. So this is basically a concept. What we realize is if you take your trash out a night before and somebody start digging through your trash, they can figure it out what you ate last day, right? So similarly, what one cell, when they secrete out, other cell also notice what you wrote. It's very similar. Your post seen by 100 million people or your best friend, one person. So that's how cell-cell communication works. One cell secretes something out, other cell react to that. And if you see on the left-hand side, I know it's a very busy slide, this is how small extracellular vesicle biogenesis occur. So when you have a lot of material, bioactive molecule is gaining the content of a cell, it sends a signal to the cellular membrane and invagination of cellular membrane occurs. And then that forms in the early endosome formation by using ATP. And then endosome formation where they use different ATPase and Alex to make smaller particle inside or vesicle inside. And that's why it's called multivesicular bodies. That multivesicular bodies then RAB27 ATPase drag it back to the cell membrane, and that's where the opening occurs and all those vesicle comes outside. I want you to just remember this blue X marker. This blue X marker came from this particular cell membrane portion. Just remember this part and then we'll talk why we are talking about that. So this goes outside in the circulation, and then there are several mechanisms, ligand receptor mechanism, they get incorporated, or the cargo get incorporated into the recipient cell, and that can be a functional. So therefore, what I try to tell with this slide is this donor cell will influence the recipient cell's function by delivering the cargo. My American friends knows this thing very, very well, and we hate this. And every time we see this thing, and we remember, where is this? I'm not going to ask you, but this is called speed camera. So in America road, you are driving with your own sound, music, and everything, and then next day or day after or week after, you get a ticket, and you have to pay $120 because your speed was 15 miles per over. So the question we ask, how this machine take a picture and figure it out, where do you live? So that's what we will use. So they have a radar. They read your license plate. Once they read your license plate, they know your address and who else lives in the house, right? So we told our team member, so why don't we come up with a molecular speed camera, and we will read what's outside, and then we will figure it out which cell is secreting this particular small extracellular vesicle. And that's what we try to do. That remember the blue box with a X in it. That's what we're going to read using our cell-specific molecular speed camera. So for next couple of slides, we will tell you the background, or what we call it, server space backend, which is how we make molecular speed camera. So this is the first step. We take primary cell. Remember, we cannot use cancer cell or any cell line which comes from cancer. So we use non-neoplastic primary cell, whichever cell we are trying to identify the speed camera, right? We will take those and we will culture those using FBS. This is the most important thing, because cow serum or plasma, they also have extracellular vesicle. We do not want to contaminate with our source of extracellular vesicle. Then from cell culture media, cultured media, we will isolate those small extracellular vesicle, and then we will use proteomics using bioinformatics. We will identify all the shotgun proteomics target, which are the protein stays on the surface. So we have list of our protein, and then we will use a validation cell-specific marker method. So this is a very simple method. What we will use is on the left-hand side, that's the surface EV, and we will use protease. Once you use protease, it's like a Pac-Man thing. It will go and eat or chew the protein on the surface. However, if you add detergent in the same mixture, so they will make holes, and now your Pac-Man could go inside and chew up inside material. So that's how we can distinguish in the EV which proteins are outside and which proteins are inside. So this is one representative figure. We're using dot plot. This particular method we use and the principle we use, and then what you see is by doing this dot plot, we can tell that protein expressed outside or our cell-specific marker. And this is a representation, and this is the way we can tell this protein is inside the vesicle. We generate a catalog of cell-specific marker for T cell, B cell, macrophages, microglia, and these positive and double positive and triple positive, how specific they are. Triple positive is the best. So using this method, we can capture or isolate the T cell specific EVs from our blood or any biomarker. So what are the implications? Let's talk about the implication. We made the speed camera for molecular purposes. We can use it for biomarker. So take one of the common conditions which is growing crazy in the whole world, obesity. So our goal is to get obesity and take it to the next step, which is obesity induced, or obesity is a major factor for different diseases. So if an obese person come to your clinic and then we can tell that, hey, you do not have any type 2 diabetes yet, but within 6 months you will develop type 2 diabetes, and if you don't change your lifestyle or take medication or start jumping, something like that. So that's our goal, and how we will do it using this. So we will take their blood, then we will isolate plasma, then we make aliquot their small extracellular vesicle fraction, and then what we developed, we will capture B cell specific EV in one of these plates, and then we take whatever unbound fraction and put it in another plate which is specific for endothelial cell. Remember, you took 500 microliter of blood at the beginning. It's very sensitive, and we are repurposing the same thing over and over. And we take the unbound fraction from endothelial cell specific plate and put it in hepatocyte specific plate, and ultimately we will put it in adipocyte specific plate. Starting material 500 microliter of blood. And what we will do is we will take these 4 plates and then we will tease out what are the risk factors in it. So this is the product, obesity product, and what you can see here is those are the 4 markers we found, and those are the microRNA beneath it, like here, that will tell us risk factor for adipocyte specific, this one pancreatic beta cell specific extracellular vesicle, and these marker, if they go up certain range, this individual may have type 2 diabetes, and then similarly in other cell type. On the right hand side, what you see is we really capture the EVs, how specific, and what is their number in 500 microliter of blood from the same blood. So now this is the predictive value, what you are saying, what we are trying to do here. We got 2 sets of samples, 2 groups. One is healthy individual or lean individual whose BMI is less than 25. And then second cohort is obese individual without any known diseases, no type 2 diabetes, no hypertension, except for their BMI is more than 30. So we've identified how many beta cell specific EVs are present, endothelial specific EVs are present. There is no difference, that much of difference. Slightly elevated in hepatocyte specific, but it's a statistically significant increase in adipocyte. And we agree, right? Obesity is just gaining fat, and so fat will release more and more EVs, and that's what we are seeing. And these are the pancreatic beta cell specific marker which I showed you earlier. These microRNAs increase the risk of type 2 diabetes or beta cell function. So now we are working on AI model to put all of this input into a machine learning algorithm, and we can come up with a time frame. So when you see a doctor get this kind of test, and then the spit out of AI will be in 7 months, you will have type 2 diabetes. Similarly, we have endothelial cell, 8 months for hypertension and fatty liver disease. We can come up with a number unless you change your diet, unless you eat healthy or do exercise, something like that. So if somebody goes to a doctor and say, hey, you are obese, please do some exercise, that person will come out and forget about what he said. But if you tell the same individual, hey, if you don't do it, 7 months later, you will have type 2 diabetes. So that's kind of emotional push we are trying to give. Now the second part, so one cell tweet, now other cell will read that tweet. So again, we come to the obesity part again back. And so this is a marker conformation of adipo-EV or adipocyte-specific extracellular vesicle. Suppose this is the method, and this is what we did for adipo-EV. We have the same method, but this time we are not doing sequence, we are just capturing adipo-EV, and then we are trying this molecular bioactive molecules, which is microRNA here. So these are the 20 microRNAs, highly expressed. And if you can see in the obese, you see a statistically increased fold change of these microRNA. They go up. Why we pick random 20 microRNA out of 6,700 microRNAs? Because when we take the fat and do a sequencing, these are the top 20 microRNAs present in fat pads, in human fat pads. So now, let's go to cooking school. And now you know who you are cooking for. And in America, if you try to put too much chili, that person will not eat it, right? So you have a mix of 20 microRNA in your spice shelf, and you know which spice you are not going to use, chili, right? So how you do it? You do an in silico analysis and figure it out, I'm making food or I'm tweeting it for cardiomyocyte. So which are the microRNA out of these 20 is functionally active in cardiomyocyte? This way we screen out from 20 to 8 microRNA, and this has a potential function by in silico in the heart. So we want to know these 8 microRNA, how they influence cardiac function from the AdipoEV cargo. So we went to the animal model and we put them in a high-fat diet for 18 weeks. It's a 60% high-fat diet. They generate obesity. And what we did is we want to know in this model which are the microRNA from our 8 microRNA list is detectable in a very low range. So that means they are most abundant. So out of 8, we found 4 is in a very, very high range, which we can play with by functional assay. So the first thing what we want to know is this hyperglycemic condition. So one can argue that, hey, looking at these 4 microRNAs, they can also upregulate in the fat in hyperglycemic condition like obesity, right? So we took some cardiac cell and we put them in or mimic them in a hyperglycemic condition by adding lipid, by increasing the glucose, and we challenge them and ask them the question, hey, will you change your expression of these 4 microRNA? The answer is no. So that means if we see in the heart with a high-fat diet, these microRNAs are going up, that means it's coming from outside, not the cardiomyocyte, the capacity to upregulate these microRNAs. So that's what we did. We took those hard from high-fat diet and we asked the same question. Your expression is up, all 4 shows up. That means these 4 microRNAs coming into cardiomyocyte from outside, mostly EVs, right? So now what we want to know, how about these 4 microRNAs in the fat pad itself? So if we put them in a high-fat diet, are any of these microRNA changes its expression from just by, in the fat expression? And what you see is, first observation will be from 4, now we are talking about 3. So what happened to LET7A5P? We did not see the expression in a measuring level in the fat. So therefore we exclude this. But what we see here is the LET7 is the only microRNA which overexpress inside the fat cell or adipocytes with the hyperglycemic condition. So we thought that's our target, because as fat grows in number, as well as LET7 itself can grow their expression in one cell, so that can generate a cumulative effect in cardiomyocyte. So from now onwards, we will focus only on LET7C. So the question is, we found the molecule, but we are not even sure whether cardiomyocyte will read a tweet from adipocyte. So we wanted to do some kind of assay to identify whether these are the assay when we stress or stimulate adipocyte with high glucose, high lipid, the EV, what will come out, we put it in the cardiomyocyte and we try to see whether the cargo inside will go to the cardiomyocyte or not. And what you see here, the confocal microscopy, because of the light problem you cannot see the green color. Yes, here. The green color is the cargo of EV, and you see it in cardiomyocyte. So that's how we are saying the uptake of AdipoEV cargo into cardiomyocyte. Okay, we can deliver it, but whether there is any functional effect or not. So what we did here is we basically use a co-culture method. We culture adipocyte on the top and cardiomyocyte stick to the bottom, and then we try to see by this EV transfer, can we change function of cardiomyocyte? The answer is yes. By using MTT as well as creatinine kinase release, we can say that just by co-culturing together, cardiomyocyte function is changing tremendously. And then what we want to do is try to identify when LET7 is the functional one or not. What we did is basically we take cardiomyocyte, we individually overexpress each of the 4 microRNA, and then what we did is we use a cocktail of 4 microRNA and express it inside the cardiomyocyte. What you can see is the LET7 here And here is same as the cocktail. That suggests that out of 4 microRNA we screened out, LET7 itself, LET7C itself can do all the bad things in cardiomyocyte. So then what we did is we manipulate the expression of LET7 in adipocyte, and then we are trying to see using siRNA, or here we call it antagomere, and try to see in the cardiomyocyte whether we can solve the problem or not. And I'm not going to take too much of your time, and what I'm going to tell you is, yes, LET7C target PPAR alpha in the cardiomyocyte, and if we reduce the expression in adipocyte, PPAR alpha level goes or normalize, and we do not see cardiac hypertrophy. But in order to prove it, we generate a mouse model. I know we do not have time, I don't want to ring that bell again, but what we generate is this mouse model. The EV which will come out from the adipocyte will be green in color. And we have an ability to capture those green EV and try to interrogate inside whether the LET7 is causing any trouble or not. And so using this method, we capture the green EVs. Now we know they are only coming from adipocyte, and then we look at the level. And what you see here is this adipocyte specific EV is almost six-fold in the high fat diet model. So basically all of these EVs goes to the cardiomyocyte and deliver LET7, and this cumulative effect influence the cardiac function. You can say that, hey, you force in the in vitro assay and put the adipocyte and cardiomyocyte and no other immune or other cell type, but show it in vivo. So we use this mouse, and using confocal microscopy, what you see, these green dots in the figure, those are the green adipo EV in the heart. And these are the section OCT fixed section. And what you see here in cardiomyocyte, the accumulation of this green and attaching to the red, which is the cardiomyocyte membrane, is way more than normal chow group, suggesting that LET7 transfer in the adipo EV is way higher in high fat diet compared to the normal diet. And these are the functional, as I said, PPAR alpha and cardiac hypertrophy. So it's all influenced by adipo EV. Finally, in summary, what I said, this cell-cell communication, again, think about your post and your friend reads your post. So adipocyte during obesity will increase the LET7, and that adipocyte will say, hey, I increased my LET7, and that adipocyte will secrete this EV. EV will reach to the heart and do all the bad things. With that, I would like to thank my group. Thank you for giving me extra time. And most of the work has been done by my talented people there. Dr. Mitra also showed Shushmita's picture. She spent some time at Hopkins, as well as Pawandeep Kaur, also from AIMS and from Dr. Mitra's lab. They work on this particular project some way or the other. And my collaborators and Dr. Mitra, we are doing some TB work, good and bad news. Hopefully next meeting I can share with you. Thank you all.
Thank you very much, Dr. Samarjit. Due to paucity of time, will not take any questions. I myself have a question which we can discuss later in the evening. Thank you so much. So now we move on to the technical talk by MNNIT Biotech India by Dr. Prudvidhar Shripuram Budhuru, if I am pronouncing it right. So from MNNIT Biotech India. So we have 15 minutes, and I would request you to stick to the time. Thank you.
please wait for a while. We'll have a memento to the speakers and the chairperson. After that, you can have a talk. Till then, you can give a presentation to the technical team.
I didn't understand. I'm sorry. Do you want me to?
Actually, you have to give a memento to the speakers and the chairperson. Till then, you can give your presentation to the technical team. Okay. So that you can give your presentation to that side where the technical team.
It's already here. I have already tested this with them.
We have to present the memento to our chairpersons and the speakers now. Just if it's possible.
Are you facing any difficulty in putting on the slides?
No, ma'am. I think there's a memento presentation before that. There's a memento.
Okay.
I'll present the memento. Then after
So now,
just it will take 5 to 6 minutes. Then you can present your presentation. I'll call you now.
Okay. Right. So I would request the speakers of the earlier session, Dr. D.K. Mitra and Dr. Samarjit Das, to come and receive the memento.
On the behalf of the organizing committee of IMMUNOCON 2025, I extend our sincere gratitude to our respected chairperson, Dr. Kalpana Luthra, for their valuable time, guidance, and for conducting this session with such grace and depth. As a token of appreciation, we would now like to present memento to our distinguished speakers. I request our chairperson, Professor Kalpana Luthra, to kindly come forward and present memento to Dr. D.K. Mitra, Department of TII, AIIMS, New Delhi, and Dr. Samarjit Das, Johns Hopkins University, USA. Yes, after. Also, a very kind request you all to please be there on the stage for the group photograph, sir. Now I request Dr. Ambak Rai to kindly present memento to our respected chairperson, Dr. Kalpana Luthra, as a token of our heartfelt gratitude. Thank you so much. Moving ahead, we now begin the technical talk of the session. It's my privilege to invite Dr. Apurva Bharadwaj, the Country Medical Affairs Director at Miltenyi Biotech India and the Cell and Gene Therapy Center of Excellence in Hyderabad. Dr. Bharadwaj is a distinguished expert in cancer therapeutics and advanced cell therapy, with a special focus on developing point-of-care CAR-T therapy manufacturing in India. She has been instrumental in advancing Miltenyi Biotech mission to make cutting-edge cell and gene therapy more accessible and affordable across the nation. Her leadership and scientific vision continue to shape India's progress in personalized medicine and next-generation cancer treatment. With great respect, please welcome her with a big hand.
Thank you so much and good evening everyone. So as you can see, I'm not Dr. Prudvidhar, our very talented Miltenyi Biotech Medical Affairs Director, who was very sweetly and very well described by the team here. I'm filling in for him, and my name is Apurva Bharadwaj. I head Product Marketing and Strategy at Miltenyi Biotech. So like I was saying, everyone, good evening. My name is Apurva, and I'm here to introduce you to Miltenyi Biotech, the global leader in cell and gene therapy, a name you might have or might not have heard so far, but something that's definitely around to stay. Our mission is to advance scientific research and medicine by providing solutions for curative cell and gene therapy, as well as biomedical research. Now, what is MNNIT Biotech and who we are? MNNIT Biotech is a German company with 35 years of CGT experience in the field. It was founded in 1989 based on the invention of MAX technology, which many of you might already have heard of. But today, our more than 19,000 products play a vital role in paving the way for over 150,000 cell therapy procedures around the world, having already treated more than 10,000 patients. With our headquarters in Germany and our R&D and engineering base in Germany and Tetro, our cell manufacturing centers are located in Bergisch Gladbach and Shanghai, and very, very happy to tell you that MNNIT Biotech this year brought the first cell and gene therapy center of India to Genome Valley, Hyderabad. As you can see over here, our integrated technologies span a broad range of applications, from cellular research to genetic sequences, high content imaging, and commercial scale manufacturing. So, in the true sense, from benchtop to bedside for patients. The end-to-end solutions, reagents, and capabilities, this is what helps us aim to be partners for anyone who's working in any of the research areas that are shown here up on the So just to let you know, these are currently the CAR T therapies out there globally, which Clinimax Prodigy is powering, with currently being used in more than 150 clinical trials across 45 countries with 6 commercially approved therapies. Now, why I'm telling you all this and why I have introduced you to this company is just so you understand the context of what comes next. Globally as well, there are challenges to cell and gene therapy. It's not just the landscape of India where we are seeing certain challenges crop up again and again. Is it accessible? Approximately 20% eligible patients are treated. Is it effective? With newer improved therapies, the complete response rate is slowly rising and is now close to 50%. Is it safe? Severe adverse effects are under control, less than 50% at the moment as of 2025, and this is improving. Is it universal? Minorities are still underrepresented. And is it affordable? Maybe heavy on the pocket, but patient access plans are arriving to support. Cell therapy is the future, and that is why we are all here discussing these trends in IMMUNOCON. Patient access, however, can be a bit of a challenge, and there are several reasons why this may happen. Limited drug registrations, reimbursements, insufficient manufacturing capacities, long vein-to-vein time. India is a large country. One can almost think of it as a microcosm, which is why we genuinely believe that the way to go ahead with making precision medicine more accessible and more sustainable for the public is to democratize it. So, I think for those of you who might have attended Dr. Hament's talk this morning, he did talk about the first clinical trial which is going on for Wellcut, and he might have also introduced the concept of point-of-care manufacturing with CAR T cell therapy, which is here to stay and can make a big difference considering the landscape of Indian medicine today. So what is point-of-care manufacturing? Point-of-care manufacturing refers to the process of producing CAR T cell therapies locally, within or near the hospital where the patient will receive this treatment. In this model, T cells are collected from the patient, genetically modified, expanded, and re-administered all within the institution. So the entire process of CAR T cell development, but with infusion within the locality. Now, why we think centralized versus decentralized models might work, and probably in India, a mixture of both of these models is required to go forward for anybody who's starting research in CAR T cell therapy and clinical application of it, is that one, it shortens the turnaround time, so there is faster treatment. It reduces transport risks. It enables personalization where products can be tailored immediately to the patient. It lowers the cost, extremely important, by eliminating large-scale centralized facilities, and increases access to advanced therapies. Some case studies for you. So at the Erlangen University Hospital, there is a GMP laboratory that manufactures CAR T cells locally, that is within the facility itself. This is not outsourced to a big external CDMO, but it is produced within the clinical research infrastructure itself, a central point of the point of care manufacturing concept. This is, of course, production is done using the closed system bioreactor, which is the Clinimax prodigy for CAR T generation. In a very, very recent development, just about October 2025, Erlangen actually treated a 21-year-old patient with therapy-resistant ulcerative colitis, a severe inflammatory bowel disease, using CD19 CAR T cells. Why this is significant? Because this was the first time globally that CD19 CAR T was used for this kind of bowel disease, which is in the autoimmunity context and not just cancer. The modified T cells were produced locally under the supervision of our dear friend Dr. Michael Eigner, and the outcome was that the patient's intestinal lining completely healed and they became independent of the disease. Another case much closer to home and also specified in a lot of detail this morning itself by Dr. Vikram Matthews' team is the trial at CMC Vellore, where Christian Medical College Vellore conducted a phase 1 clinical study called VELCART, and they manufacture anti-CD19 CAR T cells in-house, which is at the hospital, using a point-of-care strategy. They used the Clinimax Prodigy system, which is a fully automated closed system, to make the CAR T cells here. And the vein-to-vein time from collecting the T cells from the patient to administering them back to them as CAR T was 9 days. 9 days only. So this is the kind of difference that a model like this is able to make. So very, very simply put, and just to sum up the concept of centralized versus decentralized, both of which, of course, all of us are here to support with, is that some of the challenges that we usually see with this is access to treatment. Point of care beats that, and these facilities across major cancer care centers, they help democratize patient access. Time to treatment, this is something which is also taken care of when there is point of care, because when the patient's room, the ward and the clean room are right next to each other, obviously there is very, very less vein-to-vein time. Logistical complexities, cell viability is always better when fresh. And when it is manufactured close by, that is what becomes possible. And the shortage of skilled workers. The need for structured training is, and for upskilling is something which is rampant. It is definitely there, and we are here to support with that as well. An example of that is the first CAR T hands-on training for cell manufacturing, which took place earlier this month at THSTI. So all in one package, just in summary to let you know, is that Miltenyi Biotech is a solution provider here to stay, and this is the company that can support you from research, whether you're working on T cells, engineered T cells, CAR NK, HSCs, MSCs, iPSCs, and I see a lot of people who are already doing that with us. Moving on to clinical applications and regulatory. So I think with that, I would say thank you very much for giving me the time to listen, and I wish you a very pleasant evening ahead. Thank you.
Thank you, ma'am. Now we begin our plenary talk, and we invite our distinguished chairperson to kindly come forward and take their seats on the dais to lead this session. We are privileged to welcome Professor S.K. Arora, former professor in the Department of Immunopathology at PGI MER, Chandigarh. Professor Arora's work has made major contributions to understanding immune mechanism in both infectious and autoimmune diseases. With decades of academic and research excellence, he has mentored numerous postgraduate and doctorate scholars and has published extensively in leading national and international journals. His expertise in clinical and experimental immunology has significantly advanced translational research in India. Professor Arora is widely respected for his scientific leadership, mentorship, and outstanding contributions to the work of immunopathology. With our esteemed chairperson on the dais, I would like to request them to kindly take the charge of the session and invite the speakers for their respective talks.
Hello. Okay, thank you very much. Thank you for introduction. Good evening, everyone, and this is the last session of the day. I know you all are tired, but still, this looks to be very interesting talk by Dr. Abhay Dandekar from Department of Plant Sciences. It's very interesting to know that in UC Davis, he's trying to develop something edible, and the title says that it's Core, a new platform for edible plant-based manufacturing. Dr. Dandekar is a distinguished professor in the Department of Plant Sciences at the University of California, Davis. His research focuses on dissecting plant phenotypes to define the molecular, genetic, and physiological and biochemical mechanisms regulating productivity and quality in fruit and nut trees. His laboratory identifies genes, regulatory pathways, and metabolites underlying key traits to develop trait-specific diagnostics and improvement strategies for sustainable crop production. I'm sure it's going to be very interesting talk, so let's listen to Dr. Dandekar. Floor is yours. You are given 35 minutes, but I'm sure you will reduce it looking at the plight of everyone. So let's be brief.
So which one is yours?
The top one. This one? Yeah, IMMUNOCON, yeah. And then turn on the laser points.
Well, thank you very much. I appreciate the opportunity to speak. I'd like to thank the organizers for giving me the opportunity to speak to you today, and thank you for your kind words. So, if you look at the IMMUNOCON logo, you see on the left side there's a gentleman or a man or a person, and then there's a DNA molecule. And then coming from the top of that, I'll look like plants to me, right? So, well, that's going to be my topic of discussion. I'm going to present to you sort of a strategy. I'm not going to go into a lot of data and show you what we are doing. I've spent maybe 4 decades working on plants, especially walnuts. And so you're going to all want to eat walnuts at the end of my talk. Okay, so what we mean by core is clonal, organogenic, renewable edibles. So what we've done, we've been working on walnuts, and one of the things that we discovered long back was the ability to remove stem cells from these walnuts at early stages of development. So seeds are embryos, and embryos are what you eat, right? So these kernels, at an early stage, we can extract stem cells and proliferate them in culture, and they will divide, and you have now these stem cells. And then what we would do, we developed a series of technologies to be able to transform them
genetically modify them, all sorts of things. And then we were able to put those embryos back into soil and get plants out and then grow those plants. And we've conducted maybe about 100 field trials with those plants, with this stick. That's how we developed the technology. Well, what I'm going to talk to you today is about actually taking these embryos into solution and then growing them in a bioreactor at scale and having really a form of low resource biomanufacturing. We've all been hearing about the lamentations of not having adequate biomanufacturing facilities. Well, this will be a very simple way to grow these embryos, and they are edible. So they can be consumed, much like walnuts can be consumed. You can think of them as micronuts. And these micronuts are consumable, and they will work through the gut axis. And you've heard from Satya talking about how the gut has 80% of the immune responsive cells and has all these axes. There's a gut-brain axis, the gut-liver axis, and that's how we are hoping to, in a nutshell, that's what I'm going to talk about. Okay, so what is our target? Our target is chronic diseases, and you've heard a lot of talks about chronic diseases. They cause 75% of the death worldwide. And inflammation is the major underlying cause, and it's a global burden. So we really need new platforms to address and provide therapy to reduce these diseases. There's an increasing demand for biologics, and these could be different peptides and proteins. They could be specialty metabolites, and we've heard some discussion on those. But there's also, we need food-safe additives. As you know, when you drink any beverage, it has a color to it. And that color is a fossil fuel-derived chemical. And so you're consuming, is really a poisonous chemical. When you drink Coke, it's a brown color F. There's now people are trying to actually reduce the red color. The red color is a really nasty dye. So can we make additives that give you pigments? So current systems are really limited. We are really talking about cells or fungi, growing them in bioreactors to produce a lot of metabolites. But they're limited by cost and scale. You need stainless steel fermenters, you need stirred tank reactors. So plants really offer an untapped potential. So why plants? This slide goes through, makes an argument why we should think of plants. If you think of globally how much carbon is fixed, 82% of the carbon fixed in all terrestrial locations is fixed by plants. So it's about 450 gigatons of the 550 gigatons that are fixed in the biosphere. So plants are scalable, right? The other thing I want to mention is that all the food we eat is seed derived. That 75% of all your food comes from seeds. Those seeds you eat, they go through in your gut and they provide you all the nutrition, but also provide you all these useful metabolites that are good for your health. So medicine plants are the first ones to give you medicine. They came from plants, aspirin, for example. So that's an equivalent of about 0.5 gigatons. That's a lot of scale, right? 0.5 gigatons, to give you an example of what that is 5,000, the weight of 5,000 aircraft carriers. That's an amount of protein that we eat, or seeds that we eat. So plant cells also have the coding capacity. They have the ability to convert carbon dioxide or sugars into any useful products, and you don't need any complex media components. It's just mineral solutions, sugar, and you can make any metabolite protein that you want. And so what I'm focusing on is having the edible plants, and the reason for that is we know that food is medicine, and walnuts have a lot of what can provide diet-derived metabolites. And so we've been really interested in understanding what are these diet-derived metabolites. I want to explain to you that walnut is regarded as a superfood. So if you Google walnuts and superfood, you'll see that it's one of the top superfoods. And the reason for that is it has a very high content of the omega-3 fat, the alpha-linolenic acid, right? And very few plants or seeds accumulate alpha-linolenic acid. And alpha-linolenic acid is created because of the omega-3 desaturase that's present in the walnut genome. And that forms the basis of all the long-chain fatty acids. So the eicosanoids and the docosanoids require the omega-3 desaturation. Humans don't have or any animal doesn't have omega-3 desaturations. There was a study conducted some time back in 1958 where they looked at the Seventh-day Adventists in Loma Linda, California, and they found that they had the lowest incidence of coronary artery disease. And the reason for that is they ate a handful of walnuts every day. So it's a landmark study, and they try to understand what is it in walnuts that reduces cardiovascular disease. Now, they are Loma Linda is regarded as a blue zone, so they not only have lower incidence cardiovascular, they live long life, and they're very productive and very active. And so there's been a search of what are the metabolites in walnuts and so there've been lots of studies done with consuming walnuts, and written here is some of the positive benefits that have been found. So they're obviously metabolites that are mediating these health benefits, and we are interested in how do they work from the gut, and can we provide them in an edible form. So mental and mood disorders have shown that walnut consumption actually improves mood and decreases mental disorders. It counteracts addiction, and those are due to the satiety factors that walnuts have. So that means if you eat walnuts, they prevent you from consuming too much. And those are satiety factors that work through the gut-brain barrier, and we've been interested in trying to identify what these satiety factors are. You've seen a lot of work on the GLP-1 inhibitors, and you're seeing remarkable differences in side effects, very positive side effects of GLP-1 inhibitors. And they work through the similar mechanism that some of these metabolites that are present in walnut. Pain management, improving the composition of gut microbiome. Clearly, there are metabolites that are being made in walnuts that actually influence the gut microbiome. Aging and lifespan increase, the Loma Linda study. Male fertility is the latest one, increasing male fertility. We don't really know how that is, but there are obviously metabolites that are doing that. So what we are trying to do is work, as I mentioned to you, take these embryo lines and put them into solution and then make these micronuts. And if you compare these micronuts to walnut kernels, so you have here walnut kernels, a handful of walnuts is about 200 calories, and you can see that what all the different contents, this is just a food label that we've made for our micronuts. So walnuts have 65% of their weight fat, and that fat is polyunsaturated fat, as I mentioned, it contains. They don't have very much carbohydrate, they have a good amount of protein. So if you look at the walnut micronuts, this is just the micronuts, they are much less. However, they are 95% water. The walnut kernels are about 2 to 4% water. So if we dry them down, dry these micronuts down into micro flakes, or little walnut flakes, you see that now you have much higher calorie content, good amount of protein, and we found that it has a lot of the very interesting fatty acid-derived metabolites and phenolic compounds that are really important for health. And that's something that we are investigating. So this is the workflow. So if you wanted to go about creating these, you'd have to take walnut kernels at early stages, put them into culture, so you now have an organ. An organ is different to a cell culture. A cell culture is a two-dimensional structure. Organ is a three-dimensional structure. So an embryo has a apical and root axis. It has phyllotaxy. It has got all this three-dimensional structure. It's got the infrastructure. It has got vascular systems. It's got all the infrastructure to do biomanufacturing. And the biomanufacturing can do is create oil bodies, protein bodies that sequester proteins, so that they are released, slow release. The oil is in oil bodies and then they are slowly released. Therefore, that's why when you eat walnuts, you don't get fat, even though you're eating something that contains a lot of fat, 65% fat, as I mentioned, by weight. If you eat potato chips, on the other hand, which are replacing the water with oil, you put on weight. So then we reprogram. So since we know all about have the complete map of the genome, and I'll show you that, we can reprogram them to do anything, and then we harvest them and you consume them. There's no processing involved in providing the benefits of these metabolites that are made in these bioreactors. So CORE, I want to mention, is a functional design platform. To think about it, we have the complete sequence of the walnut genome. We know that it has 16 chromosomes. We know what's located, about 32,000 genes. We know where those are located on these chromosomes. We have very high resolution sequence of the genome, so we have them phased. So that means the two diploid copies, the haplotypes, we have the sequence of each chromosome, but each haplotype. And this way, we can examine allelic differences because that's very important. Trees like walnuts and many of these plants, which are vegetatively propagated, are highly heterozygous. So we need to choose the right genes, choose right locations to make changes in the genome. And so we can use, decode that information. We use a lot of data science. We have RNA-seq analysis data, we have proteomics and metabolomic data, so we know exactly what's made, what genes are involved in those, what pathways exist, and then we can change those pathways. We have epigenetic, we've spent a lot of time shutting down pathways using epigenetically, using siRNA, targeted siRNA. We also have CRISPR genome editing capacity that we've put, not only genome editing without any residue. That means we don't have transgenes as part of the, we can introduce DNA-free editing systems. It's sort of shown here. The red ones are the wild type and the white ones are edited. So we can very easily edit it. So we're also creating RNA-inducible promoters. We are looking at what promoters are active in the embryo stage. So we can engineer things with using resources from the walnut genome itself. And then we can do the metabolic engineering, create new pathways, pigments and proteins, and I'll give you examples of that. And then we need to design a bioreactor, and these are very simple bioreactors that we are starting off by 3D printing these bioreactors so that we can test the prototypes. So they have tremendous application of this technology to immunology and therapeutics because this is an oral delivery system. A lot of people do not like to be injected with needles. So oral delivery is something that's not been looked at that carefully, and this is another way to think about it. Proteins, antibodies, metabolites, immunoregulators, on-demand antigen. We were involved in producing on-demand, really fast antigen production for vaccines. It can be decentralized. You don't need these bioreactors can be fabricated and put in low resource environments. And that's something we are looking at. So we were involved in this rapid antigen production. We actually introduced the COVID spike protein and the RBD, and we were able to express that. So this thing, you can go from sequence to product in 9 months. You can have a protein that can be ready to be injected. You have cell lines that are overproducing the protein, and it's ready to go. We are just starting a project with the US space program, because the ultimate low resource environment is space. And there's a lot of interest in improving the bone density of astronauts and people who are in space. And so we have built a PTH hormone, which is it's a 34 amino acid peptide. We have linked it to the FC part of the IgG, and we will express that in walnuts, so that these could be provided to astronauts in space. The bioreactor can be put in space and works in space, and that's something we are at the moment experimenting on. This is the first prototype of bioreactor that we are going to send into space. It is a simple little chamber here where the embryos will sit, and this is put into the media, and then the media is just turned over and wets the embryos. It's called temporary immersion. It's a temporary immersion bioreactor. And so this is going to be launched. There are about 6 of these. They're going to be launched into on the International Station in 27. So we are sort of getting ready, printing it and testing these, because there's a really need for having food for long missions. So this is something we are thinking about. Regarding the food safe colors that I was talking about, this is an example of the red color. So as you know, beetroots have this wonderful red color. It's called betainin. It's this molecule here. And so we were able to put 3 genes from beetroot that convert tyrosine, the amino acid tyrosine, into betaine. So you can look at here, look at these embryos. They are highly red. And so we are actually making more betainin in our embryos than beetroots make. So just think of a bioreactor making a bunch of these embryos, and you can just extract it. It's in an edible form, so you can just dry it and sprinkle it and make the red color. We are also able to make melanin. I don't know if you have heard of melanin. Of course, we have it in our skin, right? So melanin is a really important biomolecule, and the only sources of melanin are cuttlefish and insects. That's how they extract melanin. And so walnuts naturally make melanin. And so so you can see, so we have the genes that make it, and so we can make melanin. Melanin is a really important pigment. I'm not sure you've heard about it, but melanin is, people are thinking of using it as in chips. So these will be biodegradable electronics. They're thinking of printing circuits on your skin so that you can have mechanical devices that are triggered by the melanin. Melanin is also used for radio protection as much as it works in our bodies. Melanin is used to protect against UV radiation. But for hypersonics, hypersonic missiles systems, protecting avionics, spraying on aircraft, high-performance aircraft wings, these all require melanin is one of the best forms of protection against heat that occurs at high speeds. So there are different types of melanins, and we can make all the different forms of melanin in our system. So in summary, what we are proposing is you go from orchards, that's where walnut trees grow, to core-based living devices. And so we will create these devices. These are embryo lines that, or micronuts that are producing various things. We are actively looking at various of these. So we call them living devices. They will grow in bioreactors and perform and give you, there'll be next generation things to help health. And I've talked about all of these things, and so you can see that. So the future really is rooted in low resource manufacturing. We really need that. There are lots of places that require, that can be involved in manufacturing, and this type of manufacturing is easy to install. The Army is really interested in forward deploying bioreactors to provide meals for warfighters in various altitudes, having specialized meals, so we can add to that rather than have the CREs, which is having MREs, which is meals ready to eat. You have cultures ready to eat. And so one of the things we are thinking of, this is space travel. Long distance space travel requires your ability to manufacture your food. There's not enough space on a spacecraft to take the food for a Mars mission, for example. So there's a lot of interest in understanding how to create food that is, hey buddy. So we have I think he's interested in my talk. Okay, yeah, I'm almost done. So why now? I think this slide just explains why this idea is going to receive traction. Space and defense missions require very high, long shelf life, compact food. Synthetic biology enables all of this to happen. The climate crisis is going to move production indoors, and so we are ready for that. We can also, I didn't have time to go over this, rewilding, we can actually put back lost, when we breed plants, we lose a lot of interesting molecules. I think this allows us to get those back. By looking at wild plants, we can simply transfer those genes into plants. So the path forward, you've got to optimize yield. We've got to figure out the regulatory pathways for scalable, edible biofactories. We should be expanding these. I talked about walnuts, but you could expand these to other crops. Any crop that makes seed, we can culture the embryos, and now you have the embryo version of those plants. And then this is going to be integrated with automation and synthetic biology and AI, so it's going to happen really fast. Okay, so I think this just repeats that. So I'll just stop here and take any questions that you have. Thank you for your time.
Thank you very much. Very interesting. And the only thing I want to know is, every time, you said the time span is 9 months you will have from the start to get the product out. And
yes. So
it's for walnut, right?
Yeah.
You can try other things also.
Yes, actually, if you you could do it in 3 weeks if you just allowed transient expression to occur. So that's possible. That means you deliver the new genetic information into the embryos, and then so in 3 weeks' time you can go from sequence to product. But if you wanted a stably transformed system, it takes about 2 months to select the lines, and then takes time to grow them up in a form that you can extract in a large scale.
Very nice. So it's open for question. Shyamal and.
So for this plant-based like spike protein that you have mentioned.
Yes.
So the pattern of glycosylation.
Yes.
In mammalian versus plant, they're very different.
Yes.
So how do you really take care of this kind of issue?
Yeah, no, it's a great question. So we've spent a lot of time on glycosylations. So as you know, anything that's secreted goes through the glycosylation, because the glycosylation pathway is present in the endoplasmic reticulum. And so you can do a couple of things that we've been doing. One is that you could retain the protein in the rough endoplasmic reticulum. So in order for it to be secreted in a plant, we put a signal peptide so it enters the endoplasmic reticulum, and then we have a retention sequence that holds it there. When you hold it there, you get a high mannose protein. And a high mannose structure is humanized. So we could do that. The other thing we could do is we could knock out the xylosyl transferase and the fucosal transferase. If we knock those out, then you don't have the plant-specific fucosal and xylosyl residues. And since we have the technology to RNAi knock out things, we can do that. We can actually introduce silation. We can put the whole pathway for silation, in. So you could create a line that it would create glycosylation residues that are good for plant. The other thing I would mention is that we eat plant proteins, so our system, especially when you go through the gut system, are accustomed to plant glycosylation because we've been eating plants for a long time. So these are some of the.
Yeah, can we cut the discussion a little bit less?
Yes.
We can ask questions, short one.
One clarification actually, when you express the PTH as well as the spike protein of COVID, how can you give orally actually? How can you eat the walnuts containing this expressor protein?
So that's where we've got to go through regulation. We've got to go through FDA so that you can consume them. The micronuts are consumable because they are no different to walnuts. So now all they have is this extra protein that's being expressed. So it's tantalizing to think whether you could deliver PTH hormone from the gut. If you look how PTH hormone works, it uses calcium, and that calcium distribution comes from the gut, and then it goes, to different parts of the body. So it's something that we will test, in some animal system. I'm hoping we'll be able to test the PTH hormone. We have tried to use the infrastructure of the walnut embryo. So we've put PTH hormone, just the 34 amino acids, as part of the oil body. So it sits on the oleosin protein that sits in the membrane of the oil body. So we've made a fusion protein of that protein. We've also put it into a plastid thylakoid membrane specific protein. So we've embedded it there. So what happens is it has to be digested. That'll be the neat thing when you eat the micronuts. They've got to be digested, and then it's presented in a form that your body is naturally able to assimilate these. We are naturally able to assimilate the oil bodies, and that's how we get the essential fatty acids. Walnuts make both essential fatty acids, linoleic and alpha linoleic acid. I don't know whether I answered your question. Yeah.
The degradation is one of the problem mentioned in all these cases. The protein degradation inside the gut is one of the major problems.
Yes. So this is what I'm thinking, if it's encapsulated, because what you're doing is you're putting your protein, but it's encapsulated inside the walnut matrix, right? And in the walnut matrix, it's deposited in a subcellular system. Not all proteins we can secrete. We do that for, to harvest, antigens for therapy. But if it's going to go through the digestive system, you're in a matrix, in a food matrix, which has got fiber and all sorts of cool stuff.
So can we stop the extended thing?
Okay. I'm around, so you can come and talk to me. Thank you.
On the behalf of Organizing Committee of IMMUNOCON 2025, I extend our sincere gratitude to our respected chairperson, Professor S.K. Arora, for their valuable time, guidance, and for conducting this session with such grace and depth. Your insights and leadership have truly enriched today's discussion. As a token of appreciation, we would now like to present memento to our distinguished speaker. I request our chairperson, Professor S.K. Arora, to kindly come forward and present memento to Professor Abhay M. Dandekar. Can we have a huge round of applause? Sir, I request you to please stay on the stage. I request you to, sir, please stay on the stage, sir, for a group photograph. Now, I request Professor Vishnu Agarwal to kindly present memento to our respected chairperson, Professor S.K. Arora, as a token of our heartfelt gratitude. Thank you so much, sir. Yes. Sir, can we have a group photograph, sir? Now, it's time to add a splash of color and joy to this evening, presenting the cultural event of IMMUNOCON 2025.
Hello, hello, check, 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 hello, 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 check, hello, hello, check, check, one, check, check, hello, hello, hello.
I request everyone to please take their seats as we are about to commence our cultural performance very soon. I request everyone to please take their seats. We are about to begin very soon.
Hello, hello, hello. Hello, 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 hello. Hello, hello, hello. Hello, hello. Hello, hello. Hello, hello. Hello, hello. हेलो 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 इको कम कीजिए वॉल्यूम दीजिए, वॉल्यूम। हेलो हेलो हेलो, स्टेज पे मॉनिटर वॉइस। हेलो 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 हेलो, माई टेस्टिंग। हेलो 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 हेलो, प्रॉपर नहीं आ रहा है, थोड़ा सा वॉल्यूम दे दीजिए। हेलो 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 हेलो, श्री राधे श्री राधे। हल्का सा मिड बढ़ाइए। हेलो
हेलो हेलो, श्री राधे।
हेलो
राधे राधे, जय जय श्री राधे, जय जय श्री राधे। श्री राधे, राधे राधे, श्री राधे। श्री राधे राधे, जय जय श्री राधे, जय जय श्री राधे, श्री राधे। श्री राधे, राधे राधे, जय जय श्री राधे, जय जय श्री राधे। श्री राधे, राधे राधे, जय जय श्री राधे।
हेलो हेलो, थोड़ा सा गेन।
हेलो हेलो
गेन गेन। हेलो चेक। हेलो हाइस। हेलो श्री राधे। हेलो स्विच। हेलो हेलो श्री राधे। हेलो श्री राधे। इसमें इफेक्ट्स जो आ रहा है थोड़ा कम कर दीजिए, इफेक्ट कम कर दीजिए, इफेक्ट कम कर दीजिए इसका। हेलो हेलो चेक। हेलो श्री राधे। हेलो हेलो, थोड़ा सा वॉल्यूम, थोड़ा सा वॉल्यूम।
श्री राधे।
हेलो हेलो चेक। ठीक है।
श्री राधे, जय राधे, श्री राधे, श्री राधे, जय राधे, श्री राधे, श्री राधे, जय राधे, श्री राधे।
हेलो श्री राधे, राधे जय जय श्री राधे।
I request everybody to please take their seats as we are about to start. Braj, the land where every color, rhythm and melody celebrate the eternal love of Krishna and Radha. Today, we have team from Kana Academy Vrindavan as they bring the divine charm alive on stage. Their performance is not just dance, it's devotion. Every moment reflects the innocence of Braj, the playfulness of Krishna, and the spiritual beauty that has inspired generations. So we are about to step into the vibrant world of Braj. Let our hearts sway with its rhythm and emotion. Please welcome the team of Braj Folk Dance with a huge round of applause.
शुक्रिया शिवम। भक्ति, प्रेम, आनंद की ये खूबसूरत धरती प्रयागराज, जहां संगम है, प्रेमियों का मिलन है, उन खूबसूरत विचारों का जो विचार हमारे मन से, दिमाग से निकलते हैं, और इन विचारों को एक दूसरे तक पहुंचाने के लिए ये जो आज इस मंच से लगभग 132 देशों से जो प्रोफेसर्स यहां आए हैं, अपने अपने फील्ड्स के प्रभावशाली जितने भी हमारे प्रोफेसर्स हैं, वैज्ञानिक हैं, इम्यूनोकॉन 2025 के लिए यहां पधारे हैं। मैं चाहूंगा उनके लिए एक बार अपनी जोरदार तालियां जरूर बजाइए। स्वागत, अभिनंदन। जब मन से कोई विचार उठता है तो वो हमारे दिल तक नहीं रहना चाहिए, उन विचारों को उन तक जरूर पहुंचना चाहिए जो उन विचारों की कद्र करते हैं, एक सोच की कद्र करते हैं। और ये सोच इतनी दूर तक जब जाती है तो नया एक संसार, एक नया आयाम बनता है। और इसको बनाने के लिए हम सब यहाँ आज एकत्रित हैं। 19 तारीख से 22 तारीख तक चलने वाला ये सेमिनार हमको एक ऐसी ऊंचाइयों तक ले जाने के लिए तैयार है जिसमें 132 देशों से आए हुए अनुभवी प्रोफेसर्स, वैज्ञानिक्स अपने अपने मास्टर स्ट्रोक्स के जरिए, अपनी अपनी स्पेशलिटी के जरिए आपको कुछ नया दिखाने वाले हैं। लेकिन ये नया वास्तव में है, यकीनन हमारी अप्रोच को आगे बढ़ाने वाला है। लेकिन अप्रोच कब आगे बढ़ती है, हम कब आगे बढ़ते हैं, जब हम कर्मशील हों, ज्ञानशील हों, और उसके साथ साथ सबसे बड़ी बात, हम अपने मन के भाव को समर्पित करने वाले हों। और मन का भाव समर्पित कब होता है, जब हम मुस्कुराते हैं। कई बार हम इतने ज़्यादा सीरियस हो जाते हैं ज़िंदगी के प्रति कि हम ये नहीं समझ पाते कि हम क्या करने वाले हैं। क्या हमारा उद्देश्य केवल एक गंभीर चिंतन है? नहीं, चिंतन भी तभी अच्छा लगता है जब चेहरे पर मुस्कुराहट हो। आपसे एक रिक्वेस्ट है, प्रार्थना है, गुजारिश है, एक बार मुस्कुराइए और अपने लिए तालियां बजाइए जोरदार। हमने जिंदगी में हर इंसान के लिए तालियां बजाई जो हमको प्रभावशाली लगा, इफेक्टिव लगा। लेकिन कभी हमने ये नहीं सोचा कि हम कहां खड़े हैं, क्या हमारे लिए भी कोई ताली बजाने वाला होगा? अगर कोई नहीं मिले तो आप आईने में खुद को देखकर अपने लिए तालियां बजाइए, आपके लिए नए रास्ते खुलते चले जाएंगे। श्री कृष्ण ने कहा है, कर्म ही जीवन का प्रधान है। बिना कर्म के इंसान अपनी मंजिल नहीं पा सकता। और कर्म यह कहता है कि आप जिस सपने को बुनकर आए हैं, जिस सपने को सोचकर आए हैं, उस रास्ते पर चलना शुरू कीजिए। सपना आपको मंजिल तक पहुंचाएगा, लेकिन आपका कर्म पूरी ईमानदारी के साथ होना चाहिए। सभी जानते हैं कृष्ण के बारे में। कृष्ण हमारा आधार हैं। जग में सुंदर हैं दो नाम, चाहे कृष्ण कहो या राम। आजकल लोग राम और कृष्ण को भूलकर उस दुनिया की ओर जा रहे हैं जहां सिर्फ अंधकार है, रोशनी नहीं है। लेकिन जब हमें थोड़ा सा रोशनी की तरफ बढ़ना होता है तो हमें वापस कृष्ण और राम पर आना होता है। और आज हम उसी रोशनी में आपको ले जाने के लिए तैयार हैं, जहां वृंदावन से भगवान कृष्ण अपने प्रेम, भक्ति और आनंद के साथ आपको यहां ले जाने वाले हैं। वृंदावन से पधारे कान्हा एकेडमी के सभी कलाकारों की ओर से आपका अभिनंदन, स्वागत। कम समय है, लेकिन उस कम समय में हम आपके दिल में, आपके मन में वो भक्ति, प्रेम और आनंद को भरना चाहते हैं जिससे आपके जीवन में एक नई ऊर्जा का संचार हो, आपके चेहरे पर मुस्कुराहट हो और आपको लगे कि जिंदगी का सही मायने क्या है, क्योंकि कृष्ण की मुस्कुराहट ही जिंदगी का आधार है। आइए हम इस प्रोग्राम का स्टार्ट कर रहे हैं, श्री गणेश कर रहे हैं। हमारे साथ हैं कान्हा एकेडमी के डायरेक्टर डॉक्टर अनूप शर्मा साहब और उनकी पूरी टीम। हमारे म्यूजिशियन श्री दिनेश मिश्रा जी, कीबोर्ड प्लेयर। उनके साथ हैं योगेश शर्मा जी ढोलक पर, सचिन शर्मा जी पैड पर, शालिनी शर्मा जी जिनकी आवाज आप सुनेंगे। साथ में हैं हमारे दीपक जी, पूरी टीम को कोऑर्डिनेट करते हैं। मैं आशु शर्मा। लेकिन इन सब में सबसे महत्वपूर्ण बात है कि ये पूरी टीम को संगठित कौन कर रहा है, टीम को आगे कौन ले जा रहा है, लीडर कौन है, और वो लीडर सिर्फ एक ही है, कान्हा एकेडमी से डॉक्टर अनूप शर्मा साहब, वृंदावन की पावन धरा से। आइए मिलते हैं भगवान कृष्ण से आपके प्रेम के साथ। श्रीमती शालिनी शर्मा।
हेलो, श्री राधे, श्री राधे, जय जय श्री राधे। वक्रतुंडे महाकाय सूर्यकोटि समप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा। मंगलं भगवान विष्णु, मंगलं गरुडध्वज, मंगलं पुंडरीकाक्ष, मंगलाय तनोहरि। सर्वमंगल्ये मांगल्ये शिवे सर्वार्थ साधिके, शरण्ये त्र्यंबकं गौरी नारायणी नमस्तुते। गुरुर्ब्रह्मा गुरुर्विष्णु गुरुर्देवा महेश्वरा, ध्यानमूलं गुरुमूर्ति पूजामूलं गुरुपदं मन्त्रमूलं गुरुवाक्यं मोक्षमूलं गुरुकृपा अखण्डमण्डलाकारं व्याप्तं येन चराचरं तत्पदं दर्शितं येन तस्मै श्री गुरुवे नमः तस्मै श्री गुरुवे नमः तस्मै श्री गुरुवे नमः।
मैं एक निवेदन आपसे करूंगा, जब हमारे मन का चाहने वाला कोई अपना आता है तो हम बड़े प्रसन्न होते हैं, खुश होते हैं। आज दुनिया को अपना बनाने वाला एक नाम आ रहा है, कृष्ण आ रहे हैं वृंदावन से। एक बार अपने दोनों हाथ ऊपर कर लीजिए, अपने आप को भगवान की शरणागत हो जाइए और प्रेम से बोलिए श्री बांके बिहारी लाल की जय जय श्री। आइए, चेहरे पर मुस्कान, हाथों में बांसुरी और उस बांसुरी को अपने अधरों पर सजाते हुए श्री कृष्ण राधा के साथ आ रहे हैं आपसे मिलने के लिए।
जब से राधा श्याम की नैन हुए हैं जान, श्याम बने हैं राधिका, राधा बन गई श्याम, श्याम बने हैं राधिका, राधा बन गई श्याम। कि ब्रज 84 कोस की परिक्रमा एक दे तो 184 योनि के संकट हरि हर ले। ओ जय जय ब्रज भूमि, जय जय ब्रज भूमि, जय जय ब्रज भूमि, हाँ जय जय ब्रज भूमि, जय जय ब्रज भूमि, जय जय ब्रज भूमि, जय जय ब्रज भूमि। हो चारों धामों से निराला ब्रज धाम की दर्शन कर लियो जी, चारों धामों से निराला ब्रज धाम की दर्शन कर लियो जी। दर्शन कर लियो, सुमिरन कर लियो, दर्शन कर लियो, सुमिरन कर लियो, वंदन कर लियो जी। होली की राधा जी बिहारी जी को नाम की दर्शन कर लियो जी, चारों धामों से निराला ब्रज धाम की दर्शन कर लियो जी। गोकुल में कौना मथुरा से आयो, गोकुल में माखन चोरी खायो, गोकुल में हो माखन खायो यानी बाटी हूँ खाई, पकड़ो यशोदा ने तो दीनी सफाई। मैया मुरी कसम तुझी मैं नहीं माखन खायो, ओ मैया मुरी कसम तुझी मैं नहीं माखन खायो। मुख में धरोई ब्रह्मांड दिखायो, ऐसे मोहन को सहर्ष प्रणाम की दर्शन कर लियो जी। चारों धामों से निराला ब्रज धाम की दर्शन कर ले। श्री राधे। बरसाने की राधा राधा बोल, जिसने मोहन को बांधा राधा राधा बोल। बरसाने की राधा राधा बोल, जिसने मोहन को बांधा राधा राधा बोल। होरी खेले तो आजा कृष्ण मुरारी, बरसाने में बुलाई गई राधा प्यारी। होरी खेले तो
आजा कृष्ण मुरारी, बरसाने में बुलाई गई राधा प्यारी।
हो कान्हा बरसाने में आई जइयो, बुलाई गई राधा प्यारी। कान्हा बरसाने में आई जइयो, बुलाई गई राधा प्यारी। बुलाई गई राधा प्यारी, बुलाई गई श्यामा प्यारी। बुलाई गई राधा प्यारी, बुलाई गई श्यामा प्यारी। हो कान्हा बरसाने में आई जइयो, बुलाई गई राधा प्यारी। कान्हा बरसाने में आ जइयो,
बुलाई गई राधा प्यारी।
दोनों एक दूजे के प्रेम पुजारी, दोनों मिल के बने हैं एक नाम की दर्शन कर लियो जी। चारों धामों से निराला ब्रज धाम की दर्शन कर लियो जी। ओ दर्शन कर लियो, सुमिरन कर लियो।
दर्शन कर लियो, सुमिरन कर लियो,
वंदन कर लियो जी। बोले के राधा जी बिहारी जी को नाम के दर्शन कर लियो जी। चारों धामों से निराला ब्रज धाम के दर्शन कर लियो जी। चारों धामों से निराला ब्रज धाम के दर्शन कर लियो जी।
चारों धामों से निराला ब्रज धाम की दर्शन कर लियो जी।
चारों
धामों से निराला ब्रज धाम की दर्शन कर लियो जी।
जय जय ब्रज भूमि, जय जय ब्रज भूमि, जय जय ब्रज भूमि,
जय जय ब्रज भूमि, जय जय ब्रज भूमि, जय जय ब्रज भूमि, जय जय ब्रज भूमि, जय जय ब्रज भूमि, जय जय ब्रज भूमि, जय जय ब्रज भूमि।
आज का आगाज है पावन धरती ठाकुर की, श्री वृंदावन धाम है पावन भूमि ठाकुर की, श्री वृंदावन धाम है पावन भूमि ठाकुर की, श्री वृंदावन धाम है पावन भूमि ठाकुर की, श्री वृंदावन धाम है।
आपकी तालियां निश्चित रूप से ये बता रही हैं कि आपके हृदय में एक प्रेम की ज्योत जग गई है। एक ऐसा दीपक जलने वाला है जिसकी रोशनी से आपका मन और आपका घर परिवार हमेशा हमेशा के लिए रोशन हो जाएगा। लेकिन ये रोशन तब और होगा जब कृष्ण के प्रति आप अपना पूर्णतः समर्पण करेंगे। और समर्पण कब होगा? जब एक बार फिर से दोनों हाथ उठाएंगे। आपको पता है जब ये दोनों हाथ उठते हैं तो ईश्वर क्या करते हैं? अपने बच्चों के हाथों को पकड़ते हैं। लेकिन हम कई बार, सर हाथ ऊपर ही रखिएगा, लेकिन कई बार हम गलत जगह हाथ उठा देते हैं जहां से सिर्फ हमें केवल धोखा मिलता है। लेकिन परम पिता परमेश्वर के सामने हाथ जब आप उठाएंगे तो उनके साथ एक संबंध बनेगा विश्वास का, और आवाज आएगी। दोनों हाथ उठा के बोलिए, बोले कृष्ण कन्हैया लाल की जय जय श्री। वृंदावन कौन कौन गया है? श्री धाम वृंदावन जा चुके हैं। निश्चित रूप से एक बड़ी अनुभूति हुई होगी। हमारे सर बैठे हैं सामने, सोच रहे हैं मेरा भी मन कर रहा है वृंदावन जाने का, लेकिन अगर आप तन से नहीं जा पाए, फिजिकली नहीं जा पाए, तो आप मानसिक रूप से जरूर वृंदावन जाएंगे, यह हमारा वादा है। और जो मन से काम किया जाता है उसमें जो आनंद मिलता है उसको शब्दों में बयां करना बड़ा मुश्किल है। लोग प्रैक्टिकल करते हैं, लेकिन प्रैक्टिकल तभी अच्छा लगता है जब उस प्रैक्टिकल में कोई जुनून हो, पागलपन हो, तब वो प्रैक्टिकल आपको मंजिल तक ले जाता है। आज हम यहाँ युवाओं के साथ बैठे हैं जो देश का भविष्य हैं। भगवान ने कहा है कि अपनी जिंदगी में इतने ऊर्जावान, इतने एनर्जेटिक बनो कि आपको देखकर आपकी ओर लोग खिंचे चले आएं, ऐसा काम करो। और भगवान के लिए ताली बजाना यह सबसे अच्छा काम है। एक बार ईश्वर के लिए ताली फिर बजाइए। आइए, सबने कृष्ण और राधा के बारे में सुन रखा है। ये वो नाम नहीं है जो अनजाना नाम हो। कृष्ण प्रेम के एक बहुत बड़ा अध्याय है जिसके हर पन्ने पर प्रेम का, भक्ति का शब्द लिखा है, लेकिन राधा उसमें नाम ना जुड़े तो वो भक्ति अधूरी रह जाती है। आज बरसाने वाली हमारी राधा रानी, जिनके प्रेम में कृष्ण बरसाने में नृत्य कर रहे हैं, और राधा रानी प्रेम के आनंद को कृष्ण के साथ आइए प्रेजेंट कर रही हैं। मैं बरसाने की छोरी। इसको आपको लगेगा कि हम इस समय प्रयाग में नहीं हैं, वृंदावन पहुंच चुके हैं। आनंद लीजिए इस खूबसूरत प्रस्तुति का। कान्हा एकेडमी वृंदावन।
मोते बरजोरी, मैं बरसाने की छोरी, नगर मोते बरजोरी, मैं बरसाने की छोरी, नगर मोते बरजोरी, तू कारो और मैं गोरी, अपनों मेल नहीं।
मैं तो से बांधू प्रीत की डोरी, करता खेल नहीं।
ना ना मैं बरसाने की छोरी, ना कर मोते बरजोरी, तू कारो और मैं गोरी, अपनों मेल नहीं।
मैं तो ते बांधू प्रीत की डोरी, करता खेल नहीं।
आहा मैं बरसाने की छोरी, ना कर मोते बरजोरी, तू कारो और मैं गोरी, अपनों मेल नहीं।
मैं तो ते बांधू प्रीत की डोरी, करता खेल नहीं।
अरे मैं बरसाने की छोरी।
आपकी तालियों से आप ज्वाइन कीजिए भगवान को, दोनों हाथ खोलिए तालियों के साथ। क्या बात है, ये ऑडिटोरियम प्रेम के रंग में रंग जाना चाहिए। श्री राधे।
शुक्र करो के पड़े नहीं जसुदा मैया के डंडे।
अच्छा।
एक डांट में है जाते अरमान तुम्हारे ठंडे। शुक्र करो के पड़े नहीं जसुदा मैया के डंडे। एक डांट में है जाते अरमान तुम्हारे ठंडे।
मैं नंद बाबा का लाला, मैं तो ना डरने वाला, माखन खाना, शर्मा है श्याम दीवाना, करता खेल नहीं।
अरे तू गोकुल में बरसानो अपनों मेल नहीं, मैं बरसाने की छोरी ना कर मोते बरजोरी, तू कारो पर मैं गोरी अपनों मेल नहीं।
मैं बांधू तो से प्रीत की डोरी, करता खेल नहीं।
ना ना मैं बरसाने की छोरी ना कर मोते बरजोरी, तू कारो और मैं गोरी अपनों मेल नहीं।
मैं तो से बांधू प्रीत की डोरी, करता खेल नहीं।
अरे मैं बरसाने की छोरी।
एक बार फिर से आपकी आवाज, जय जय श्री। किन किन के पास अपनी आवाज है, वॉल्यूम है, बताइए जरा। जय जय श्री। कुछ लोगों के पास आवाज तो है लेकिन वो एक्सप्लेन नहीं करना चाहते, बोले क्यों बोले, हम मतलब की बात होगी तो बोलेंगे। लेकिन यकीन मानिए ये मतलब की नहीं, बहुत महत्वपूर्ण बात है। जब आपके मुंह से किसी के नाम की जय जयकार निकलती है और वो भी प्रभु की, तो दुनिया में आपके नाम की जय जयकार होती है। इसलिए जरा दोनों हाथ उठाइए, प्रेम से बोलिए बंसी वारे की। अग्रिम पंक्ति में जो हमारे आदरणीय बैठे हैं, सर बचपन आपका भी रहा है और बचपन यहां भी बैठा है। बचपन की अपनी एक खास बात होती है, एक सहजता होती है, एक मासूमियत होती है, और उस मासूमियत में ऐसा लगता है दुनिया में हम ऐसा क्या कर दें कि लोग हमारी तरफ देखें, पागलपन होता है। लेकिन जब हम बड़े हो जाते हैं, समझदार हो जाते हैं, तो कई बार लोग कहते हैं ज़्यादा समझदार हो गया है, पागल हो गया है। कभी कभी बच्चा बन जाना चाहिए। तो कौन कौन बच्चा बनकर यहां आनंद लेना चाहता है, जरा ताली बजाइए। अरे वाह, क्या बात है। एक छोटा सा क्वेश्चन मैं आपसे कर रहा हूं, मेरे सीनियर्स बैठे हैं, मेरी मातृ शक्तियां यहां बैठी हैं, सवाल का जवाब जरूर दीजिएगा। मिश्रा जी, किस किसने, यहां यूथ भी बैठा है, सीनियर्स भी हैं, हमारी मातृ शक्तियां भी हैं, किस किसने अपने जीवन में प्रेम किया है, हाथ उठा दीजिए। वन, ओहो क्या बात है, लवली लवली। मुझे ऐसा लगता है सर की डायरेक्ट कॉल चल रही है बिहारी जी से। सर ओके ओके, अच्छा है, ये ज़माना ही सर इसका है, इसके बिना तो कुछ हो ही नहीं सकता। लेकिन जब हम लाइव हों तो इसको छोड़ देना चाहिए। थैंक यू सर। एक बार दोनों हाथ उठाइए जिसने प्रेम किया है। और मुझे लगता है मेरे सवाल को लगभग समझ चुके हैं सभी, सबने प्रेम किया है। यकीन मानिए प्रेम जिंदगी की वो धरोहर है, प्रेम जिंदगी का वो तोहफा है जो हमें माँ से मिलता है। जब पहली बार माँ का स्पर्श बच्चे के साथ होता है, वहीं से प्रेम की शुरुआत होती है। मातृत्व झलकता है, स्नेह झलकता है, और माँ बताती है ये तेरे पिताजी हैं, ये तेरे भाई हैं। वहाँ से प्रेम शुरू होता है। लेकिन हम प्रेम को कई अलग दिशा में जब ले जाते हैं तो वो प्रेम जिंदगी के लिए नासूर बन जाता है। लेकिन वही प्रेम जब हम ईश्वर से करने लगते हैं तो हमारा परिवार मुस्कुराता है। और मैं चाहूँगा आपका परिवार मुस्कुराता रहे। तो अगर जो हमारा यूथ यहाँ बैठा है कृष्ण को चाहता है, प्रेम करता है, हमारे सीनियर्स बैठे हैं, तो मैं आप सबको इन्वाइट कर रहा हूँ 2 मिनट के लिए यहाँ आइए, ब्रज के उस आनंद में रंग जाइए जिस आनंद को लेने के लिए दुनिया जाती है। कॉल कर रहा हूँ मैं आपको, आइए थोड़ा सा एंजॉय ठाकुर जी के साथ वृंदावन चलते हैं। आइए। लीड कौन करेगा ये देखने वाली बात होगी, लीड कौन करेगा।
तालियां ही नहीं बजा दी है, थोड़ा सा उठ के ठाकुर जी के साथ आना भी है। तो जो पीछे जिन्हें लगता है कि वो ठाकुर जी की गोपियां हैं और कान्हा के साथ आनंद लेना चाहती हैं, वो उठकर आएंगी और आनंद लेंगे।
कई बार हमने ये भी देखा है, उम्र 55 की लेकिन दिल बचपन का। तो अपने को 55 बना लीजिए, 2 मिनट उठ के आ जाइए, सभी सीट छोड़ दीजिए 2 मिनट के लिए। आइएगा सर प्लीज।
कि शाम देखा घनी शाम देखा, शाम देखा घनी शाम देखा, राधा राधा राधा।
क्या बात है,
राधा राधा राधा, अरे राधा ढूंढ रही किसी ने मेरा शाम देखा,
राधा ढूंढ रही किसी ने मेरा शाम देखा,
अरे शाम देखा घनी शाम देखा, शाम देखा घनी शाम
देखा घन श्याम देखा, श्याम देखा घन श्याम देखा।
अरे राधा ढूंढ रही किसी ने मेरा श्याम देखा,
अरे राधा ढूंढ रही किसी ने मेरा श्याम देखा।
राधे दीवाने हो गए कृष्ण के रंग गए। दो करके
जय हो।
क्या बात है मैम। ओहो ये प्रेम का रंग है। राधे सर्कल।
जमुना किनारे मेरी ऊंची हवेली,
जमुना किनारे मेरी ऊंची हवेली,
अली में बेचती गोपिका नवेली,
रंगी गोपिका नवेली,
अली राधा रंगली मेरो नाम ये हसपति रहे जहियो, जमुना किनारे मेरो गाँव सावरी
आए जहियो,
जमुना किनारे मेरो गाँव।
यार है
दीवाने हो गए कृष्ण के चूंदर उड़ गई। राधे ओहो
वृंदावन जाओगी सखी, वृंदावन जाओगी।
ओ मेरे उठे, ओ मेरे उठे, ओ मेरे उठे पीर की पीर सखी, वृंदावन जाऊंगी।
मेरे उठे की पीर सखी, वृंदावन जाओगी।
श्री राम
प्रयागराज। हरि बोल।
छोटी छोटी गैया छोटी छोटी ग्वाल,
छोटी छोटी गैया छोटी छोटी ग्वाल,
छोटी छोटी गैया छोटी छोटी ग्वाल,
छोटी छोटी गैया छोटी छोटी ग्वाल।
मेरो मदन गोपाल,
हरी छोटा सो मेरो मदन गोपाल।
कारी कारी गैया गोरे गोरे ग्वाल,
कारी कारी गैया गोरे गोरे ग्वाल।
ओ श्याम वरण मेरो मदन गोपाल,
हरी श्याम वरण मेरो मदन गोपाल।
छोटी 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 गैया छोटी छोटी ग्वाल,
छोटी छोटी गैया छोटी छोटी ग्वाल। छोटो सो मेरो मदन गोपाल।
अरे आएंगे बिहारी चले आएंगे बिहारी,
आएंगे बिहारी चले आएंगे बिहारी,
अरे आएंगे बिहारी चले आएंगे बिहारी,
आएंगे बिहारी चले आएंगे बिहारी।
अरे राधे 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 जपो चले आएंगे बिहारी,
राधे राधे जपो चले आएंगे बिहारी,
राधे राधे जपो चले आएंगे बिहारी,
राधे राधे जपो चले आएंगे बिहारी।
और सखियों
राधा रानी भोली भाली चंचल बिहारी,
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 राधा रानी भोली भाली चंचल बिहारी।
राधा रानी भोली भाली चंचल बिहारी,
राधा रानी भोली भाली चंचल बिहारी,
राधा रानी भोली भाली चंचल बिहारी।
अरे राधे 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 जब चले आएंगे बिहारी,
राधे राधे जब चले आएंगे बिहारी, राधे राधे जब चले आएंगे बिहारी, राधे राधे जब चले आएंगे बिहारी, राधे राधे जब चले आएंगे बिहारी, राधे राधे जब चले आएंगे बिहारी, चले आएंगे बिहारी, चले आएंगे चले आएंगे
थोड़ा सा स्ट्रेस कम हुआ, जिसका स्ट्रेस थोड़ा सा कम हुआ है, रिलैक्स हो गए हैं, तो एक बार फिर से दोनों हाथ उठाइए, बोलिए बांके बिहारी लाल की। जिंदगी में ये बहुत ज़रूरी है अपने आप को उस स्ट्रेस से बाहर निकालना। मुस्कुराइए, हंसिए, किसी से बात कीजिए, अपने मन की बात को शेयर कीजिए। आज राधा रानी ने अपने मन की बात कृष्ण से कही है। आप बहुत सुंदर हैं ठाकुर जी, लेकिन मैं चाहती हूँ जिस प्रकार आपके मुकुट पर एक मोर पंख धारण जब हो जाता है तो वो अत्यंत दिव्य लगता है। मैं चाहती हूँ कि आप मोर स्वयं बनकर आएँ, पीकॉक का रूप धारण। भगवान कृष्ण मोर बने हैं, और वो कैसे बने हैं, आइए समय न ज़्यादा न लेते हुए आपको वो दर्शन करा रहे हैं जिनके दर्शनों के लिए लोग पागल होकर, दीवाने होकर वृंदावन आते हैं, बरसाने आते हैं। आज श्री राधा रानी बरसाने में भगवान कृष्ण का इंतजार कर रही हैं। आइए।
आज गोपाल लिए ब्रज कोल, बांस बसो वंशी वट छैया, बीन पखा बज बांसुरी की धुन, आनंद सिंधु कदम्ब के ठैया। भाव उठो हे राधिका के मन, मोर नचे नंद राज के छैया। देख प्रिया जुगी दीन दशा, बन आए गए हैं मोर कन्हैया। एक दिन श्री कुंवरी राधिका मोर कुटी आई, एक दिन श्री कुंवरी राधिका मोर कुटी आई। नित मयूर ने कौ देखने मने उत्कंठा छाई, नित मयूर ने कौ देखने मने उत्कंठा छाई। मोर वहाँ मोर वहाँ एको नहीं पायो जी, मोर वहाँ एको नहीं पायो। जान प्रियावन प्रीतम प्यारो मोर बनी आयो, हाँ जान प्रियावन प्रीतम प्यारो मोर बनी आयो।
भगवान दौड़े चले आ रहे हैं मोर बनकर।
मोर बन आयो रे सिया, ओ बरसाने ओ बरसाने ओ बरसाने की मोर कुटी में मोर बन आयो रे सिया। ओ
बरसाने की मोर कुटी में मोर बन आयो रे सिया। आयो रसिया मोर बन आयो रसिया, आयो रसिया मोर बन आयो रसिया। ओ बरसाने ओ बरसाने
ओ बरसाने की
मोर कुटी में
मोर बन आयो रे सिया।
ओ बरसाने की मोर कुटी में मोर बन आयो रे सिया। कितना सुंदर दृश्य है, भगवान जब मोर बनकर आए हैं, राधा जी नृत्य कर रही हैं।
एक दिन बोली राधा प्यारी,
एक दिन बोली राधा प्यारी,
दो दिन तीन मिले मिले मुरारी,
2 दिन के न मिले मुरारी।
अरे बिना श्याम सुंदर दर्शन के प्यासे अखियां,
बिना श्याम सुंदर दर्शन के प्यासे अखियां।
उबरसाने
राधे
उबरसाने
राधे
उबरसाने की मोर कुटी में मोर बन आयो रे सियाम। उबरसाने की मोर कुटी में मोर बन आयो रे सियाम। श्री राधे।
ब्रज की सारी सखियां कृष्ण के प्रेम में मोरनी बन गई हैं।
मोरा बन गई मदन मुरारी,
मोरा बन गए मदन मुरारी।
ऐसो नृत्य कियो है मनोहारी,
ऐसो नृत्य कियो है मनोहारी।
अरे मोरा बन गए मदन मुरारी,
मोरा बन गए मदन मुरारी।
ऐसो नृत्य कियो है मनोहारी,
ऐसो नृत्य कियो है मनोहारी।
राधा कभी कृष्ण है राधा, राधा
कभी कृष्ण है राधा।
अरे रूप रंग रहस्य युग रास में साधा,
रूप रंग रहस्य युग रास में साधा।
अरे अष्टमी का आय हाय अष्टमी का चंद्र दोनों ओर राधा, अष्टमी का चंद्र दोनों ओर राधा। पवन मंद मंद तेरा कोई भी बाधा,
पवन मंद मंद तेरे घोर है आधा।
अरे हिल मिल के हिल मिल के दोनों नाचे श्याम और राधा,
हिल मिल के दोनों नाम श्याम है राधा।
अरे श्याम और राधा घनी श्याम और राधा,
श्याम और राधा घन श्याम और राधा।
श्याम और राधा घनी श्याम और राधा,
श्याम और राधा घन श्याम और राधा।
मोहित है रे या चुबे ऊपर ब्रज की सखियां, मोहित है रे या चुबे ऊपर ब्रज की सखियां। उबरसाने 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 की मोर कुटी में मोर बन आयो रे सियां।
बरसाने की मोर कुटी में मोर बन आयो रसिया। मुस्कुरा रहे हैं श्याम सुंदर।
बजता मोरा तुम तन नन, बजता मोरा
तुम तन नन,
बजता मोरा तुम तन नन,
बजता मोरा तुम तन नन।
तुम तिगदा दिग थे, तुम तिगदा दिग थे, तुम तिगदा दिग थे, तिगदा दिग थे, तिगदा दिग थे। बजता मोरा तुम तन नन,
बजता मोरा तुम तन नन।
पायल बज रही छुम छुम चनन, पायल बज रही छुम छुम चनन, छुम 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 चनन, छुम 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 चनन, छुम 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 चनन, छुम 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 चनन। पायल बज रही छुम छुम चनन,
पायल बज रही छुम छुम चनन।
सप्त सुरन सो नारायण की बाजी बांसुरिया, सप्त सुरन सो नारायण की बाजे बांसुरिया। ओ बरसाने की मोर कुटी में मोर बन आयो रे सियाम। ओ बरसाने की मोर कुटी में मोर बन आयो रे सियाम।
विष्णु अवतार योगी राज कृष्णा सुदर्शन चक्रधारी 16 कलाओं से अवतरित योगी राज कृष्णा, दर्शन कीजिए अद्भुत दर्शन।
कदम पर बैठे,
उड़ उड़ मोर कदम पर बैठे,
अरे उड़ उड़ मोर कदम पर बैठे,
उड़ उड़ मोर कदम पर
बैठे हैं, वो बैठे हैं, हाँ बैठे हैं पंख मरूर मरूर को शोर भयो भारी वृंदावन बोले मोर चकोर गोकुल में शोर भयो हरि
अद्भुत है भगवान का दर्शन।
टप टप पेच गिरे यमुना में,
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 दिन बीने नंद किशोरी किशोरी गोकुल में शोर भयो भारी वृंदावन बोले मोर चकोरी गोकुल में शोर भयो भारी, गोकुल में शोर भयो भारी,
गोकुल में शोर भयो भारी,
गोकुल में शोर भयो भारी,
गोकुल में शोर भयो भारी,
ओ गोकुल में शोर भयो भारी, गोकुल में शोर भयो भारी, वृंदावन ओ वृंदावन आ वृंदावन बोले मोर चकोरी गोकुल में शोर भयो भारी,
वृंदावन बोले मोर चकोर गोकुल में शोर भयो भारी।
राधे राधे बोलो राधे राधे बोलो राधे राधे राधे
राधे,
श्यामा श्यामा बोलो श्यामा श्यामा बोलो श्यामा श्यामा श्यामा,
राधे राधे बोलो राधे राधे बोलो राधे राधे राधे,
जरा ताली बजा लीजिए।
श्यामा श्यामा बोलो
राधे,
बोलो श्यामा श्यामा श्यामा,
राधे 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 राधे
राधे राधे राधे
राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे राधे
जय जय श्री
राधे 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 राधे
जय जय श्री राधे जय जय श्री राधे।
किसी दृश्य को देखकर, किसी इमेज को देखकर हमारे चेहरे पर अगर खुशी आ जाए, वो दृश्य बहुत खूबसूरत है, और अगर हाथों से तालियां बजने लग जाएं तो दिव्य है, अलौकिक है। प्रेम से बोलिए जय जय श्री। कितनी सुंदरता है भगवान के इस दृश्य में। आप हाथ उठाकर बताएंगे, जिनके हाथ ऊपर उठेंगे ऐसा लगेगा कि आप हृदय से भगवान के इस आनंद को स्वीकार कर पा रहे हैं। प्रेम से बोलिए जय जय श्री। लगभग 132 देशों से प्रोफेसर्स यहां आए और अपने अपने एक्सप्लेन करेंगे अपने अपने विषय पर। 2 लाइन कहना चाहूंगा ऐसे लोगों के लिए जिनसे हम कभी मिले नहीं सर, जिनके बारे में केवल सुना है, लेकिन जब हम उनके विचारों से सहमत हुए तो एक शेर के रूप में मैं उनको अवगत करा रहा हूं। खूबसूरत सा पल एक किस्सा बन जाता है, कौन कब न जाने जिंदगी का हिस्सा बन जाता है। लोग मिलते हैं कुछ इस तरह से जिनसे कभी न टूटने वाला एक मजबूत रिश्ता बन जाता है। यह रिश्ता ही तो है। अगर यह रिश्ता ना होता तो आज यहां नहीं बैठे होते। सबकी अपनी अपनी दुनिया है, सब अपने अपने क्षेत्र के प्रभावशाली लोग हैं, एक्सपर्ट्स हैं, लेकिन सबसे बड़ा एक्सपर्ट्स अगर कोई यहां है जिसको देखने के लिए हम बैठे हैं तो वो कृष्ण हैं, जिसके सारे कलाकार बेकार हैं जिसके आगे। समूचा ज्ञान बेकार है। जिसने भगवत गीता का ज्ञान दे दिया हो उसके आगे ज्ञान की क्या चलती है, उसके आगे तो बस सहजता चलती है। हर पेरेंट्स अपने बच्चे को डॉक्टर, इंजीनियर, एक बड़ा कोई कलाकार बनाना चाहता है, लेकिन एक गुजारिश करूंगा जितने भी पेरेंट्स यहाँ बैठे हैं, अपने बच्चों को शिक्षा दीजिए, ज्ञान दीजिए, ऊंचाइयों तक ले जाने की क्षमता दीजिए, लेकिन जिंदगी में उन्हें सहज सरल होना सिखाइए। आप सरल बनिए, सहज बनाइए, मुस्कुराते हुए जीवन गुजारिए, मंजिलें आपकी होंगी। प्रेम से बोलिए राधे राधे। और एक बात और कहना चाहूँगा, वृंदावन जो गए हैं फिर से एक बार हाथ उठाएंगे। श्री धाम वृंदावन, क्या बात है। जो लोग जाना चाहते हैं वो भी हाथ उठाइए। अब सोच रहे होंगे भाई कौन सी ऐसी व्यवस्था होगी जिससे यहाँ वृंदावन जाएं। व्यवस्था आपके मन की है, आपके हृदय की है, जो आपको यहाँ से मन की गति से वृंदावन ले जाएगी। श्री बांके बिहारी जी का नाम सुना है? प्रेम से बोलो श्री बांके बिहारी लाल की। आप जब वहाँ गए होंगे ये सोचकर, श्री हरिदास जी महाराज ने अपने प्रेम के भाव से उन्हें प्रकट किया था। एक मूर्ति स्थापित हुई थी श्री बांके बिहारी जी के नाम से। दुनिया में कृष्ण के अनेक विग्रह हैं, अनेक मूर्तियां हैं, लेकिन बिहारी जी का विग्रह अपने आप में आकर्षित करने वाला है। क्यों? क्योंकि उनके जो नैन है ना, उनमें समस्त ब्रह्मांड समाया है। और जब हम नैनों को देखते हैं तो दिल से आवाज आती है, कजरारे तेरे मोटे मोटे नैन नजर न लग जाए। हमारे सर बैठे हैं सामने। सर, यकीन मानिए आपकी चेहरे की मुस्कान मुझे अपनी ओर खींच रही है। पता है क्यों सर? मुझे नहीं लग रहा कोई एक बहुत बड़ा व्यक्ति बैठा है सामने, ऐसा लग रहा है कोई एक बच्चा बैठा है गोपाल का रूप धारण करके। क्या बात है सर, एक्सीलेंट। जब घर के बुजुर्ग मुस्कुराते हैं ना तो घर का आंगन मुस्कुराता है, लेकिन जब घर के चेहरे बेजल हो जाते हैं तो घर की तरक्की रुक जाती है। इसके लिए बच्चे अपने बुजुर्गों को, सीनियर्स को मुस्कुराने का मौका दीजिए, उनके साथ 2 पल बिताइए, आपकी जिंदगी आराम से गुजर जाएगी। लेकिन आज हम पर्दे के पीछे जिनको देखने वाले हैं, स्वयं बिहारी जी आ रहे हैं प्रयागराज में। 132 देशों से प्रोफेसर्स आए, लेकिन केवल एक आ रहा है कलाकार बिहारी जी बनकर वृंदावन से। निवेदन, जिस प्रकार भक्त भगवान के लिए कुर्सी छोड़ देता है, अगर हो सके तो एक बार अपनी जगह छोड़ दीजिए, खड़े हो जाइए 2 मिनट के लिए। यह भाव है, समर्पण है, डेडिकेशन है, और जहां डेडिकेशन है वहां जिंदगी है। थोड़ा करीब आ जाएंगे, थोड़ा सा करीब, क्योंकि गोल्ड बहुत महंगा हो गया है, सब चाहते हैं गोल्ड लें। अब सुनिए, रहोगे दूर तो खोना ही खोना है, वहां पीतल है, यहां सोना ही सोना है, प्योर गोल्ड है। आगे आ जाइए सर, आगे आ जाइए। और मेरे सर ने बहुत अच्छी बात की है, अपने शूज खोल दिए हैं भगवान को पाने के लिए। मुझे लगता है अगर कोई ऐसा भाव है तो हो सकता है 2 मिनट के लिए अपने सैंडल शूज रिलीज कर दीजिए, बड़ा आनंद आने वाला है। क्या बात है सर, आहा। अच्छा, धीरे धीरे करीब आ रहे हैं। सबने सुना है सर, मैं प्रणाम करता हूं आपको। एक बहुत बड़े व्यक्तित्व के रूप में आप यहाँ हैं। आपकी ज़िंदगी में आपका परिवार आपके साथ हो, और आपकी जितनी भी आयु है उसमें ठाकुर जी की कृपा से आप स्वस्थ रहें, ये मंगल कामना है आपकी। मैं चाहूँगा सब थोड़ा थोड़ा आगे आइए। कीर्तन सभी के घर में होता है, किस किस के घर में होता है? करते हैं ना? क्योंकि कीर्तन से भगवान प्रसन्न होते हैं, और भगवान प्रसन्न होते हैं तो घर में तरक्की होती है, आनंद आता है। आइए, पर्दा खुल रहा है, जिनको दर्शन हो जाएं।
मोटी मोटी
तो फूल ठाकुर जी के चरणों में जरूर अर्पण करेंगे।
हो मीठे मीठे बेनने के तू, सावरी सलोनी सूरत के तू, आई प्यारी प्यारी मूरत के तू, अरे सांवली सलोनी सूरत के तू, आई प्यारी प्यारी मूरत सूरत के तू हो गजरारी मोटे मोटे तेरे
जय हो। बोलिए बाके बिहारी लाल की।
हो गजरारी मोटी मोटी तेरी नैन हो नजर ना लग जाए
हाय हाय हाय
नजर ना लग जाए
होए होए होए
नजर ना लग जाए बाकी बिहारी गजरारी मोटे मोटे तेरे नैन हो नजर ना लग जाए
हाय हाय हाय
नजर ना लग जाए
होए होए होए
नजर ना लग जाए।
मैं रिक्वेस्ट करूंगा इस पूरी मीट के संयोजक श्री अंबर कुमार साहब से वो मंच पर आए। श्री अंबर कुमार साहब और हमारी मैम जिनकी वजह से कृष्ण के दर्शन कर पा रहे हैं, मधु शुक्ला जी, प्लीज मंच पर आइए। मधु शुक्ला जी।
ओ काजल की कुरी
हाय हाय हाय
काजल की कुरी
होए होए होए
मेरा जिगर मरूरी
होए होए होए
मेरा जिगर मरूरी
हाय
होए
हाय
होए रंग रस में बूरी पहले हारी रे गजरारी मोटे मोटे तेरे रे ओ नजर ना लग जाए
हाय हाय हाय
नजर ना लग जाए ओए होए होए नजर ना लग जाए।
आइए सर, स्पेशल दुपट्टा आया है बिहारी जी के दरबार से। आइए सर। श्री अंबा कुमार जी ठाकुर जी की नजर उतार रहे हैं, सभी के साथ भगवान के प्रेम में रंग रहे हैं।
ओ ब्रज का ये खिलौना
होए होए होए
ब्रज का ये खिलौना
होए होए होए
तेरा रूप सलोना
होए होए होए
तेरा रूप सलोना
हाय हाय हाय
करता रोटोना बलिहारी रे गजरारे मोटे मोटे तेरी नैन ओ नजर ना लग जाए
हाय हाय हाय
नजर ना लग जाए
होए होए होए
नजर ना लग जाए
बाके बिहारी गजरारे मोटे मोटे तेरे नैन
लग जाए
हाय हाय हाय
नजर ना लग जाए
होए होए होए
नजर ना लग जाए
होए होए होए
नजर ना लग जाए
होए होए होए
नजर ना लग जाए
हाय हाय हाय
नजर ना लग जाए
होए होए होए
नजर ना लग जाए।
जिनको दर्शन करके आनंद आ रहा है जरा हाथ उठाइए। कुछ लोग ऐसे भी वृंदावन में आते हैं, बिहारी जी के चरणों में अपनी तरफ से समर्पण करते हैं जो ठाकुर जी ने दिया है प्रेम के भाव। आइए थोड़ा सा वही खड़े होकर नृत्य करेंगे ठाकुर जी के चरणों में, बिहारी जी के चरणों में।
मनमोहन तेरे कर मुरली उर्वा
राधे
ये बालिक मोमन बसो
श्री अम्मा कुमार साहब आनंद के प्रस्तुत करता।
ये बालिक मोमन बसो खुशी सदा बिहारी लाल
झूमे के सभी
वृंदावन की इन कुंज गलिन में वृंदावन की इन कुंज गलिन में खुशबू बिहारी जी की आती है वृंदावन की
इन कुंज गलिन में वृंदावन की इन कुंज गलिन में
खुशबू बिहारी जी की आती है वृंदावन की।
थोड़ा झूम लीजिए अपनी जगह पे, ऐसा लगेगा वृंदावन में है आप लोग। ओहो राधे
ऐसी सुगंधे छाई है चहूरी रस को
ऐसी सुगंध छाई है चहुओरी, रसिकों को खींच लेती बात प्रेम डोरी।
मन को भुलाए मदहोश बनाए,
मन को भुलाए मदहोश बनाए।
हाँ दर पे बिहारी ले जाते हैं
वृंदावन की
इन कुंज गलिन में,
वृंदावन की
इन कुंज गलिन में हो खुशबू बिहारी जी की आती है, खुशबू बिहारी जी की आती है, खुशबू बिहारी जी की आती है,
खुशबू बिहारी जी की आती है।
खुशबू।
प्रेम से बोलिए राधे राधे। देखिए सर, फिर कहूंगा आपसे, बोलने को दी जुबान तुम बोलते क्यों नहीं, लेने को कृष्ण का नाम अपना मुंह खोलते क्यों नहीं। आज का न बोलना उस दिन याद आएगा जिस दिन बोलना तो चाहेगा मगर। तो आज प्रयागराज में मौका मिला है, आप सभी को एक बार हाथ उठाइए, प्रेम से बोलिए बाके बिहारी लाल की जय जय श्री। ये प्रेम का रंग है, जो चढ़ जाता है उतरता नहीं है। श्याम पिया मोरी रंग दे चुंदरिया, ऐसी रंग दे रंग नाई छूटे सारी उमरिया। अंबक सर ने जो प्रेम का रंग चढ़ा दिया है आप लोगों के ऊपर, एक कोई ना कोई ऐसा सूत्रधार होता है जिसके साथ पूरा परिवार बंध जाता है। अंबक कुमार जी के लिए एक बार जोरदार तालियां। और हमारी मधु शुक्ला मैम, एक बार मैम खड़ी हो जाइए, जिनकी वजह से आज दूसरी बार प्रयागराज की धरती पर कृष्ण आए हैं फिर से मिलने के लिए। कान्हा एकेडमी डॉक्टर अनूप शर्मा साहब और ये सारी टीम अब आपको उस ओर ले जा रही है जहां मस्ती, प्रेम, भक्ति और उन्माद होता है, जहां दिल की आवाज दिल से निकलकर सामने दिल तक टकराती है और उसे कहते हैं हार्ट टचिंग होली वृंदावन की। नंदगांव और बरसाने की वो होली जिसमें कपड़े नहीं रंगते, मन रंग जाता है, दिल रंग जाता है। आइए उस होली का आनंद लेते हैं। सभी जिसका मन करे वो आइएगा, होली खेलिएगा।
बरसाने होरी मची और खेल रहे नंदला, रंग गुलाल उड़ाते तो भैया नाच दे दे ताला।
पोजीशन शुरू। होली है।
अरे आई गई हुरिया रे, हुरी के आई गए हुरिया रे, ग्वालिन निकसा दिया गन में आए ग्वाल तेरे
द्वारे।
अरे किवारन की रे, किवारन की हो, किवारन की हो, कैसे ठाड़ी है, हाँ कैसे ठाड़ी है, हो कैसे ठाड़ी है उठ किवारन की। कि तू तो जाने कावे दी ससुराए गोरी,
तू तो जाने कावे
दिखत नहीं
ओ तो
गोरी।
नजरी लगी है इन ग्वालन की, कैसे ठाड़ी हाँ उठ किवारन की, कैसे ठाड़ी है
होठ किवारन की।
अरे नंदगाम को टोल आई गए हो सज धज के गोरी, नंदगाम को टोल आई गए हो सज धज के गोरी। अरे बरसाने में धूम मची है आए गई हो रे, बरसाने में
धूम मची है आए गई होली।
कि दिन में दिख जाए दारे, कि दिन में दिख जाए दारे, लाला सारे आंगन में आई ग्वाल तेरे द्वारे, लाला निक सारे आंगन में आई ग्वाल तेरे सारे, ओ छोरा निक सारे आंगन में आई ग्वाल तेरे सारे, लाला निक सारे आंगन में आई ग्वाल तेरे सारे। पाक के मन भर नटवर नंद किशोर, अरे नटवर नंद किशोरी लाला, नटवर नंद किशोरी, हाँ नटवर नंद किशोर लाला, नटवर नंद किशोर, आगे के नटवर जान में आए हैं, नटवर नंद किशोर, आगे के नटवर जान में आए हैं, नटवर नंद किशोर।
हरि बोल।
घेर लिए है सब गली रंगीली,
घेर लई सब गली रंगीली,
छाई रही है छवि छटा जबीली,
छाई रही छवि छटा छबीली,
डफडोली 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 मृदंग बजाए हैं, बंशी की बुनी घोर भाग के नटवर सान में आए हैं, नटवर नंद किशोर।
सारी गोपियां कृष्ण से होली खेलने आई है, नंदगांव और बरसाने की होली।
जुर्मिल के सब सखियां आई,
जुर्मिल के सब सखियां आई,
अरे उमड़ घटा अंबर पे छाई,
उमड़ घटा अंबर पर छाई,
जानी अबीर ओ जानी अबीर ओ जानी अबीर गुलाल उड़ाए हैं, मारत बर बर जोर भाग के नटवर सान में आए हैं, नटवर नंद किशोर।
सारी सखियों ने मिलकर भगवान कृष्ण को घेर लिया, पकड़ लिया। आज चलो, यहाँ नंद के लाला है, मजा चखाएंगी। राधे।
सखियन ने पकड़ो है गिरधारी,
सखियन ने पकड़ो गिरधारी,
नरती शाम बनाए दियो नारी,
नरते शाम बनाए दियो नारी,
जखू गड्डी लेहेगा, जखू गड्डी लेहेगा, जखू गड्डी लेहेगा में
भगवान को नारी बना दिया, साड़ी पहना दी।
नटवर सान में आए हैं, नटवर नंद किशोर।
प्रेम के कारण भगवान नृत्य कर रहे हैं सखियों के साथ, साड़ी पहन ली, लहंगा पहना दिया। राधे। तैयार है?
ले रे चोट ग्वाल ढालन में,
ले रे चोट ग्वाल ढालन में,
अरे केसर कीच मलर गालन में,
केसर कीच मले गालन में,
जाने हरियल जाने हरियल ओ जाने हरियल बांस मंगाए,
लठ मार होली
चल लगे चहुर भाग
के नटवर सान में आए हैं, नटवर नंद किशोर।
आए जयो शाम बरसान धाम तोए, होरी खूब खिलाई दूंगी, लट्ठन को मजा चखाए दूंगी। आए जयो शाम बरसान धाम तोए, होरी खूब खिलाई दूंगी, लट्ठन को मजा चखाए दूंगी। भक्ति की पराकाष्ठा हो गई। ब्रजवासी बैकुंठ से लोग जब भगवान के स्पर्श के लिए आने लगे तो भगवान सोचने लगे कि जब ये मेरे नज़दीक आए हैं तो क्यों ना इन्हें मैं आने का मौका दूं। प्रभु ब्रज भूमि पर विराजमान हो गए राधा रानी के साथ, और जितने भी ब्रजवासी थे
वो आए और अपने हाथों में पुष्प लेकर भगवान का श्रृंगार करने लगे। यदि आपको लगता है कि आपका मन है भगवान का श्रृंगार करने का, तो जो कपल्स हैं वो आइएगा, जो अकेले हैं वो ठाकुर जी का साथ लेकर जरूर आइए। स्टेज पर एक एक अम्बर कुमार जी सर, सबको आप लीड करेंगे, एक एक करके सभी पुष्प हाथ में लेकर भगवान को अर्पण करेंगे। आइए सर, प्रयागराज वृंदावन बनने वाला है। आइए सर प्लीज।
अरे फूलों में सज रही है कि फूलों में
मंजू मैम
सज रही
आइए, मधु मैम आइए।
श्री वृंदावन बिहारी फूलों में सज रही है, वृंदावन बिहारी फूलों में सज रहे हैं, श्री वृंदावन बिहारी
फूलों में सज रहे हैं श्री वृंदावन बिहारी,
और साथ सज रही है, हाँ और साथ सज रही।
आइए सर प्लीज आइए सर।
वृषभान की दुलारी फूलों में सज रहे हैं, श्री वृंदावन बिहारी फूलों में सज रहे हैं।
तेरा सब कुट सर पे रखा है किस अदा से,
और करुणा बरस रही है, करुणा भरी निगाह से,
करुणा बरस रही है, करुणा भरी निगाह से।
बेमूल बिक गए हैं, हाँ बेमूल बिक गए हैं, जब से छवि निहारी फूलों में सज रहे हैं, श्री वृंदावन बिहारी फूलों में सज रहे हैं।
आइए मैम आइए सर, पीछे से जो हमारी बहन है, यूथ है, आइए ठाकुर जी को प्रेम के रंग में रंग दीजिए। आइए
श्रृंगार तेरा प्यारे, शोभा कहूँ क्या इसकी,
श्रृंगार तेरा प्यारे, शोभा कहूँ क्या इसकी।
अरे श्रृंगार तेरा प्यारे, शोभा कहूँ क्या इसकी,
श्रृंगार तेरा प्यारे, शोभा कहूँ क्या इसकी।
अरे उन पर गुलाबी पटका, उन पर गुलाबी साड़ी,
उन पर गुलाबी पटका, उन पर गुलाबी साड़ी।
बेमूल बिक गई है, हाँ
बेमूल बिक गई
है, जब से छवि निहारी फूलों में सज रहे हैं, श्री वृंदावन बिहारी फूलों में सज रहे हैं।
अब एक छोटा सा रिक्वेस्ट है, ये वो समय आ गया है जब बिल्कुल भी आप उस ऊंचाइयों पर जाने वाले हैं जब प्रेम आपके हृदय में बस जाएगा। खड़े होकर 2 मिनट आगे आ जाइए और हमारी जो सीनियर्स हैं सर आगे आइए। जो आपने फूल भगवान को सजाया है उससे आपको आंगन को सजाने वाले हैं। यहाँ आइए, स्वयं कृष्ण आपको अपने रंग में रंगने वाले हैं। आइए
आप सभी जो बैठे हुए हैं वो खड़े हो जाइए, क्योंकि जो ठाकुर जी के जो रंग हैं वो आप सब पर भी बिखरे।
जिस जिसको प्रेम के रंग में रंगना है आगे आ जाइए जल्दी से।
अरे रंग बरसे रे गुलाल बरसे, अरे रंग बरसे रे गुलाल बरसे, राधा रानी हमारे रंग बरसा है,
राधा रानी हमारी पे रंग बरसे,
आगे आ जाइए आगे।
हमारी पे रंग बरसा है,
राधा रानी हमारी पे रंग बरसे।
अरे होरी में तबाही मच जाएगी,
अरे होरी में तबाही मच जाएगी,
तबाही मच जाएगी, तबाही मच जाएगी,
तबाही मच जाएगी, तबाही मच जाएगी,
ओ तबाही मच जाएगी, तबाही मच जाएगी,
तबाही मच जाएगी, तबाही मच जाएगी।
अरे होरी में तबाही मच जाएगी,
होरी में तबाही मच जाएगी,
अरे हुरिया में उड़े रे गुलाल तो कयोरी मंगी तरने,
हुरिया में उड़े रे गुलाल कयोरी मंगी तरने,
अरे मारी रे मंगी तर काजल वाले तो मारी रे मंगी तर, मारी रे मंगी तर काजल वाली भैया पूछो बारो रे नबा तो कयोरी मंगी तरने, हुरिया में उड़े रे गुलाल कयोरी मंगी तरने, अरे मारी रे मंगी तर नकरा वाले तो मारी रे मंगी तर, मारी रे मंगी तर नकरा वाली लाला पीछे भागे रे नवाब तो कयोरी मंगी तरने,
होलिया
में उड़े
ओ कन्हैया
तो कयोरी मंगी तरने, रंग बरसे ओ रंग बरसे ओ रंग बरसे भीगे शमा प्यारी,
रंग बरसे भीगे शमा प्यारी, रंग बरसे
ओ रंग बरसे भीगे शमा प्यारी, रंग बरसे
ओ रंग बरसे भीगे शमा प्यारी, रंग बरसे
ओ रंग बरसे भीगे राधा रानी, रंग बरसे
ओ रंग बरसे भीगे शमा प्यारी, रंग बरसे। होली है, होली है, अरे नाच लो भैया,
लौंगा इलायची का, लौंगा इलायची का बीड़ा लगाया,
लौंगा इलायची का,
लौंगा इलायची का बीड़ा लगाया, अरे चबे गोरी का, चबे गोरी का, चबे गोरी का यार बलम बरसे रंग बरसे,
ओ रंग बरसे भीगे चुनर वाली, रंग बरसे,
अरे चलत मुसाफिर मोहलिया रे पिंजरे वाली मुनिया,
चलत मुसाफिर मोहलिया रे पिंजरे वाली मुनिया,
अरे उड़ उड़ बैठी हलवैया तू कनिया,
उड़ उड़ बैठी,
उड़ उड़ बैठी हलवैया तू कनिया, बर्फी के सब रस ले लिया रे पिंजरे वाली मुनिया,
चलत मुसाफिर मोहलिया रे पिंजरे वाली मुनिया,
अरे मस्ती भरी रात है, राधा रानी मेरी जात है, कि जीने का ले लो मजा, डरने की क्या बात है, ना 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 नारी नारी नारी, ना 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 नारी नारी, ना 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 नारी नारी नारी, ना 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 नारी नारी ना, राधा संग रहोगे तो ऐश करोगे, जिंदगी के सारे मजे ऐश करोगे,
रंग सहोगे तो ऐश करोगे, जिंदगी के सारे मजे ऐश करोगे, शमनल
ओ शमनल ओ रमनल, ओ रंग लेके खेलते गुलाल लेके खेलते, रंग लेके खेलते गुलाल लेके खेलते, राधा संग होली नंद जाते, बोलो होली आई, बोलो होली आई, मदन गोपाल के, बोलो
होली आई, होली आई, होली
मस्ती लाई मस्ती लाई
मस्ती आई, होली आई, होली आई, होली आई, मस्ती लाई, मस्ती लाई, मस्ती लाई,
वृंदावन में डोलोगे तो राधे राधे बोलोगे, रोक न पाओगे खुद को तुम गाना के ही बोलोगे, वृंदावन में डोलोगे तो राधे राधे बोलोगे, रोक न पाओगे खुद को तुम गाना के ही बोलोगे, राधे 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 राधे
राधे राधे राधे राधे राधे श्याम से राधे
श्री राधे
आना यहाँ सोच समझ के शाम की लत लग जाएगी, नाचोगे तुम गली गली में लोग लाज बढ़ जाएंगे। आना यहाँ सोच समझ के शाम की लत लग जाएगी, नाचोगे तुम गली गली में लोग लाज बढ़ जाएगी। हाथों का पागल खाना या बिना पिए ही झूमोगे, गले में कंठी माला डाले राधे राधे बोलोगे। हाथों का पागल खाना या बिना पिए ही झूमोगे, गले में कंठी माला डाले राधे राधे बोलोगे। राधे 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 राधे
राधे राधे राधे राधे राधे श्याम से मिला दे।
सावन में लग गई है कि दिल मेरा।
राधे 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 राधे
ओ सुनो हसीना पागल दीवानी,
सुनो हसीना पागल दीवानी।
आज न आज न आज न आज न आज न सोया सारी रात कि दिल मेरा। ओ करुणामयी कृपामयी मेरी दयामयी राधे।
करुणामयी कृपामयी मेरी दयामयी राधे।
करुणामयी
करुणामयी
कृपामयी
कृपामयी
ओ मेरी दयामयी शाबा। करुणामयी कृपामयी मेरी दयामयी राधे। करुणामयी
कृपामयी मेरी दयामयी राधे।
ओ मेरी दयामयी शामा,
मेरी दयामयी श्यामा,
मेरी दयामयी राधे।
मेरी दयामयी श्यामा राधे।
बोल मांगे बिहारी लाल की।
मेरी दयामयी मेरी राधे।
बोलना धरारी सरकार की।
जय हो।
बोल मांगे बिहारी लाल की।
जय हो।
बोल बोल जोड़ी सरकार की।
जय हो।
ठाकुर जी थोड़ा सामने आ जाइए सभी लोग। जिनको ऐसा लगा है कि आप यहाँ नहीं हैं, कहीं दूसरी दुनिया में, वो आगे आ जाइए 2 मिनट के लिए। सभी आगे आ जाए। आइए। ये प्रयागराज वृंदावन बन गया, वो आनंद बन गया जिस आनंद को आप सालों तक याद रखेंगे। एक बार मैं चाहूँगा जयकारा एक ऐसी आवाज़ से जो आपके दिल से निकले भगवान कृष्ण के लिए। प्रेम से बोलिए बोल बाके बिहारी लाल की जय जय श्री। मैं यहाँ जितने भी हमारी नई जनरेशन है, यूथ है, हमारे सीनियर्स हैं, हमारी मातृशक्तियाँ हैं, कान्हा एकेडमी के डायरेक्टर श्री डॉक्टर अनूप शर्मा साहब की तरफ से 2 लाइन आपके लिए कहना चाहता हूँ, ज़रा ध्यान से सुनिएगा। आज हम अपने जीवन को खो रहे हैं जात मजहब के नाम पर। देश और दुनिया में हम केवल इसी का नाम लेते हैं, लेकिन हमारे प्रेम में कहीं ना कहीं एक दीवार खड़ी हो रही है। 2 लाइन सुनिएगा। अब तो मजहब कोई ऐसा चलाया जाए जिसमें इंसान को इंसान बनाया जाए, और जिस फूल से महक जाए पड़ोसी का घर, ऐसा फूल हर घर में खिलाया जाए। प्रेम से बोलिए जय जय श्री। इन्हीं शुभकामनाओं के साथ कि ये 19 से लेकर 22 तक चलने वाला सेमिनार मीट्स आपको बहुत कुछ देकर जाएगा, जिससे आपके जीवन में एक नई ऊर्जा का संचार होगा। इन्हीं शुभकामनाओं के साथ फिर मिलेंगे, फिर मुलाकात होगी। हंसते रहिए, खिलखिलाते रहिए और राधे राधे कहते रहिए। प्रेम से बोलिए राधे राधे राधे। और मैं शिवम जी से कहूंगा एक बार फिर से आइए।
It was a truly energetic and blissful performance by the Bridge Folk team. I would now request Dr. Amba Kumar Rai and Dr. Amit Avasthi to kindly present a token of our heartfelt gratitude to the director of the team, Dr. Anup Sharma. Can we have a huge round of applause for the team?
और स्पेशल थैंक्स हमारे साउंड ऑपरेटर इंजीनियर साहब का, जिन्होंने इतना सुंदर आनंद दिया है। थैंक यू सर, यहाँ आपकी उंगलियों का कमाल है। थैंक यू, शुक्रिया कान्हा एकेडमी वृंदावन की तरफ से।
हम सभी ने ऐसा ही एहसास किया कि हम प्रयागराज में रहते हुए वृंदावन में आ गए हैं, तो आनंद बहुत आया। मतलब उसकी अनुभूति की कोई सीमा नहीं, और मैं शायद उसे बयां भी ना कर पाऊँ। बड़ी अच्छी बात है।
थैंक यू सर।
Can we have a huge round of applause? With that, we conclude an inspiring second day of IMMUNOCON 2025. Thank you for your energy, curiosity, and the passion you brought into every session. As the lights dim on today's discussion, we invite you to return tomorrow for another day of breakthrough ideas and unforgettable conversations. Before we all disperse, we warmly invite all student participants into the dining area. We also request all the faculty scientists and speakers to please gather near registration desk. Please enjoy the evening, relax, and connect with the fellow participants. Rest well. Day 3 awaits, and it promises even more brilliance. Thank you. I repeat, student participants are asked to please assemble in the dining area, and we request all the faculty, scientists, and speakers to please gather near registration desk. Thank you.